instruments purchased at Ingaba is well taken care of. A fleet of vehicles commissioned for local trips and deliveries assures we arrive at your lab on time. In the past 18 years, we have started subsidiaries in East and West Africa, and we have offices in Kenya, Nigeria, Ghana, Tanzania, Senegal, and still maintain a strong presence in our neighboring countries. All this is achieved with a highly trained and professional team that is always ready to assist. Welcome to Ingaba Biotech.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, was established in 2014 as one of the World Bank's Africa Centers of Excellence. WACCBIP is located on the campus of the University of Ghana, Legon.
WACCBIP was established as one of the World Bank African Centers of Excellence. And the inspiration behind it is basically that a number of us who were at the Department of Biochemistry and the Noguchi Memorial Institute for Medical Research, we had trained outside, and we did PhDs in various science disciplines outside the country. When we got back and realized that there was not much by way of facilities to train scientists, we saw this as an opportunity to create an environment which would enable many young scientists to get the opportunity to get the quality of training that we got overseas.
This world-class research center started off as an infectious disease center, but along the line, it's added non-communicable diseases to its focus areas.
WACCBIP is providing something to local and regional students that we probably don't have in many places within the country or within the region. Facilities, financial resources, as well as an environment that encourages student learning and research. And that has significantly increased students' output in the Department of Biochemistry, Cell and Molecular Biology.
WACCBIP has a whole range of collaborators at the institutional, national, regional, and international levels. These collaborations include the Noguchi Memorial Institute for Medical Research, Ghana, the Department of Biomedical Sciences at the University of Ghana, among other departments, the Ghana Health Service, and the Ministry of Health. The center also has collaborations with institutions within and outside Africa.
Currently, there are about 6 labs in the center, and these labs do research based on the faculty who is in charge of these labs. We have a malaria group that has 3 different units. There's an immunology unit, there's a vaccine development unit, and also a transmission unit in there. We also have a molecular biology lab that has 2 faculty in charge there. One does work with African animal trypanosomes, and the other one, who is myself, works in Buruli ulcer and antimicrobial resistance. We have a huge virology group. They do research, anything that has to do with viruses. So they do HIV research. They even look at some bat viruses and gastroenteric viruses. More recently, too, we've added a COVID lab, and this lab does routine testing for COVID positivity and negativity. And we also look for seroprevalence across the population. So people who have been prior exposed to COVID, but maybe have been asymptomatic, so they don't know whether they had COVID or not. And we actually just recently published a paper on our findings.
The virology laboratory of WACCBIP was set up right from the beginning with one main lab, and they were doing things like rotaviral, researching into rotaviral infections in Ghana. The virology lab at WACCBIP was also involved in the first identification of a dengue case in Ghana, and also doing HIV research, as well as research on viruses which might which are in bats, but which might be have a risk of causing disease in humans. So these are filoviruses, the same type of viruses that cause Ebola. So that's work that was being done.
The center currently has 6 research laboratories, namely the Cell Biology and Immunology Lab, commonly known as the Malaria Lab, the Molecular Biology Lab, the Virology Lab, the Medicinal and Natural Products Lab, the Next Generation Sequencing Lab, and the Forensic Chemistry Lab. At WACCBIP, the scientific tools and equipment are always and readily available to students, regardless of the courses being studied at the center.
Really, the driving force was to try to create a world-class research and training environment where young African scientists from across the continent can come here and develop their talent and do high-quality research that can be comparable to research that's done in any part of the world. So basically, that was the vision that we were pursuing.
Throughout my academic journey, I'd always looked for opportunities to come back home. My plan has always been to establish myself, to spend my most productive, my most useful years in Africa and hopefully in Ghana. So even though I studied abroad altogether for 13 years, followed by 6 years as a postdoc also abroad. So altogether, I was away from Ghana for about 18 years. I was always looking for opportunities to return. During my second postdoc, I actually met Professor Wandare, the director of WACCBIP, in London when he came to actually receive an award. That was the first time I heard about WACCBIP. And so hearing about WACCBIP, hearing what Professor Wandare was trying to do in Ghana, I felt that it was a place that would welcome someone like me, a place that someone like me potentially could thrive. So I began to look for opportunities to return, and I was able to get funding to support my return to Ghana, and so applied for a position and was appointed as a research fellow. It was basically a fulfillment of something I'd always wanted to do, and WACCBIP stood out as a place that, even though it was a young center, was led by somebody who himself was very dynamic, who identified a lot with the ideals that I myself identify with. And so was somebody that I thought I could work for, somebody who could mentor me.
WACCBIP has evolved to become a world-class research center, thriving on initial funding from the World Bank and subsequent support from the Wellcome Trust and the Africa Academy.
We were very fortunate to get the funding through World Bank to establish the Center of Excellence here. And part of that funding gave us a huge chunk of money to buy core equipment that could be used and shared by all faculty in the department and opened up to faculty within the university and within the sub-region. We have a core facility for flow cytometry. We also have a core facility for sequencing of DNA. Just recently, we've even upgraded it to include a NextSeq Illumina sequencer, which is probably one of 2 in the entire Africa. And that has the capacity to sequence both DNA and RNA, which is world-class. We have a confocal microscope core facility, which can be used to take images of cells and for us to understand actual cell biology as it happens in real time. We also have the protein expression lab that I talked about, and we have state-of-the-art equipment in these laboratories that help us, again, make proteins, express proteins, and take it to the next level of trying to identify what these proteins actually do within cells.
Since I've been in the virology department, I've been doing mostly HIV research, that's my specialty, looking at how the different genotypes we have in Ghana affect the ability of treatment and the progression of the disease. And of course, since the pandemic, we have been right at the forefront of doing research of SARS-CoV-2, that's the virus that causes COVID-19 in Ghana. The HIV research that we've done here at Virology, prior to my coming, there's been some findings that in the Ghanaian population, some of the genes that may be involved in progression are deleted in some of the population. Actually, a few of my colleagues did that work. And since I've been here, we've been looking at the new drug which has been introduced in Ghana, dolutegravir, and our data, which is yet to be published, is showing that it is one of the best drugs to be used, and we are also selecting some mutations for this drug.
My experience in WACCBIP here has been very great. I came here in 2014. So after my undergraduate, I joined the lab in 2015 and I served as a research assistant. When I entered, I didn't know anything about research, but I've got opportunity through mentorship and training. I've had great experience here. Sure, I think there's fairness in the enrollment process. For what I know, I was fortunate after my first year master's, I was enrolled into the PhD program. Then what I have seen so far, really, before you get admission to the program, you write in exams and they try to look at gender balance and also the international and the local students. So there's fairness in the selection process. So my PhD thesis or project seeks to develop novel diagnosis for detecting Plasmodium species. So Plasmodium species are parasite that causes malaria, and currently we have RDTs and microscopy at the point of care for detecting this species. But unfortunately, these tools are not sensitive or specific enough for detecting this species. So there are more reliable tools, which are the PCR or molecular-based diagnostic tools. And if I can give you an example, that's what we are using to detect COVID now. The unfortunate thing is these tools are not readily available in terms of the reagent. So my project seeks to develop these reagents here in Ghana so that we can make them readily available and also cost-effective for detecting infectious diseases.
The center runs 3 programs at the master's level, which includes Master of Philosophy in Molecular Cell Biology of Cell and Infectious Diseases, Master of Philosophy in Biochemistry, and Master of Philosophy in Molecular Biology. The PhD level includes PhD in Biochemistry and PhD in Molecular and Cell Biology of Infectious Diseases. The center also engages the community in its activities from time to time.
What was of common interest to all of us was that we wanted to create a world-class research and training environment to do basic science. Now, this was led by me, but with support from, several colleagues here at the Department of Biochemistry and also at the Noguchi Memorial Institute for Medical Research. In terms of the resources we need sustainable funding, predictable funding. If you look at all the countries that have developed, they have predictable funding for research and for science. If you look at the USA, they have something called the NIH, which they pump billions and billions of dollars in every year as part of their national budget. It's locked in. Ghana, we still don't have that. We've been talking about a research and innovation bill for a long time. Still not been enacted. We need to have such things so that we as scientists, there's a fund that we can apply to access funds to do research and to innovate on a sustainable basis. As we say, we compete for funds from overseas, and that is not predictable nor sustainable because a lot of the funders, they keep asking us, how much is your government contributing? Okay, we've given you 8 million, we've given you 6 million, we've given you 5 million. How much is your government contributing?
Everybody knows that Africa has problems. Our problems are all over the news. We get offended when people point out only the problem. But it's also very clear that the only people who will solve Africa's problems are Africans. We are waiting for malaria vaccines to be developed in Europe and North America for us to use. We are waiting for people, for President Jimmy Carter to come and eradicate guinea worm in Ghana. If Africa and in Ghana is going to achieve what I believe and we all believe we can achieve, we have to take responsibility for our problems and come up with locally relevant solutions to solve those problems. For me, WACCBIP represents a place that is trying to do its bit to contribute towards it. We are trying to train the next generation of scientists, not to come and work at WACCBIP. We are training the next generation of scientists to think of ways to solve problems they see in their own community.
International students are helped through WACCBIP to obtain accommodation. And basically what we do is that we liaise with the hostels and the halls on campus and arrange some of the rooms for them before they actually come in. At WACCBIP, we provide at least two forms of scholarship. One from the ACE World Bank grant and the other from the Delta's program.
Because we are in the tropics, a lot of patients report to the hospitals with high temperature. This is usually attributed to malaria, but we found out that malaria answers, let's say, 40% of those cases. For the other 60%, they are presumptively treated as malaria. And in most cases, the patients keep going back because it's not malaria. So what else is causing it? It's not clearly known. And the diagnostic systems in the hospitals are not advanced enough to take care of a variety of disease conditions. So this project was undertaken by one Nicholas, who looked at a whole panel of pathogens. So this was a discovery project. Okay, so once a child reports with fever and it's not attributed to anything else but the infection, and they check for malaria and malaria is not there, they then take those samples and look for all manner of pathogens. Okay, and they were able to discover a whole variety of viruses and bacterial pathogens that explain those other febrile illnesses. So going forward, that would, instruct a new way of attending to patients in that category, so that we can. What we haven't done is to develop diagnostics that can be used in the hospitals, but that is the next phase that we can look at. The second is diagnostic technologies that have been developed by two PhD students, Francis Krampa and Felix Ansa. So here they are looking for electronic ways of detecting malaria. Okay, so what we have in the system right now is glucometer. You put a drop of blood and it tells you the level of glucose in your blood. We are trying to do the same for malaria so that people can even test at home, right, and not have to go to the hospital when this becomes available on the market. The third one is a project that concerns a new way of developing antibiotics. And what we are trying to do is to target what we call the genome stability of pathogens. This has not been done before, and we are pioneering that from this center for the first time. And we're hoping that over the years we will make progress that will lead to the next generation of antibiotics.
The first output that we had from our sequencing was a manuscript which was published, and that was the first 46 sequences of SARS-CoV-2 in Ghana. We published that last year, and since then we've done almost 1,000 sequences. So we are preparing that also to publish it. And why that is important is we are tracking basically the changes in the virus locally, because there's, if you look at literature or the news, you see how it's changing globally, but you have to know the local outbreak, you have to know the local epidemic. And what we see in Ghana is that sometimes the variants which are increasing are not necessarily what is increasing in the rest of the world. For now, for instance, we have Delta, which is also on the rise in Ghana. But there's another variant which is nobody's talking about, but it's rising almost faster than Delta in Ghana. So that's the sort of things that we do. And when we get the results, we write what we call policy briefs, advising the government that this is what we found. Earlier on in this year, when we did our sequencing of airport samples, and we first found the alpha variant, that's the UK variant. And then we found it in local circulation, and we wrote it, we communicated to the government, and that was what led to the reinstatement of some of the restrictions, like restricting the funeral size, party size, and things like that. That was as a direct result of the work that we're doing.
The key message that we want to put out there is that Africa has a lot of talent. If you look across the world, there are African scientists doing great things all over the place. Some of these COVID vaccines that we are seeing, African scientists played key roles in developing them so quickly in different countries. If we create the right environment for them, a lot of these people will come back home. We have already brought 7 scientists, African scientists who were in the diaspora, we brought them here. And the reason why they were able to come back, or they were convinced to come back, is because they came and looked around, like you guys have looked around, and they saw that they can do good science here, and that this environment looks like where they've been working in the UK or in the US, and they are convinced that this is world class enough. If we create that, we can start to bring all our talent back, and Africa can start to develop our own vaccines, can develop our own drugs, we can be more innovative and build new technologies that we can even export to other parts of the world, and not sit as we are now waiting for people to develop vaccines, use for their people. When they have had enough, then they share with us. That's where we are now. Okay, that situation has to change. That means we have to take matters into our own hands. That means the African governments have to start to put some money into science on a consistent basis. Let's have a fund, such as we have National Health Insurance Fund, we have GET Fund, we have all these funds that a percentage of the taxes goes directly into that fund every year. So that fund is available for scientists to access. Let's do that. Let's put some money aside for science, because this type of investment have to be done for us to develop. Otherwise, we keep marking time, we keep just treading water. Because if you keep relying on people for your innovations, that means you are not making progress. You know, I keep telling people that if you think about investment in science, think about it like the way we invested in petroleum exploration. You know, ever since I was a child, I was hearing.
Good morning. I've been waiting for the room to fill up, and it seems people have a hangover or something. So we're going to get started, and hopefully people will stagger their way here. All right, so this morning we're having the third and final keynote talk, and once again it's one of our own. Today we are going to host Dr. Bismarck Dinko. So Bismarck is a member of the WACCBIP family, and he has done a fantastic job of building a research group from scratch, you know, at the University of Health and Allied Sciences in Ho, UHAS. Those of us who have been through these types of efforts, we appreciate the achievements that he has gathered over the last few years. You know, we had the opportunity to do that in the University of Ghana, but he did it in a new university, which basically had nothing. And he has managed to stand up a research group that is now vibrant. He has managed to win several highly competitive and prestigious grants, and he is now well positioned to join the top echelons of African scientists. Bismarck started his education at the other university in Ghana. You know, the other university that is second to us. And he did biology over there in Kumasi. And after that, he went to the London School of Hygiene and Tropical Medicine, where he did his PhD in infectious and tropical diseases. After that, he returned home. He was bold enough to come back, and he came back, and he has, initially he came back through the Gates postdoc fellowship at Noguchi. And he did that for a short while, and then he moved to UHAS to start his group. Since he has been at UHAS, he has won several prestigious fellowships, including what we call FLAIR, which is Future Leaders African Independent Research Fellow, which is a collaboration between the Royal Society UK and the African Academy of Sciences. This is one of the most competitive fellowships on the continent. He also won a Wellcome Trust Training Fellowship, which again is a highly competitive grant scheme. Before then, he was a fellow in the Fogarty Global Health Research Fellowship, in this case with a partnership with Keck's group in Cornell, as part of these Fogarty Capacity Development Fellowships. At UHAS, he was the founding head of the Department of Biomedical Sciences, and he basically established the department and stood it up and got accreditation for their courses before he moved on. He started as a lecturer and then was promoted to senior lecturer in that same department. So, ladies and gentlemen, I am proud and honored to welcome my friend Bismarck Dinko.
Thank you very much, Professor Wandari, for the kind introduction, and also thank you to the entire WACCBIP for inviting me to give the third keynote and one of the keynotes for this year. Myself and my research group in Ho and the entire UHAS community is very grateful for this opportunity. So today I will talk to you about what we know about gametocytes of Plasmodium falciparum with respect to the biology and the immunology, particularly antibody immune responses, and how that information can be useful in the development of transmission reducing interventions. So in the first part of the talk, I will give you a little background about gametocyte biology and also antibody immune responses that we already know. And then I will go ahead to talk about gene expression in gametocytes of clinical isolates, then antibody responses in a cohort that we looked at for using gametocyte-specific antigens. And then I will move on to another important part, which is looking for gametocytes in the bone marrow of people who are still living, not autopsy cases. And then the last part will look at cellular immunology, looking at immune cells. Since we have the opportunity to work on bone marrow, then we'll look at immune cells that are present in the bone marrow and how the accumulation of parasites in the bone marrow impact on the ability of individuals to mount immune responses. So just a brief introduction, just to say that in the malaria infection, we have asexual cycle and then the sexual cycle. So after every round of asexual replication, there's a small portion of parasites that will diverge to form the sexual stages. And these will go through up to 10 to 12 days of development to give you these developmental stages of gametocytes. What I need you to keep in mind is that these stages 1 to 4, are not seen in peripheral circulation. They are known to be sequestered in the bone marrow or the spleen. So when we do diagnosis for malaria and there are gametocytes, these are the ones that we see. Then we know now that there are differences in the distribution of the different stages of gametocytes in the bone marrow, but most of this data is coming from autopsy cases, people who have already died from malaria, and then we look in their bone marrow. But more recently, we also have data to confirm that this is also the case when we look in the spleen. So we can see gametocytes as well as asexual stages in the spleen as well. And it's even thought that there's a de novo generation of gametocytes. So commitment happens here to give rise to development in the different stages. And then, of course, this study also showed that there are more parasites that you see in the spleen compared to what we see in peripheral circulation. So with respect to antibody responses, we know that there are specific immune responses that are raised against the mature stages based on the previous study, and then these antibody responses are very distinct from asexual stages. And then I took this study further to show that you can actually detect these antibody responses developmentally as part of my PhD work. And very recent data has also shown that it's not only the mature stages that have antibody reactivity, but also the immature stages also elicit antibody responses. But despite all this knowledge we have already about antibody responses against gametocytes, we still don't know specifically what the gametocyte antigens are. But we think that there could be members of the gene families that are known to be expressing gametocytes, or there could also be some novel antigens. And one of the gene families that we are interested in looking at is TVOS, because we know that TVOS has been shown to be involved in the deformability, the changes in the shape of the gametocyte stages from the earlier stages to the mature stages. It's also been shown to be present in the gametocyte-infected erythrocyte surface. But there are several TVOS, so the question we had at the beginning is which specific TVOS is the one that is directly involved. So our general rationale is the fact that we don't know the mechanisms that are responsible. How do gametocyte stages arrive in the bone marrow, and how do they come out? How do they come out back into circulation? And then we don't also know the identity of the antigens that are responsible for gametocyte immune responses, regardless of whether the mature stages or the immature stages. So our strategy or the aim was to analyze in clinical isolates if we could generate gametocyte from clinical isolates, several clinical isolates, and look for common genes commonly expressed genes that are found in these clinical isolates. Could that give us an indication of whether these are the ones that are responsible? And the focus was to look in immature stages so that we can be able to look to find if these genes, if we are able to identify some genes, then could these be the ones that are important for sequestration and possibly also for immunity? And the way we did that was to obtain clinical isolates and then perform this analysis. And this strategy was also important because in most laboratories that work on malaria, mostly they work with a lab line. So it will give us an opportunity to compare the transcriptome that we get from clinical isolates, from clinical isolates against the laboratory lines to see whether the observations are comparable. So just to give you the first aspect, looking at the global transcriptome, like all the transcripts that we obtain in immature gametocyte. So the first aspect, what we did was to grow gametocytes from 3 different isolates, clinical isolates, And then alongside, we grow a laboratory line, in this case, NF54. And then we induce the gametocytes, we grow the clinical isolates and induce gametocytes. And when we have a mixture of mid stages, then these are harvested, and then we subject these to sequencing. And then we can take through the analysis at the end to look at differentially expressed genes, and then also the gene clustering. So for the quality control, you can see the different samples that we obtained here, and they are arranged or clustered on the basis of the strain of the parasite as well as the stage of the parasite. This is what is used in grouping them. But what is important here is if you look at the principal component analysis, you will see that gametocytes will cluster around the same area, but you see also there are some asexual gametocyte groups that cluster in the area where we have asexual stages. So at this point, we're thinking whether this could possibly be asexual contamination of our gametocytes. So we term this the gametocyte group 2 or outliers, and I will talk to you in more detail about these outliers with respect to their gene expression. So first, we did 2 sets of analysis or comparisons just to see what we have. So we have clinical isolates and we have lab lines, we have asexual parasites, and we have gametocytes. So we compared gametocytes and asexual parasites for both the clinical isolates, as you can see here, and then also we compared for the lab line sexual stages or gametocyte with asexual stages. And for both comparisons, we realized that there was, we had in the first 100 genes, there are gametocytes that are specific, genes that are specific for gametocytes were differentially expressed in the first 100 genes. And this was the same for the lab line comparison as well as the clinical isolate comparison. And this tells us that there are similar biological processes that are commonly enriched in the clinical isolates as well as the NF54. And this is just a diagram just to show the different the numbers of the genes when we did the comparison. But because we are interested in gametocytes, we focus more on the gametocytes to see what's happening in the clinical isolates versus the laboratory line. And here we can show that with respect, the gametocyte-specific genes are highly expressed when we look in the clinical isolate compared to when we look in the laboratory line. And this might suggest that in clinical isolate, gametocyte production is a much more active process compared to the laboratory strain. So there's a higher expression of gametocyte-specific genes in clinical isolates. So we did also a k-mean clustering and pathway analysis, some of which are already showed. And then we had 4 clusters. We have cluster A and then B. And what we realized is that the genes that were present in cluster B were upregulated in clinical isolates, and these were not the case for cluster A. Then when we looked also for cluster C and D genes, they were largely asexual, highly expressed in asexual stages and not gametocytes. So coming back to the outliers, the gametocyte samples that clustered in the region where we expected to have clusters of asexual parasites, we did 2 basic analyses just to tell if they are indeed gametocytes or is a contamination of asexual parasites. So the first analysis is to compare these gametocyte group 2 with their asexual parasite progenitors. And when we did that, we saw that we made the same observation as we made with the first comparison, which is that there's an upregulation of gametocyte-specific genes in the clinically derived, in the outliers that we have, compared to the asexuals, their asexual progenitor cells. Then next was to compare these gametocytes from the outliers or the outliers with the other asexual, clinical gametocytes to see what we have. And then we found out that they are actually gametocytes, just that they have a lower expression compared to the first group. And this is why they appeared closer in the region of the asexual parasites. So for this part, we know now that the core transcriptome is well conserved between short-term cultured clinical isolates and NF54 laboratory strain, regardless of whether you are looking at gametocytes or asexual parasites. However, there are some important differences, and one difference is that some genes are differentially expressed in clinical isolates and in laboratory lines, example, genes related to motility. We also found that some gametocytes exhibit some transcriptional diversity, particularly for the clinical isolates. And pathways that are associated with movement we found to be more enriched in clinical isolates, which suggests that if you were doing laboratory studies looking at physical structures of the parasite, you have a better resolution if you work with clinical isolates compared to laboratory lines. So we are now doing further analysis, looking at the multi-gene families, which is taking a long time to be able to find in these 3 clinical isolates and the lab line that we worked on, what are the commonly expressed genes. If you take STIVO, for instance, given that there are many of them, then whatever we find, we can apply these in infected individuals to identify potential targets. So while we were waiting to discover our novel antigens, we had gametocyte-specific antigens from Patrick Duffy at the NIH, particularly PFS 4845 and PFS 230, to analyze in a cohort of children that we had already recruited and sampled to be able to analyze antibody responses to gametocytes. So in this study, we worked in 3 hospitals. So we recruited 2 to 14-year-olds. They were positive for RDT and microscopy at the same time. And then we took a blood sample day 0 and day 7, and then we processed them as follows. So if we look at the patient characteristics, we were able to recruit over 2 years during the malaria season 427 children, and a little more than half of them were available 7 days later after treatment for a follow-up, and they gave an additional blood sample. We can see that the majority of them are between the ages of 6 to 10 years. And in the lab also, because our interest is on gametocytes, we developed a multiplex assay that has the ability to detect male and female gametocytes and also ring stages, so that in one single tube, you can find who has gametocytes, who has ring stages, and who has what proportion of these are male and female gametocytes. And this assay is quantitative and, of course, is RNA-based. So when we use this assay to screen these 200 out of the over 400 children, and then we could see that when we look at male and female gametocytes, there are more female gametocytes at day 0. And when we go to day 7, we even see more female gametocytes compared to male gametocytes. But if you put the 2 together, you still have slightly higher gametocyte at day 7 compared to day 0. So if we look in more detail, this is just what I showed on the table, but if we look in particular only focusing on just the gametocyte, you can see more clearly that if you put the male and female gametocyte prevalence together, you have more gametocytes at day 7, and most of them are female gametocytes compared to the male. And if you quantify these, you see it even much more clearer that you have a higher number of gametocytes that are coming up 7 days after treatment, and most of these are female. And we think that when you treat with an antimalarial, there's premature release of gametocytes, but also because these are symptomatic individuals, 7 days after treatment is enough time for immature gametocytes that were not killed by the drug to develop to give you mature stages, which we are detecting in this assay. So when we now look at the antibody responses using the gametocyte-specific antigens, we added 2 asexual stage antigens just for the purpose of comparison. You can see that we don't see any significant differences in terms of antibody levels when we looked at day 0 gametocyte positive and day 0 gametocyte negative compared to day 7 gametocyte positive. But when we look in, we see some difference with respect to AMA1, which was not really our focus. If you look again for only focusing on those that were gametocyte positive for the 2 time points, we don't still see something significant. If you do a more detailed comparison, we find the same scenario, and then we think that in these individuals are children, and also we don't tend to see a lot of gametocytes in terms of numbers and also prevalence in children because they are symptomatic. If we were to look in asymptomatic individuals, we will see much more stronger responses. So here we find that there's a significant increase in anti-AMA1 antibody level at day 7 in gametocyte positive children when we compare to the negative and positive day 0 counterparts. And we don't see any association in terms of antibody levels between antibody levels for these ones and gametocytemia. And there's no difference in antibody level between gametocyte positive and negative children at the same time point. So given that we know that much of what we know about gametocyte development biologically comes from autopsy cases and then also from in vitro culture, we designed a new study to be able to look and find gametocytes in individuals who are still alive. And this study was designed around orthopedic surgeries to see whether that will help us to understand gametocyte sequestration and development. So in this study, we worked in 4 different hospitals, 3 hospitals in the Volta region and 1 hospital in the northern part of the country. And I will tell you later on which part of the country you have more broken bones. So here, what we expect is that when there's an there are 2 major categories to get some bone marrow samples here. The main one is from accidents. So when there's an accident, it will come to the orthopedic clinic as an emergency, and then we can get consent, a verbal consent initially, and then a written one afterwards. And then the accident must involve an open bone that will allow easy access of bone marrow, and we can take this using a syringe. We don't need to use a needle. Another category is you can have children who are born with malformed bones, and then they have to be a surgery to repair the bone, or even as we grow older, the joints part of the joints of our bones can overgrow, and these have to be operated upon. So this is how we get bone marrow samples. And we get a bone marrow and then a biopsy at the same time. So the biopsy is basically the cancellous bone or the subpart of the bone. And that is meant for the imaging analysis, which we will do later on for localization of gametocytes or other parasites. And the bone marrow is particularly for our molecular analysis. So if we look at the patient characteristics, so in 2 years also, we were able to get up to 227 bone marrow donations, and at the same time, we collected PET peripheral blood so that we can compare what we are seeing in the bone marrow with peripheral circulation. So we used the first assay that we developed detecting asexual stages, male, female gametocytes, and to be able to determine the prevalence of gametocytes in all of these. We developed also a second assay. Because we are interested in gametocyte, we know that there are different stages, up to 5. We developed an assay that could detect early gametocyte, mid-stage gametocyte, and then late-stage gametocyte, and this assay also was applied. So if we look at this table, we find early gametocyte, intermediate, and late. So early gametocytes, we saw more in bone marrow compared to peripheral blood, as you can see here, 10.6% and then 9.2%. And intermediate gametocytes were largely found in the bone marrow compared to peripheral blood. And then late gametocytes were more in peripheral blood compared to bone marrow. And we could actually look at these with respect to the age, the gender, and the HB level of these individuals to see the distribution. So if we look in more detail, also first, just to show which part of the various, which type of bones we got our bone marrow from, we can see that most of the samples came from the femur, and then some from followed by the tibia and all the others. And we could analyze the site of bone marrow collection and the numbers or the prevalence of gametocytes, which, so for male and female, and then also the at the early, mid, and late stage gametocyte. And of course, if we are getting most donations from the femur, you see that the femur in most cases is where you have more parasites. So just to show in more detail what I showed on the table, so we see that in bone marrow, you have more female gametocytes, and then compared to, in bone marrow, you have more gametocytes if you put the male and female together, compared to peripheral blood. And then, of course, most of these gametocytes that you see here are female. And then if you do quantitatively, the representation is the same, it doesn't change. And then here we are just showing if you have asexual parasites. So a good number of them had parasites by PCR. So we looked further to see if we look for early, mid, mature, and if you put all of them together, we find there are more gametocytes in bone marrow compared to peripheral blood. And then, as already indicated, for mature stages, you have more gametocytes in peripheral blood compared to bone marrow. It is the reverse for the intermediate or the mid stages, but in early, you have about roughly about the same. And you can find the same representation for all gametocytes. If you quantify, the numbers are very similar, but if you look for mature, you have very few in the bone marrow, and a lot of them are in peripheral blood. And for intermediate, most of them we find, in terms of numbers or density, are in the bone marrow. So we're also interested to see these bone marrow donors, whether they had antibody responses to PFS48, PFS230, and the other asexual antigens. And we could see in the case of 48-45, there's a significant response between when we compare the negative versus the positive ones, negative for and positive for gametocytemia. So regardless of whether we are using the first assay looking at asexual male and female or the different developmental stages of gametocytes, the presence of gametocytes is somewhat similar in terms of numbers and prevalence in the bone marrow and the peripheral blood. However, we find female gametocyte prevalence and density to be relatively higher in the bone marrow. What I forgot to say is that in gametocytes of falciparum generally is female bias. And there was more distribution of female, the mid stages, late gametocytes in various bone types, as I showed, and then the intermediate gametocytes were more abundant in bone marrow, while mature gametocytes were abundant in peripheral blood. And we saw a strong PFS48-45 antibody response in gametocyte positive bone marrow samples. So now we know that from the bone marrow donations that we have, which ones have the particular type of gametocytes. The next step was actually to visualize them using an imaging technique. And this is very, very preliminary. We just show you now the optimization that we have done. So when we get the biopsy, it goes through a decalcification step to get rid of the bone, and then we can then, first it's in formalin just to kill whatever is there, and then it's decalcified, and then we can do paraffin embedding, and then we cut them into thin slices before we react with the antibody. And here we have shown that it is possible to detect gametocytes using rabbit anti-PFG27 and also using mouse anti-PFS16 antibody. So just to show that it's possible to do this also in bone marrow, here we don't show positive gametocytes yet because this was just preliminary, but we do show that it is possible to perform our procedure and be able to identify the different components of the bone marrow. So we can see adipose cells, we can see the bone, and then we can also see the bone marrow and then the nuclei of the cells within. So when we, as parasite biologists, when we work with bone marrow, we are interested in the impact of the bone marrow on the development of the parasites. But the opposite is also important. If you have parasites accumulating in your bone marrow, is it in any way affecting the function or the primary function of the bone marrow? So, so the way to look at this is we came up with a hypothesis which is based on the fact that depending on the malaria antigen, immune responses to some antigens are known to be very poor, and it could be due to a number of issues. One of them is there's dislocation of older plasma cells that are located. We know that plasma cells, most of them are located in the bone marrow, and then also the memory cells are in circulation. So it's either dislocation of these cells or there's central tolerance, because you have the parasite located in the region where the immune cells are generated. So are they beginning to make friends so that they don't attack each other later on in the future? That's what we wanted to know. So during our bone marrow study, we already had mononuclear cells from the bone marrow and then also cells from the periphery. So we stain these with antibodies that we use in the ELISA, and we can show here very briefly that the output that you get from the flow, you have your lymphocytes, and then you can determine all the different cells as shown here. And then we then do further analysis to look at the proportions that are B cells, memory cells, and then plasma cells. And we show here that you have an enrichment of malaria antigen-specific B cells in the bone marrow compared to peripheral blood, as you can see here. And this is the case for the malaria antigens, and it's not the case for tetanus. So just to summarize that, we are currently working on the localization of gametocytes in bone marrow, but we have shown that B cells that are specific to malaria antigens are enriched in the bone marrow compared to peripheral blood. So, and we tend to work a lot collaboratively, as I was introduced. If you are isolated in some corner, that is how you can do very good science. So we work with a lot of people. These are the orthopedic surgeons and the clinicians in obtaining our samples, and these are different scientists that we collaborate with. So we work closely with Britta on the B cell analysis, and with Gordon, we work on every aspect of our research. And Patrick Duffy is the one who donated the antigens for us. Oliver gave us the opportunity to do the sequencing at Sanga. The bioinformatic analysis was done by Jonas. Jonas was a PhD student under the WACCBIP program, and Hamza is the one who developed the assay. So, and for the imaging, we work very closely with Pietro Alano, who is based in Rome. And we are also grateful for all these institutions and the funders who have been supporting our work all these years. Thank you very much.
All right. Thank you, Ms. Mark. So we have just a few minutes for questions. So can take one or two questions, comments. Let's start with Ken.
So, Bismarck, I was puzzled by your comparison, your transcriptome comparison between your wild isolate gametocytes and your NF54. And you said that the wild isolates were enriched in motility-related genes. But gametocytes aren't motile, so what do you think that means?
So I think physically gametocytes may not move, but during fertilization, gametocytes have to invade, male gametocytes have to invade the female, and there must be some form of movement there.
So you think it's a storage of proteins for fertilization in the gut of the mosquito?
Exactly, that's what I think, yeah.
Yes, sir.
Hi, Bismarck. Just to follow on from that, what kind, if that was true, what kind of motility are you talking about? Actin-based motility or microtubule-based motility? I could imagine gametocytes possibly having stored things for building the flagellum, but, you know, so motility, two major types, cilia-based or microtubule-based or actin-myosin-based. So which type of proteins are you talking about?
I think in this case is microtubule based,
Fine.
not the use of cilia.
Yeah. Okay.
Yeah, thank you. I think interesting work. I was only intrigued by the fact that antibody responses in symptomatic children were compared with those of, I think these were parasite negative children. I thought probably if we had asymptomatic but parasitemic, probably that could give us a better sense of the gametocyte response, as we know either drug-induced gametocytemia or some other stress-related. So I thought that would be a good comparison to look at.
Yeah, thank you very much. Yeah. So I think in this case, we wanted just to compare within the first cohort, who has gametocytes, who does not have gametocytes. In the future, when we do another study looking at asymptomatics, then we will have alternatives for better comparisons. Yeah, so the positivity and the negativity is not a sexual parasite. All these individuals were positive. All the children were malaria positive. Yeah.
Okay, I'm Philip. So I was wondering, you used NF54 lab strains.
I can see the one.
Yeah. Okay. So during my master's, I used some of these lab strains and tried to switch them to gametocytes. And I observed that NF54 strains had, when you try to do the switching, they go more to gametocytogenesis. So I was wondering if in your analysis you found any difference between these NF54 strains and other laboratory strains, and why they switch easily compared to the other strains. And then my second question is, the participants you had for the bone marrow experiments, were there anyone who were asymptomatic for malaria, or were they all active cases of malaria plasmodium infection? Yeah, so I'll stop there.
All right. So I'll take the second question first. So we had about 40% of the over 200 participants being positive by our ring stage marker, which is skeleton binding protein 1. So that's about 40% out of the over 200. And these were asymptomatics because the study was not designed to look specifically for malaria-infected individuals. We were just taking advantage of the orthopedic cases and sampling the people. So we did not, and we only had about 10 of these who were positive by microscopy. Yeah, but we found more positive individuals when we looked internally compared in the Volta region, because at the younger age group, so they are younger adults in northern region who ride motorbikes and get accidents compared to people in the Volta region. So most of the data from the Volta region, they were largely adults. Yeah. And with respect to NF54 in gametocyte production, actually in our comparing NF54 with the 3 clinical isolates, we found a higher conversion rate with the clinical isolates if we compared with gametocyte. But we did not work with any other laboratory line. But I previously worked on 3D7. And I think you are right. When you compare 3D7 and NF54, you get more gametocytes from NF54 relative to 3D7.
Yeah. Awesome talk, Bismarck. So I was wondering, have you looked also on the drug treatment history of your patients? If I understand correctly, in Ghana, the first-line treatments are 50-50, artemether-lumefantrine and artesunate amodiaquine. Now, amodiaquine was usually associated with higher gametocytemia. So I was wondering whether this treatment is affecting the presence of gametocytes and the level of gametocytes. Have you looked at the treatment history of your patients?
Oh, very important question. So for all, that's for the first assay looking at the children. All the first cohort study looking at the children. All the hospitals in which we sampled the children were using just one drug, which is artemether lumefantrine. So we took the day 0 sample before treatment, and they came back 7 days later. So we had no opportunity to compare between different treatments. Yeah. And we relied on the hospital procedure, so we didn't have to treat them by ourselves.
Hi, thank you for that. Very interesting talk. Very interesting. My question is sort of twofold. First of all, do you think that the female gametocytes that are sequestered are there for the long run? And then the second question is really, what do you consider to be the signal to then release them from the bone marrow if they're going to get back into the circulation for transmission?
Thank you very much. So regardless of whether you have male or female gametocytes, we think that all gametocytes that are in the bone marrow at some point get out of the bone marrow into circulation. And there is data to suggest that the changes in the formability of the different stages, that is from stage 1 up to stage 4. So stage 5 are more deformable. And then keep in mind that the bone marrow has an open circulation, so when they are deformable, it's much easier for them to get out of the bone marrow. But we find that the immature stages that are not deformable are not located in the blood vessel. They are in the parenchyma of the bone marrow. So the open circulation might not facilitate their exit. But I'm just using open circulation as a way to explain how they come out. But we don't have any idea how they get in the first place and how they come out. Yeah, but we think deformability is one of them.
Okay, so let's appreciate this now. Thank you very much. All right, so before we move to the next session, I would like to acknowledge the arrival of the venerable board chair of WACCBIP, Professor Kivgol. He's in our midst today. Professor Kivgol is a distinguished professor who was at the University of Oxford, and he's now a full-time fisherman. All right, so we have Professor Mark Carrington too, who has joined us today. Mark Carrington is one of our long-time collaborators and a member of the advisory board as well. And Keck was here yesterday, so I'm sure you've seen Keck around. Keck is also a member of the advisory board. Yeah. All right. So next session, I think the chairs are Professor Niels Kwashi and Teresa Manford-Jura. Teresa is not here. You are standing in for, okay. So Dr. Elma Ametepe is standing in for Teresa. So thank you very much.
So thank you all, ladies and gentlemen, for the next hour and 20 minutes. We're going to be listening to about 4 speakers, and we have for our first speaker, who will be taking the plenary talk, Emma Wall, and she's online.
I can hear you beautifully.
Good. Good to hear you, Emma. She's going to give us a talk on the infection with Omicron BA.2 increases the breadth of neutralizing antibodies induced by COVID-19 vaccine. So Emma, you have 20 minutes you have 15 minutes of a talk time and 5 minutes question session. So.
Okay, wonderful. Can you all see my slides?
Yes, we can.
Great. Thank you so much for the invitation to talk today. I'm really delighted to be joining your meeting, although I'm very, very sorry I'm not in Accra and I'm currently in London, although I believe it has been hotter here in London than it has been in Ghana. I'm Emma Wall. I'm a clinical infectious diseases physician and research fellow here at the Francis Crick Institute. And I'm building a collaboration with Yao Badako and WACCBIP over our work on neutralizing antibodies and SARS-CoV-2, which is why I'm presenting at your meeting. So I'm going to talk about some of the work we've been doing here at the Crick, and you'll understand by the end of the talk how that will link up with the work with WACCBIP and Yao. So I'm just going to try and progress my slides. Hopefully they'll progress. There we go. So at the beginning of the pandemic in London, our local hospital was completely without any form of SARS-CoV-2 testing, as many hospitals were. And so the Crick repurposed its laboratory facilities. The Crick is a huge research institution in central London, repurposed an entire floor of the laboratory facilities here to turn into a diagnostic testing pipeline for SARS-CoV-2. And over the course of 2 years, conducted nearly 750,000 tests for 9 hospitals, 10 care homes, 1 mental health trust, and the Francis Crick Institute. And the figure I'm showing you now, I'm just going to put my laser pointer on, is the number of tests that were done every day, so a green line is a day, across 2 years of the pipeline. And you can see that at the peak, here in December 2020, we were reaching nearly 4,000 tests a day through this high throughput pipeline. And what we were also able to show is we captured every strain of SARS-CoV-2 that came through London at the time. And one of the interesting things we found is that at the beginning of the pandemic, remember these are mostly healthcare workers, there was a whole mixture of viruses, including European viruses and original Wuhan ancestral viruses. But when Alpha here in green turned up, the pipeline was completely dominated by Alpha until Delta started to pitch up here, and then Delta completely dominated until Omicron pitched up here, and then Omicron is now completely dominating. I'll progress my slide. So Legacy is a research study that was built out of the testing pipeline, and it's a partnership between the clinical side of the testing pipeline, of which I'm part of, and the research centre in the hospital where I have my clinical contract, and the research groups at the Crick. And that links up with a worldwide influenza centre that's a WHO centre for monitoring influenza, and a resource that we have at the Crick that is unique called the Science Technology Platforms. And these are funded groups of scientists, core funded groups of scientists who provide resource for group leaders and other scientists within the Crick. We interface with high throughput screening, and I'll talk about that a lot, our sequencing facility, our flow cytometry facility, and underpinned by scientific computing. And we also interface externally with 2 UK national consortiums, one which was COG UK, you may have heard about our genomics consortium, and the other one is G2P, a consortium of virologists who are understanding the biology of the viruses across the country. So the research questions we've been working on in Legacy are broad. One is about immune boosting, which breakthrough infection or another subsequent dose of vaccine optimizes your immunity of these new variants as they emerge. And then looking at the vaccine specifically, this really crucial question is the immunity generated by the 2020 vaccines that were designed around the original ancestral Wuhan strain remain durable and effective against new variants as they emerge? And what is the correlate of protection? How do we know someone is protected against SARS-CoV-2? That's the holy grail of SARS-CoV-2. We've been studying new variants through our testing pipeline. We've been studying new vaccine targets. And we're building collaborations across the UK and with WACCBIP, as I've already mentioned. Yao runs a study I'm sure you've heard of called Heritage, which has been designed in parallel with Legacy, so we have identical collection points. And the aim at the end is to be able to compare 2 urban cohorts, one in Accra and one in London, and responses to SARS-CoV-2 infection and vaccine. I'm just advancing my slides, sorry they're being slow. There we go. So this is one of the problems we have with SARS-CoV-2 is we have this really close interface between infection and the genomics of the virus and the clinical phenotype of infection and the humoral response, the antibodies that we make, and the cells, which I've put a cartoon up here, all the different cells which are contributing to cellular immunity. And people always mention and talk about T cells, but actually there's a conglomerate of cells that are responding. And so we're trying to work out how best to analyze these data. And we've got a modeling approach, an observational approach, and an experimental approach that we've been working on. Our cohort captures London's high rates of COVID-19. So we've now got 750 participants within our prospective observational cohort. And we have recipients, unlike most cohorts, of all of the different types of vaccine that are currently being used. We've had now 60% of our cohort have had a symptomatic COVID infection, and about 6.6% have asymptomatic COVID. And 44% of those infections have occurred post-vaccine. And the reason this is important is when we consider the biology of the virus, here's the different structural mutations that have occurred in these different variants that we're now very familiar with, Alpha, Delta, and Omicron. And you can see that the mutations are clustering as they accumulate across these new variants in the receptor binding domain and in the furin cleavage sites here in the spike protein. And the furin cleavage site protein is very important because it facilitates the spike protein to open up into an open conformation, which means it can bind with ACE2. Our host response partly produces binding antibodies, which bind the virus, and their role is to flag up to the immune system, here's a pathogen, and produce a subset of binding antibodies which actively neutralize the virus. They bind directly to the receptor binding domain and mean that the virus can't enter cells and replicate. And that's why the virus keeps mutating at the receptor binding domain to evade neutralizing antibodies. So neutralizing antibodies are incredibly important. And we know that they're the best correlate of protection we have in the vaccine trials, where we're looking at here a summary of all vaccine trials, predictive efficacy on the y-axis up to 100% and neutralization level on the y-axis. And there's a very neat correlation across all the different types of vaccines in the amount of neutralizing antibodies they induce and the protection they induce. But we also know from the recovery trial, a large clinical trial run in the UK testing interventions, that when participants in recovery, these are unvaccinated, predominantly unvaccinated participants in the trial, were randomized to synthetic monoclonal antibody combination versus usual care. Those that were antibody negative at the time of recruitment to the trial had a survival advantage. They were more likely to live if they were given the synthetic neutralizing antibody than if they had no antibodies detected. If they had existing antibodies, there was no difference. The neutralizing antibodies are important, but they're hard to measure in blood. So the CRIC has developed a high-throughput live virus neutralizing antibodies assay, which runs 384 samples. So we can run 1,000 samples against 3 variants in a week. So we rack up our samples, we heat inactivate them, and then we dilute them into 4 dilutions. They then get set up with cells, and then virus is added. They're incubated overnight in category 3, and then they're stained with an anti-N protein, which fluoresces. So we can quantify the amount of N protein or virus replication, and that's imaged by an automated microscope. And this is what the plate looks like. So here are our negative controls down here. Blue is cells, and here are our virus controls here. In dilutions looking nice and green, the virus is replicating. So if we look at one participant in the first 4 dilutions, this person here has got no neutralizing antibodies. They're making masses of N protein, bright green. Whereas this person here, actually at the highest dilution, neutralizes the virus entirely. And as you go down the dilutions, only at low dilutions do they actually start to fail to inhibit full virus entry. So we use this system to automatically measure the fluorescence, and then we can quantify the neutralizing titer. And we've had a lot of success with this assay so far. So most of these types of assays are done manually and low throughput, and you can generate between 10 and 20 data points. We're looking at 250, 500 data points at a time. So we're looking at the y-axis here on the IC50, that 50% neutralization level. And here's our first study that was published last year, looking at people who'd had Pfizer vaccine and those original doses of one first dose and then second dose. These are blood draws after each dose. And you can see here against the wild type, this type the vaccine was designed against, after 2 doses, you get great levels of neutralization. But actually, as you go across the variants, here's an ancestral related variant going to alpha, beta, and delta for this study. You can see that even after 2 doses, there's a slide down in your neutralizing titer. So you get less antibodies, about a fifth less antibodies against delta than you do against wild type. And when we looked at this against different types of vaccines, so this is now looking at ChAdOx, the Oxford AstraZeneca vaccine versus Pfizer-BioNTech, and this is after 2 doses, you can see that while you still, across both vaccines, get great antibodies, as you go down the variants, back down to Delta, people who received ChAdOx, they were really split. There were some people who responded and a lot of people who didn't respond at all. Then we were asked by the government to look at waning and how long after vaccine did people still have detectable antibodies. So these are now people who had either Pfizer-BioNTech or Oxford AstraZeneca against Alpha, Delta, and now Omicron. And we were looking at the waning at either 2 to 6 weeks post-vaccine or 12 to 16 weeks post-vaccine, so 4 months post-vaccine. And you can see that against Alpha there was a wane, Delta there was a wane, and Omicron there was a significant wane. So these are people who'd had 2 doses, 4 months later, a lot of them had no neutralization against Omicron. And if you'd had the ChAdOx Oxford AstraZeneca vaccine, there was no neutralization by 4 months. So then we started to stratify people because we were starting to see this difference between those bunch of responders and those ones sitting at the bottom who didn't respond. So these are again looking at the neutralization after the 2nd dose, stratified by COVID symptoms. To see, if you've had a prior infection, you'd seen another dose of spike, did that help you? And yes, it did. Particularly against Omicron, any prior wild type spike exposure boosted your antibody titers against Omicron. And then if you had your dose of virus after your vaccine, so you'd seen vaccine wild type, then you had much higher levels of antibodies. So the order in which you see your doses of spike, your immune system sees your spike is important. And then when we look at the 3rd dose, we can see that most people picked up and had a really good response after the 3rd dose against Omicron, which is really reassuring. And these are some of the data, along with other groups, that really spurred the UK to have a very significant 3rd dose campaign at a population level last Christmas. So then we started to ask questions, this is now moving into our unpublished data, about what could we say about these people with their narrow response, the people who don't really neutralize any of our variants, and particularly not Omicron, and the broad neutralizers. In this figure, each column is a person. We're looking at neutralizing titers. Yellow means they've got high titers, blue means they don't neutralize, across the different variants. And we can see some people who don't do anything at all, some of these narrow people who do manage to neutralize wild type alpha a bit, but can't neutralize Omicron and Delta particularly, and the broad neutralizers who neutralize everything. And then those people who had had a prior proven infection, you can see they light up like a Christmas tree. They're right in the middle, they neutralize everything. So then we stratified by symptoms, by vaccine type, by sex, and by anti-N, and certainly the broad and the prior infections do have anti-N antibodies, but everybody has a positive S1 ELISA. S1 ELISA does not discriminate between these 2 groups. Neither does age, sex, or vaccine type. And when we look at these 2 groups over time, looking at neutralizing level on the y-axis here, over time on the x-axis, the red are the broad and the blue are the narrow neutralizers, you can see that against wild type, mostly they do all respond after the 3rd dose. And certainly after the 2nd dose, there's a reasonable response. But the 2 curves diverge significantly against Delta and even more so against Omicron. But after the 3rd dose, the neutralizing titers for the blue dots, the narrow responders, do pick up. So that narrow response is rescued by another dose of spike. And then when we ask the question, well, what about what type of your order you've seen the spikes in? So these are people who'd had Oxford AstraZeneca for 2 doses and then been given a mRNA Pfizer boost, or these people who'd had 3 doses of Pfizer. Actually, that initial low response we saw against ChAdOx picks up massively when you've got your 3rd dose of the mRNA vaccine. In line with all the data to suggest the mRNA vaccines are more immunogenic in generating neutralizing antibody titers. And actually against BA2 now here, as well as BA1, we saw that that 3rd dose you had great response. And what's really interesting is after this 3rd dose, the breadth of everybody's antibodies, even though this is still Wuhan spike that people are being vaccinated against, actually they still neutralize everything, including BA2, at a good level. Oh, my slides again, sorry. So now we started to ask the question about poor spike exposures, these people who'd seen 3 doses of vaccine and a dose of COVID, and these are stratified now by anti-N antibody. And these, again, unpublished data. I'm just asking everybody not to screenshot and share these slides. They're being prepared for publication as we speak, and we really don't want them shared on social media, please. So these are people who had a heterologous boost, AZ, Pfizer, or homologous boost, Pfizer, and then we're stratified into narrow and broad, and actually that narrow response is rescued in those people who had a positive N. So irrespective of whether you were a narrow or broad at the beginning, by the time you've had another dose of N, another dose of wild type spike, that irrespective of which order you saw that in, your breadth of your response, you get more antibodies made. So in summary, 2 doses of vaccines are essential to mount an effective neutralizing response. 3 doses of vaccine generate much more pan-variant broad neutralizing antibodies, and post-vaccine infection increases your antibody breadth against BA1 and BA2. So the impact of this has been that we've built this responsive research platform in the CRIC. It's been called by the editor of The Lancet a living lab, and we're developing now a COVID-19 research resource within the CRIC. And we've established partnerships within our hospital and developing a component of the hospital, the way the hospital and CRIC science together can respond to future pandemic threats. And we've had impacts in the UK on policy and patient care. And our papers so far have been cited over 600 times. So there's been an enormous group of people who've been involved in this work, and there's no time within my 15 minutes to thank everybody, but I really have to acknowledge the leadership of the study, particularly Sonia Gandhi and Charlie Swanton, who set up the testing pipeline and the high throughput neutralizing assay, supported by Steve Gamblin and colleagues David Bauer, particularly, who I work very closely with on this, Ed Carr, Kathleen Wilkinson, and Simon and Jules and their team. Mary Wu developed the assay that we've been using for all of this, and a huge amount of support. There's also a huge amount of acknowledgements on the UCLH, the hospital side, without whom this study would not have happened. And I have to also acknowledge our funders and our external collaborators. So I'm going to end there and open the floor to any questions. Thank you very much.
Thank you for your presentation, Dr. Wall. We'll take questions now from the audience. So I see Dr. Kwashi's hand up.
Hi, Emma.
Hi, Peter. How are you?
I'm good. Thanks for the fantastic talk. I had a question about the breadth of. I was surprised to see that S1 lights up across board, and it doesn't seem to correlate with any neutralization whatsoever.
Yep.
Were you guys surprised by that? Because I would have expected to see that for nucleocapsid, but not necessarily S1.
Yeah, I'm just going to see if I can stop. All right, what have I done? I don't want to do that. I'm trying to stop my screen share, so I'm just trying to make my mouse work so I can see you. There you go. It is really interesting. So S1 ELISA looks at binding antibodies, but doesn't quantify that neutralizing component. And when we've looked at the relative association between binding antibodies and neutralizing antibodies against Wuhan strain, they correlate very nicely. But certainly as you get more divergence away from Wuhan towards Alpha, Delta, now Omicron, now BA5 we're looking at, you definitely see that there's a really big disconnect. You still make loads of anti-S binding antibodies induced by the vaccine. They're long-lived and they hang around, but people don't make anywhere near as good anti-BA5, particularly neutralizing antibodies, and across all the Omicron subvariants.
Okay. I don't know have you tried using whole, I mean, I don't know what point that would be, but just to see whole spike antibodies?
Yes, so the Roche anti-1 ELISA, S1 ELISA, we originally developed our own in-house ELISA because it was cheap and high throughput. And we've now we bought the Roche platform finally, and we've been running the Roche anti-spike ELISA, which is a whole spike ELISA, rather than, and looking, it has a specific receptor binding component, domain component within there, and we're looking at both, and actually we see the same correlation with both the whole spike and the receptor binding domain.
Okay, all right, thanks.
So the message really from that is that we can't say whether someone has got quality neutralizing antibodies against a new variant based on the S1 response, unfortunately.
Yeah, so that means that the company selling spike antibodies at neutralizing antibody detection should really rebrand.
Yeah, so they can rebrand them as like you've made an immune response to the vaccine, definitely, but they don't tell you whether you've got antibodies against the new variants. The MSD platform does have more subvariant assays, but they're still binding rather than pure neutralizing. And as I know, you're developing your pseudovirus assay. There's lots of nuances into looking at neutralizing antibodies that need to be standardized, and that's something you and I will continue to talk about, I think.
Hello, Emma. My name's Keith Gull. In terms of the living laboratory concept, and I may have sort of been confused at the end, in which case I'm sorry, but I take the point about 3 doses and then exposure. Did you do an experiment which sort of said versus 4 doses?
So we haven't got enough people in our cohort yet who've had 4 doses. Because the UK government has prioritised immunocompromised people for 4th dose, and over 50s are going to get a 4th dose in the autumn, so we will be able to answer that. The other thing that the testing pipeline gives us in our healthy adults who've been under occupational health screening is we know exactly when they were infected and with which variant at what time point. So we can say definitively, we bled you here, we know your vaccine dose, we know your infection exposure, we know which variant it was. We've had people who've had now 3 spikes from wild type as well as 3 vaccines. So we've got people who've had up to 6 exposures, and we're working with Adam Kucharski at the London School to try to model that, because you can imagine as we ingest all of these complex data, and this is something Yao and I have been talking about, how do you keep collecting the data and analysing it in real time? And we've built this R object that really cleans the data as it's ingested and allows us to analyze with code as it's going forward, but it's getting increasingly complicated. And it's a really important question about that 4th dose of vaccine, what's going to happen. It looks like in the UK, they're probably going to give a combination Omicron spike, Wuhan spike vaccine, but that hasn't been announced formally yet. And we're really interested to study that as that's rolled out. But within our, going back to your original question, within our original cohort, we don't have enough people who've had a 4th dose, and those people who have done are immunocompromised, so they're not representative of the wider healthy cohort.
So given that policy issue that you just talked about, what would you do? Would you actually inoculate people with a 4th dose, or would you just let people get the virus?
Oh, it's such a good question. So this is something we are thinking about all the time. So there's a definite wane. So the data I've showed you up till January, and we're bleeding everybody at the moment. The entire cohort's being bled, and we're running the data this week. So it's not hot, I can't show it to you yet. But we're looking at the wane, and I suspect that what we who's vulnerable should get a 4th dose in the autumn. And I certainly, if I was clinically vulnerable, I'll get a 4th dose in the autumn.
Yeah, you got kind of...
Oh, sorry.
Yeah, could you just go back to the last statement?
Yeah, sorry, I saw that my screen froze. The connection between London and Accra is clearly, hopefully the pipeline will keep up. So I think that what we're going to see now, we're bleeding our entire cohort now. What I think that we should do, what we're looking at is that the wane that's happened between those January bloods that I showed you, that post-3rd dose boost, and now everyone's going to wane significantly. And we're going to see very low levels of antibodies. And so that over 50s boost is going to be entirely appropriate and for clinically vulnerable adults. Whether or not the wider population should have a boost is a much more moot question, and something that we're continuing to talk to the government and use that assay about. But we don't know yet.
Hi, it's just comforting data, right? Having the proper vaccination really can protect in a lot of differences among individuals. So my question is, neutralizing antibody is important, but you didn't have a time to share any data. But I'm just wondering what happened with the cellular immunity. I assume it's also boosted, but I mean, any comments about that?
Yeah, so our cellular immunity I've deliberately left out of the talk, because it's very hot, and hopefully it's going to Nature in the next couple of weeks. And so I've been asked not to present it, but we are seeing definite differences in the cell population between the broad and the narrow responders. And there's a specific cellular subtype that we think may be involved in generating the differences between these broad and narrow responders. And so we've been developing a moderate throughput pipeline, looking first at ELISpots, which are interferon gamma production by T cells, particularly CD4 T cells in response to SARS-CoV-2 peptides. And that combined with mass cytometry, which is a moderate high throughput way of looking at up to 37 different parameters to define cell populations. The next question is going to be with that interesting cell subset is, what's the function of those cells? What are they doing? And we need to define that experimentally. That's another piece of work that we're building on. But you can't see behind me on the wall, but within the study, we've got a large number now of interesting questions about the virus, the cell biology, the humoral response. And we do have to remember that neutralizing antibodies are made in a combination where B cells meet T cells in the germinal center, and that somatic hypermutation and selection. So they do represent a bit of the T cell response, because you have to have functioning T cells to make a good quality neutralizing antibodies. But what this cell subset we're in process of investigating is doing, watch this space.
Thank you.
Okay, thank you very much, Dr. Wall, for your talk and for your time.
Thank you for inviting me. It was a real pleasure to be here.
So now we'll take our fellow session. And first on the list is Isaiah Deborah. In 12 minutes, Isaiah is going to tell us about the genetic diversity and population structure of Anopheles finestus in Western Kenya based on mitochondrial DNA markers. So, Isaiah, you have to do this in 12 minutes.
Thank you very much. Thank you very much. My talk will be on the genetic diversity and population structure of Anopheles finestus in Western Kenya. This is a fraction of my PhD work. This is the outline of my presentation. We all know there are many mosquitoes, and mosquitoes are known to transmit various pathogens. Some transmit arboviruses, and then some are also known to transmit the malaria parasite. But in the malaria ecosystem, the genus Anopheles is responsible for transmitting malaria across the whole world. And in sub-Saharan Africa, the Anopheles funestus group and the Anopheles gambiae SL sensulato groups are responsible for malaria transmission. And in the Anopheles funestus group, Anopheles funestus sensus tritu, hereafter Anopheles funestus, is the major vector responsible for malaria transmission. It is a member of the 13 sibling species known in endemic areas in Africa. Our study focus on Anopheles funestus, and one may ask, why Anopheles funestus? It is, like I said, is the major vector of the 13 sibling species, and is well known for its notorious anthropophagic and endophilic behaviors. Anthropophagic means that it feeds mostly on human hosts, and endophilic means is like resting mostly indoors. Its capacity to transmit the malaria parasite outpaced Anopheles gambiae in some endemic areas, and it once played a major role in malaria epidemic in southern Africa. It has the capacity to adapt and colonize different ecological niches. Therefore, this study was designed to delineate the fine-scale population structure, which could be useful to investigate the genetic basis of speciation and local ecological adaptation process. Our recent study in Western Kenya has shown that this species, Anopheles funestus, co-breeds in the same habitat with other malaria vectors like Anopheles gambiae, Anopheles arabiensis, Anopheles costani, and it has the capacity to adapt to various ecological zones. But the genetic basis of this adaptation is poorly understood. Therefore, this study sought to assess the genetic structure and diversity of Anopheles funestus in different ecological niches in Western Kenya. The study was conducted in Bungoma. Bungoma is in the highlands, in the borders Uganda, and then Port Victoria is in the Lake Basin. It also borders Uganda. And then Kombewa is also in the lowland near the Lake Basin. And then Migori is on the highlands towards Uganda. So we sample adult mosquitoes resting indoors early in the morning using Procopac. And Procopac is a mechanical device that is designed to sample mainly mosquitoes. And it has a paper cap inserted and it has a battery. So once it's powered on and I move it along the wall, it sucks every insect, including mosquitoes. Samples were then knocked down and then they were preserved, and total DNA were extracted from all of them using the chillers method. And samples were, after the extraction, cytochrome oxidase subunit 2, which is known as the COI2 gene, was amplified using PCR, and Sanger sequencing was used to amplify the applicons. And Genius Prime, Mega X, Alequin, and DNA-SP were employed for the analysis. Our results have shown that Anopheles phonesus population in Western Kenya has very low nucleotide diversity, but high haplotype diversity. High haplotype diversity. And our further test of statistical neutrality test revealed negative Tajima D, and then full statistics also, full statistics also indicate negative values. And this indicates deviation from statistical neutral model, indicating population expansion and excess of low frequency polymorphisms in the population. Our further test FST values indicate that there were negative values, and this indicates that there is a weak population structure of Anopheles phonesus in Western Kenya, and this was further confirmed by a high gene flow in the population. The high gene flow observed were from between Bungoma and then Port Victoria, and this was followed by Port Victoria and then Migori, and then the least gene flow was observed between Bungoma and Kombewa. Our inferred phylogenetic tree has shown that Anopheles phonesus-like, which is a member of the Anopheles phonesus group, is a common ancestry with Anopheles phonesus in Western Kenya. And these haplotypes, haplotype 47, 42, 57, were the closest haplotypes. And these haplotypes were mainly from Migori, which borders Tanzania, and Bungoma, which also borders Uganda, and Port Victoria, which also borders Uganda. So we hypothesized that this mutation might have originated from neighboring sub-Saharan African countries. Our inferred haplotype network, we observed that haplotype S1 to 20 were shared across all the study sites, and haplotype S1 is the recent common ancestor of all the haplotypes in Western Kenya, Anopheles phonesus population. Summary and conclusion: there was high level of gene flow across all the populations. Haplotype S could be an ancestral haplotype among the Anopheles phonesus population, and the high number of haplotype diversity per study site and the population expansion observed in this study suggests the high adaptability of Anopheles phonesus to various ecological requirements, hence sustaining its vectorial capacity and malaria transmission. Thank you.
Okay, thank you, Isaiah. You have extra time for questions now. So before I let you take the mic, can I just ask, maybe it's a bit of a naive question. Is there evidence of Anopheles funestus in other sub-Saharan African countries?
Yes, please.
Okay, like which ones? Like Ghana and...
Yes,
Oh.
Ghana, there were few papers published on Anopheles funestus. And then East Africa, very common. And then Southern Africa, and then Central Africa.
Okay, so why Kenya? Why did you do your study about tribes in Ghana?
Yeah, part of my study is still ongoing in Ghana,
Okay.
but Kenya, because my funders, they have their study, their location. Kenya is one of their study locations. And Western Kenya, because it's highly endemic zone in Kenya.
Okay, so you're also like susceptible to your funders' choice. Yeah. All right, then I'll let you take the mic now and answer your question.
Thank you, Isaiah, for a wonderful presentation. Maybe I missed it, but I wanted to know which Plasmodium species does the Anopheles funestus transmit?
No, it transmits mainly Plasmodium falciparum, because that's the major one. That's the major one in sub-Saharan Africa.
Mainly falciparum.
Yes, please.
Okay. Well, have you tried looking if they carry any of the other Plasmodium species?
We are yet to get there, but I've not come across any paper that suggests that it transmits any other parasite apart from PF.
All right, thank you.
Thank you, Isaiah, for your presentation. That was a great one. I have a question. I want to find out the population expansion that you observed. Was it the same in both the highland sites and the lowland sites?
Yes, please. There is both. It's across.
Okay, so with the gene flow patterns, do you think it was due to insecticide pressures or breeding among the populations?
Your question again?
The gene flow you observed, was it based on ecology or breeding among the populations, the different populations?
No, it could be as a result of migration. And windborne migration has been documented to play a major role in dispersing mosquitoes from one geographical area to another. So it could be as a result of windborne migration of mosquitoes. That might be a factor.
Okay, I don't see any other hands up. Isaiah, thank you very much for your time. Our next presenter is Christian Alexander Schneider. Is he here or online? Oh, okay. So in 12 minutes, Christian wants to tell us about predicting the impact of COVID-19 vaccination campaign, a flexible, age-dependent, specially stratified predictive model accounting for multiple viral variants and vaccines. Over to you, Christian.
Okay, thank you. Hopefully, is it okay with the mask?
Yes.
Okay, so you don't have to be afraid of large droplets. Okay, so we heard about a lot of communicating COVID research to the media, and that's a bit what I want to tell you also what we did with our model. So it all started like 3 years ago in 2020 as a normal day, and like I was in Cameroon, and like a week later we've been in a lockdown. And so to kind of use up our time. We started day and night working a lot. I worked a lot with Christian and Esmer. They're sitting there. Can you wave so we can see you? Yeah, we're working very hard on these models. We were programming all the time. Let's see. Yeah, and then finally, a year later or so, we predicted for the major tabloid in Germany, the third wave in Germany, pretty accurately. Yeah, and the two scenarios, one scenario actually came true with the large lockdown, and we were pretty accurate. And then it started with the media coming more and more and more, and our predictions were like, we were all over. And so we had over 100 media appearances, newspaper interviews, whatever. So how do you get accurate models? So it starts like with a base model. Yeah, we have a population of susceptibles, and then we model the disease by first a latent phase, where individuals are not infectious and have no symptoms. Then they enter a prodromal state, where they start already, being infectious, having still no symptoms. Oops. And then there is like a fully infectious period where symptoms might start, or infections stay asymptomatic, and they're not like they are now full infectious to the full extent. Now that is followed by the late infectious state, where the infectivity goes down, and finally individuals would either recover or die. That's like the base model, and you typically get this type of SIR model predictions that I'm sure you've seen. Yeah. Now, if you try and predict a pandemic accurately with such a model, you ultimately fail. Yeah? And the problem is all these models, the dynamics are not right. You want dynamics that look more like the dashed lines. So in these models, intrinsically, you don't get the timing right. So you need to have some modeling tricks doing that. And that was our USP. That's why what was one of the parts why we could always parameterize our model so much better than others. So the trick is we need to adjust the variance, and we do that by modeling all these phases in the infection by substages, where individuals progress like successively, and that allows us to adjust the variance of the durations. And that's essentially what we do. So essentially, the next thing is, let's look at the demographics. This is demographics in Germany. Yeah? And let's look at the contact behavior. Yeah, there are estimates for contact matrices. So there are contacts at work. Well, they are clearly age dependent. Yeah? Then you have contact at school. They are also clearly age dependent. Yeah? Older people are no longer in school, yeah, except they are, they get to repeat a lot of years. Then at home, we see a strong correlation, essentially, with your spouse, with your kids, or with your parents' generation. And then you have other contacts, which is like going shopping, going to sports, whatever. Yeah? But we see a clear age structure everywhere. So you can't ignore that. And you cannot ignore demography. See, for instance, here we see the demography of Ghana. That looks totally different than Germany. Yeah. So also that will affect mortality a lot. Yeah. So by the way, I was not one of the modelers that predicted that the African country would be wiped out. Yeah. On the contrary, we say it should do much better than Europe in terms of mortality. And also the contacts look different. Especially contacts at home. Family structures are different. So you get to bring in also age dependence. So we need to take all these compartments and make them age dependent. And all the model parameters, the durations, the infectivity, and so on, they all also got age dependent. Now, next thing is, let's look at the population density. So we see Germany. Germany is a rather mobile country. Right? We have these highways where you have no speed limit and so on. So you have a lot of mixture in the population. Now let's look at Ghana. Here definitely you see the population being centered at certain urban areas. No? Now definitely with such a population structure, you cannot no longer ignore some spatial structure. So you get to make the model also spatially dependent. Yeah? So all these compartments are going to be modeled for several locations. So essentially we have a discretization of a partial differential equation model there. And so all these parameters become agent and density dependent. Now that is the first story. That's the base model. Now we want to add the vaccine and we want to add mutant variants. So that model was really designed to investigate, to predict the effect of certain vaccines and viral variants. Yeah, especially once the beta mutation came out and the AstraZeneca wasn't protecting much against, we're setting up the model like that. So we have the whole model structure again, and we got to distinguish now between those individuals that are unvaccinated, but they are like willing to get vaccinated. Yeah? Then there are also some that will that are anti-vaxxers or fail to immunize. Yeah, so we need to have that whole thing in parallel. We model them separately. Then we have those, well, the unvaccinated eventually get vaccinated at some rate. Yeah? These rates are, of course, age dependent. Yeah? In all the countries, you used to have like an age prioritization of the vaccine campaigns. Yeah? So you get to capture that in a model. And then, well, we model this vaccinated individuals, and it takes, a while until the vaccination cycle is complete, and the immunization outcome manifests. So one of the outcomes was you're going to get either totally immune, at least against one variant. And so initially we model that. Or like if you're already infected and you get vaccinated, we just, remove those, put them to treat them as recovered individuals. Or the majority will get partially immunized. So that means they are not as easily infected, they don't transmit as easily, but you're still getting infected. Okay? And some might fail completely to immunize, so we just treat them as unvaccinated or as anti-vaxxers. Now, then came Omicron. That was the game changer, so the model was not complex enough. Yeah? So we just add some more complexity. And with Omicron, there was one game changer. Reinfections can no longer be ignored. So we get to capture that. The other thing we get to capture is we get to capture booster vaccines, and we get to capture fading immunity. When Omicron came along, also these concerns started coming up, so we modeled that. So the model structure got a bit more complex, but still, the nice thing of that model is it can be parameterized still rather intuitively. Now, how to do that? Well, you just, some of the parameters you get by just opening the newspaper, yeah, and what is the vaccination progression, what are the vaccination rates? They're easy to estimate. Some other you get from the literature, and some other you get to check. Particularly what you get from the newspapers are the COVID restrictions. So which lockdowns, which measures, mandatory facial masks, contact restrictions, and so on are applying. Yeah, so that's what you read from the newspapers. Sometimes you can look in the future and predict what politicians are doing. But the further we went into the pandemic, the more unpredictable the politician acts, actually. So it's being tough now, because we used to be very good like 6 months in advance, because it was rather clear what's going on. But then they started, not just that the complexity of measures started to increase, but also like the frequency in which they changed the measures. Yeah, and eventually also like the experts that would on a daily basis monitor that would get overwhelmed. So anyhow, let's see. So that was, how good is the model? That was a prediction with Omicron depending. So that was a prediction. We did it on December 8th last year when Omicron was about to enter Germany. And so we said, you know, depending on which type of contact reductions are going to be implemented now, we predicted these waves. And actually, well, that is how it looked then. Well, we predicted until May as a period, about until that time, yeah? It looked rather similar. Well, we see here this little bump here. Well, here also, you just, you know, checking the calendar helps, yeah? That was just the beginning of the winter holiday season, yeah? So they didn't test all the school kids that had super high incidence, yeah? So that is just a kind of data artifact, yeah? So usually it should be, but we were actually pretty accurate, yeah? Now that is what we predict for the fall. And winter season. We didn't, that was an old simulation where we did not include BA4 and 5, so it started kind of a bit earlier, but what we're going to predict is a rather strong wave again. Definitely they are not testing anymore, yeah? So, well, we see mainly most of these infections will stay under the radar, but we're seeing these infections. In terms of well, mortality and hospitalizations, things won't be that bad. But the problem is going to shift. That we predict. The problem is going to shift because the healthcare workers will be on sick leaves. So, kind of the hospital capacity is diminished automatically by having this high incidence. So, like in the Delta wave, what we saw is, well, we had quite a number of severe infections, and the ICUs were overwhelmed, and they had all these ECMO therapies, which are labor intense. So they just couldn't fill up all the beds because they needed like teams of 5 people then rolling down the patients in the ECMO. Now this time the problem is that all the staff is on sick leave. And so that's diminishing the capacities. So anyhow, those are our prediction in FAST. So, oops, stay healthy. That slide kind of turned up weird, but that's okay. Thank you.
Yes, thank you for that, that very awesome presentation, Christian. The lockdown turned you into someone who does lots of predictions, yeah? So maybe just a light question. Can you predict when the variants are going to stop?
No, but you know, looking at the variants is interesting to begin with. Because if you look at that, we had the wild type going on approximately for a year. And that virus is not mutating a lot, no, compared to other viruses. And now just look at the frequency of mutations. Of BA2, we have now BA275 or so on, so we have 75 subvariants. Just the frequency in which these mutations occur is much, much higher. So that tells us how high actually the unreported cases are. But it's rather clear, the more infections we have, the more likely it is that a mutation will occur and then eventually spread. And mutations can only spread if they are net more contagious than what's there. And that means, you know, in the context of the immunization of your whatever population. So definitely the vaccination status might matter now. So you see in some countries some variants spreading, some others not. But mutations are going to come up. Yeah? And so what is really the sad thing is everybody's is talking about the end of the pandemic. And like in the beginning of the pandemic, everybody was talking, we need a vaccine. Yeah? So that was kind of the right thing to do. Now, nobody's kind of saying, you know, we need a vaccine that causes sterile immunity. Yeah? And that is where we need to go to finally get out of that. Yeah? So as long as we cannot get immunity in the mucous membrane, we won't see this pandemic ending shortly. And definitely that thing is going to be always a step ahead, and it always has a joker in its hand. Yeah.
Very interesting. Just for context, because you are trying to juxtapose trends in Ghana and in Germany, can you tell us the data that comes out of Germany in terms of case numbers, mortality? How is that collected? Does that include self-testing, hospital data? What is in that data?
Okay, so in Germany, you know, the best reward anybody can tell you is not too bad. Yeah, so everybody's complaining about the data that is being collected, and it's not optimal, definitely. So what they would count as infection would be a PCR confirmed infection. That's really what is there. They don't count antigen tests, and they don't count self-tests. So usually, in the beginning, you could also kind of trust it, because they would take an antigen test and then confirm by PCR, and that worked. Right now, I doubt that it's going to be done. People just might take an antigen test, but nobody bothers anymore to get PCR tested. So we just have a huge unreported number. Now, what Germany is not doing well is we have no clue who gets infected. So they don't publish the age, and they don't publish the vaccination status. So essentially all the data that needs to be now more complex, the more advanced we are in the pandemic, is not really being collected. So data is definitely suboptimal. You can kind of get a bit of the unreported number just from the model dynamics, because you would see the up and down trends of the incidence. And you know, with a certain, if the unreported cases would be low or higher, you should see incidence going up slower or faster and so on. So in a way, these models help you also to get a grip on the unreported numbers. Yeah? But the crucial data is not collected these days, and it's getting more and more difficult. And kind of, I was in a modeling workshop last week with all the German modelers that kind of are dominating the field. Now they are all complaining about the same things, particularly those that have data-driven models that just fit their models to the data, whereas we have a more mechanistic model that gives a better advantage because we're not so vulnerable to data not being present.
Hi,
hey.
Kristan, thanks for your presentation. By observing the little schema that you showed with respect to the variables and the features of the model, as you can see, they keep expanding by things that happen, changes in policy, vaccination, etc. So with this expansion, which we all know is not going to be going to stop in the near future, the model can get very, very complex. So I assume that this was done explicitly, like you explicitly programmed the model based on those features. Is there any consideration of incorporating some kind of AI where, you know, AI has a certain, potential to accommodate such large features, especially in deep learning? So I'm asking if there is a possibility of that, your team considering that, or whether that doesn't really fit in this.
That doesn't.
Thank you.
Thanks for the question. Definitely, you can try and so you could try and do like a model selection and so on, finding which would be like the optimal model, which features you could, which part of the model you can actually neglect. The model sounds more complex than it actually is, yeah? Because if you see the waves going on, yeah, and we had it in the talk before, you have like the wild type, and then the alpha mutant would take over completely. So essentially, once the wild type is gone, you can toss this equation out of the model. Yeah? So you can just simulate the model until the new variant comes and you change the initial conditions. So you can do it like in a stepwise fashion, and then you don't need to have all these equations, because obviously they are going to increase kind of geometrically. And the same is like with the vaccines. Now actually what we're seeing, we see less and less vaccines being used. So in Germany they would just use BioNTech and Moderna, which we treat like in the model as one vaccine because they have rather similar properties. Different was the Johnson & Johnson or the AstraZeneca, which we modeled explicitly. But if you just do it in a stepwise fashion, you would always in chunks reduce the model, and so it's still doable. Yeah.
Okay. Thanks, Kristen, for the talk. I'm wondering whether you use, I think what I've noticed is sometimes in terms of number of cases is not as predictive as test positivity. Because here, this is just anecdotal, but I've noticed that the test positivity, if you follow that is more predictive of the wave as opposed to number of positive cases. And if you put that in there, you might correct or get a more, going back to when, let's say, going back to when more tests were being performed, you could find a way to...
I fully agree. I fully agree. So the test positive rate is extremely informative. Now, I wouldn't say, there's no one size fits all. Yeah. So these models were designed to be applicable to any country, and that is what we planned to do. Yeah, and we started, doing like modeling prisons in the U.S. and so on, until we said, well. We started modeling Germany, and then we are kept busy with that, and so it's getting eventually overwhelming. Now the countries are definitely different. In Germany, for instance, they would change the game, yeah, while it's going on. Yeah, so like with testing, they would just then. Switch to symptomatic testing and so on. Yeah? Yeah. So if you change that so substantially, you have the problem that you cannot compare these numbers anymore. So you get to wait for a while. Now with the positive rate, I'm skeptical for one reason. What's happening is there is a certain baseline that you test. So we would PCR test healthcare workers. Now, of those, you expect them to be negative, yeah? So you want just a confirmation that they are negative after all, right? Yeah. So you have this baseline of negative tests. Now, if incidence is really low, essentially those are the only PCR tests that show up in the statistic. Yeah? Then if it picks up, you're testing different individuals. So you get to also incorporate that. And there, the problem is the lack of transparency. That's making things worse. But definitely, yeah, I look at the positive rate too, because that's often more informative. Yeah, definitely.
Okay. And then another comment on because if you look at Ghana, take Ghana as a case study. In terms of our COVID-19 policy, I think we were A1. But in terms of implementation, I don't even want to give a grade, because we had mask policy, we had public gathering restrictions, bars were closed, everything, but in reality, none of this actually happened.
Oh yeah, those things work very well on the paper. Yeah, paper is very patient. Yeah, well, after all, Ghana did not do so bad. Yeah, and you don't need good COVID management. You have Gordo. Yeah, as he making so he's taking care of everything. Now, definitely. Compliance is a problem. Now, really in Ghana, I don't think with that demography that it's that big of a deal. The real danger was in the beginning that that you have the virus spreading like a wildfire here, and that by international mobility, you import it into more vulnerable countries. Yeah. So comparing, like, I think in Ghana, 3% is 65 plus. In Germany, it's 22%. So the risk group is substantially higher. So also the propensity to get vaccinated, the incentive to get vaccinated, is very different there. Yeah, also for young people. Yeah? Because if you weigh the risk versus the benefit, well, you still have the solidarity, the thinking. You know, well, you got to protect your grandparents too. Yeah, somehow. Now, in Ghana, the story is different. Yeah, because the risk group is so small. Yeah? So, and you have to see all those things like in the context, I guess, of the society you're really in. Yeah, this, you know, this top-down approach. Yeah, I know better. Yeah, that doesn't work, I guess.
Yeah, so I can see that a lot of people have questions for Kristen. So I'm going to take the last question, and then subsequent people, you have to catch him during lunch and then talk a lot more. So, yeah, you have the mic.
Thank you, Kristen. Just a short question.
Can you wave your hand?
Yeah.
Okay.
Okay, given that we've learned a lot from COVID-19, I would just like to know if this model can work for maybe in case we have future emergence of new viruses. Can the model still work for it?
But you always get to adapt, yeah? So I don't think there's a one-size-fits-all. So the first thing you get, you know, the mathematician can never dictate the biology of a disease, yeah? So you get to adapt it to the specificities of the virus. Definitely for airborne viruses, that is okay. If you take a pandemic that is longer, like if you want to have an influenza model, I would also, you know, kind of model the age progression and so on, because there the time duration you're looking at is different. Those models are designed to be accurate for, let's say, the next 2 months, yeah, just to respond. With influenza would be different, but definitely these type of models can be used or adapted. Now, for instance, for Marburg virus, yeah, which we have in Ghana, or we had in Ghana now, yeah. I would take a different approach, yeah, because also there like contact tracing and so on is different. Also, the incidence is much lower. Yeah? The same is with Ebola and so on. With this highly lethal viruses, usually it might be better to do it like on an individual basis, because you have so few numbers. And then there also you don't need to model like the age structure so much. Yeah, with Ebola it doesn't occur because it's just smear infections. Yeah, and so you always get to think clearly about the biology of the virus. Yeah, and so the first thing is you get to talk to as many medical people you know. Yeah, so you got to know what is the medical practice. You get to go in this thinking. The next thing is you get to talk to the biologists and to the immunologists. Yeah, because a lot of people just adapt their models that they have, yeah, but it's just more of the same, and the prediction will be as bad as usual. Yeah, you get to talk to the community, yeah, and then you just build the model based on their demands. That's the only way it works. But in principle, it can be adapted, yeah.
Great. Thank you very much.
Sure. Thank you.
Our last speaker today, our last speaker for this session is Trewa Ekwama Boateng. Are you presenting a docudrama? Okay, so please let's clap while she comes. She's presenting a docudrama. Yeah. a docudrama for strengthening hearing impairment genetics research. So, Chiriwa, you have 12 minutes.
Okay. Thank you very much, everyone. I'm Chiriwa Kyomoboti, the Community and Public Engagement Officer for WACCBIP. Okay, so I'll start with a story with a community that we have been engaging in the eastern region of Ghana. So it's called Adam Robe, which about 2,000 inhabitants, about 50 of them are deaf. And engaging them, the explanation that some of them give is that there could be, there's a deaf god in the community who, if you don't follow his rules or regulations, the spirit of deafness can come upon you. So, and these are some stories that we've been hearing among communities, and a lot in most of the communities in Ghana. So today I'm here to present to you one of our interventions that we have created at WACCBIP. Yeah, thank you. So it's a docudrama for strengthening genetics of hearing impairment. Okay, so a brief background. So at WACCBIP, one of our main research focus is on disease pathogenesis and immunity, and also pathogen genomics and human genomic research, which includes hearing impairment. And in Ghana, the 2010 census actually came out that there are over 110,625 persons who are deaf. And at WACCBIP here, we have also come up with a lot of research on hearing impairment, that we've identified a significant mutant, which is the GJB2PR143W founder, which is very significant in the Ghanaian population. And we also realized that in communities that we have been engaged in, it's very difficult to explain to them words like genomics, hearing impairment, cells. These are very big scientific words that most communities or even the lay person walking on the Ghanaian streets will find difficult in understanding. So how do we explain the deafness or the genetics of deafness to deaf persons, their communities, and then their families, and even to the Ghanaian population? How do we explain deafness in simple languages? And as I previously said, there are lots of existing cultural beliefs in Ghana, perceptions, myths about the condition. And especially our researchers also find it very difficult in recruiting persons, taking of blood samples. We know the stereotypes that is usually associated with taking of blood samples for research. Yeah. So we after trying all these things, we realized that there is also no single public engagement toolkit in the country for aiding genetic research. And we also wanted to bring in the perspective of deaf persons in the implementation of scientific toolkits or science communication toolkits for better understanding. Okay. So we came up with different engagement approaches, different engagements or scientific or community engagement approaches to come up with a solution for this problem. Okay, so these are some of the pictures, and then it's actually identifies some of the issues I've earlier mentioned before. Okay. Okay, so in order to make this a reality, we had the idea in mind, but we needed to engage the persons who actually understand how engagement with communities, and also the deaf persons themselves we needed to bring them together in a workshop which we know is very participatory and very engaging. So we came together with science engagement specialists. We also had the Ghana National Association for the Deaf, and we also had some of our research scientists, genetic researchers, to participate in a workshop which we aim to develop a toolkit to engage persons who are hearing impaired. So at the end of the workshop, this is a picture of some of you can see, some of you are here. And Claudia, Elvis, yeah, okay. So, and the person in the middle doing the translation is one of the persons from the Ghana National Association for the Deaf, who is also a translator. Okay, so the purpose was to actually engage these three important people, the researchers, deaf persons, and then actors. We had actors from the Groma dance or theater group who came in with a script that at the end of the workshop, a script was written from the stories that we have heard from the deaf communities that we've been engaged in. So these are some of the key terms and then key important discussions that came. So we wanted to be very focused as much as possible. We had issues on genetic, what we face as scientists on the field, and we also wanted our focus to be on a docudrama. We had key terms and concepts explained, how the duration of the docudrama should be, and then factors to consider for engagement. Okay. So the key outcome for the workshop was the need to effectively communicate what we do as research scientists, for our communities to understand what we do, and for them to know the approaches and the reasons why we do, or come to the communities for sample, for field work, and then other activities. Okay. So at the workshop too, we had words like DNA, chromosomes, genes, proteins, and some of these words explained into simpler languages for our communities to understand. So the conclusion was to get a docudrama or a documentary drama which can dramatize the whole information. It will be very educative in a friendly manner, which will be culturally accepted and can easily be identified with the communities that we engage with. And it will also be an effective tool in passing on information to our targeted audience. So we finally came up with the drama, and in the drama we have some real life stories that are being shared by the communities or the persons that we have been engaging, the difficulties that they have been facing in the communities, and also the stigma and the perceptions all surrounding giving of blood for samples being demystified. So the toolkit is 25 minutes. It's a science communication toolkit produced by WACCBIP, and is the first of its kind in Ghana, or let me say in Africa, only by WACCBIP. And it's very interactive, very engaging. We have one version for persons who are deaf, and there is a sign language interpretation of it. And we have one for we, the hearing people. And with further consultation and broader understanding, the feedback that we got from the committees was to translate or make it, to localize the docudrama into 5 different languages. So we have one in Akan, one in Ewe, one in Dagbani, one in Frafra, and then the other one in Ga, so that people can identify it with the different tribal and ethnic languages that we have in the country. Hopefully we might translate it into Swahili. Okay, so after we produced the drama, we needed to pilot test it and to make it to get feedback from the people that we have co-created these things with, or these docudramas with. So we did a pilot testing in the northern and southern parts of Ghana in all the languages that we have been able to translate the drama into. And in the picture, it was in Tamale, where we had Professor Godnawandari joining us for the pilot testing in Tamale. It was very interactive, very open, and we wanted to hear the feedback from the people who assisted us in creating the drama. And after that, we organized a capacity building training workshop for our scientists who will be using this docudrama on the field. In fact, the docudrama is for us here to use on the field anytime we are engaging the community. So we needed to be trained and also get to know what the practical ways that we can use the drama, and also to basically understand what the communities understand and what they think should be added to make this very suitable. Okay, so after the training workshop and then the pilot testing, even during the pilot testing, we have been able to engage persons across the country in the northern, upper east, and all these regions have been covered. And mind you, there are deaf persons in every community that we find ourselves in. So these are some of the pictures to make it very, okay, so these are some of the engagement activities that we have so far been doing across the country. Okay, and we have also visited deaf schools in all the regions, and we are still engaging with them to get a final feedback from them. Okay, so our perspective, or as scientists, I know mine is not that scientific, but it's very appreciative, and looking at the communities that we engage with here, we believe that the toolkits or the docudrama that we have produced will strengthen and share best practices for public engagement and for better science engagement with their families and their communities to understand what we do as research scientists in our various labs. And also, as scientists, what we have learned from the drama is to be very adaptive, be very, we are in a multicultural society like Ghana. Scientists need to consider the different culture and customs during community and public engagement activities. And with this drama, we believe that superstitions, myths about deafness, and a whole lot of superstitions around giving of blood samples and genetic investigation will be demystified with this. Yeah. Okay, so we have gone through a lot of phases with the creation of the drama. It's been through a lot of phases, which makes it very acceptable. We are still engaging the communities, we are still engaging the deaf schools across the country as we are implementing the drama. And hopefully, by the end of this year, we'll finish with everything. But though the drama is still there and can be used for any engagement activities by any of us here. Thank you very much. So I would want to acknowledge Professor Godnawandari, who is the main PI of the project, and also the WACCBIP team, and anyone or any person who has been involved in the success of the drama. So we will show just 10 minutes of it, and that will be after the presentation, and then the rest will be played during the sponsorship session. Thank you very much.
Yeah, thank you very much. Are you showing that now?
Yes.
Okay, so maybe a quick comment just before that goes in. I just want to congratulate you. I just want to congratulate you on presenting this work, and a comment you made about maybe our work being very scientific and yours being not so scientific. But I just want you to know that this is the next stage of our science. When we do our science in the lab and we do our research, and especially the research that must make an impact to the society, this is where you play a very big role. And I believe that maybe other groups or other research groups should be talking to you, and because you have the training, and they should find out how the science can be more impactful. Because you've taken the research and you have translated it and brought it out there to the people who it makes an impact with, and I think it makes people understand more what we do in the lab. So I just want to congratulate you once again.
Thank you.
Thank you.
Hello, how are you doing today? Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, it's not painful. Blood. Okay.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patients that we need a sample of your blood for the lab.
No. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that. Look, please, we are here.
Oh, good. I hope you are well. Don't worry, we are here to help you, all of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay? For our diagnosis, you see, please, it's okay, take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. You take her back to the ward. You'll be fine, okay? You need a sign language interpreter.
looking at them. Who told you I'm interested? I can see it in your eyes. See. Doris, they are deaf and dumb people. It's true. Ah, I hear these people are friends though. How do you even introduce them as your friends?
You know, every physical condition has its medical cause. It has nothing to do with curses.
Oh, hello, sir. Your money?
Oh, I see. You are not deaf?
No, I'm not deaf.
That is interesting.
Yes.
Aha. Oh, okay. I will need your assistance to communicate with a deaf patient.
Oh, okay. Which hospital?
Legon Hospital.
Wow. Actually, I've just been transferred from the Alpha Clinic to the X-ray department.
Really?
Yes.
Wonderful. That makes my work easier.
I think I can help.
Yes.
Okay.
Please.
Okay.
This boy, then we cases for this.
Let me see how far.
You!
Hey! What is this?
Masha! Masha!
What is it?
Warn your son! Warn your son! Or else you will see what you don't want to see and hear what you don't want to hear. How many times have I not warned him not to play with my son?
Oh.
Or come to my house?
Oh, Ejapia! What's wrong? If Kofi plays with your son, are we not from the same clan?
Hey! Don't you know there is a case on you and your husband?
Me and my husband?
Yes!
Me and my husband?
Yes!
Very good. My husband is in.
Did you
say
case?
All right. We'll see who that case is on. Deal with him! But shipping, come on. What?
Doctor, we can't find Amma.
Where is she?
I don't know, we've looked everywhere.
What? Yes. In that critical condition? Oh God. This is unbelievable. We have to find her. We have to find her at all costs. I'll handle it. My friend, thank you very much for coming. I'll call you when she turns up.
I'll shout. What more tests do you need? We know the people who did all the antenatal tests. After you tell us our child is deaf, we will solve our own problem. Let's go.
Excuse me, sir. Is there any problem?
What I say is final. We are not going to any pastor. We are going straight to the village.
I am not ready to take my daughter to the shrine.
No one in my family is deaf. And your family too, there is none. So how can you tell me my daughter is deaf? We must look into it. This is purely spiritual.
What about going to do the test? So the doctor can determine how to help her.
It is my daughter who is deaf. What do they need my blood for? Blood.
I think that is what will help him.
Blood is sacred. I am not giving my blood to any doctor. I will call Papa at the village to arrange a meeting with the diviner.
Thank you.
Oh, great diviner, eye of the gods, the great owl whose gaze pierces through the thickest shadow and the darkest night. There is a deity that must be appeased in your family. She has been abandoned and has come with the vengeance of deafness. You must pay the due and purge your child. 3 rams and 3,000 Ghana cedis.
Your Highness, can we pay in cash?
Oh, the gods can take cash, even mobile money.
Hey! Something is happening to her.
Don't worry, my daughter. The spirit is leaving her. Mali, Mali, Mali.
He said she was going to get better when we get home. But now, her condition is worsening. Please, please help us, please. This is highly demonic. She needs deliverance. I can feel it in me that there's a bad spirit inside this child.
Prophetess, can you deliver her?
That is exactly what I am going to do. Let us bow before the Lord. Jesus, we trample on every serpent and scorpion. According to the word, we rebuke every demonic power of high places, every altar that stands against the authorities of Jehovah. Be brought low. Jesus' mighty name. Jesus' name. Jesus' name. Oh, Father. Jesus. Jesus' name. Koyo, she's not getting better.
What should we do now?
Let's go back and see the doctor.
Okay.
Let's go.
Doctor, thank you very much for the explanation. We got it all wrong. We now have a better understanding of what genetics is and its implications on deafness.
How can we thank you, doctor? Many people out there are not privy to this information.
Especially the people of our village. Doctor, there is a deaf community in our village. Is it possible for you and your team to come and explain this to the people?
Wow, that's a very good idea. It's an opportunity to reach out to the community and educate them. We will. Why not?
Thank you very much, Doctor. then I'll quickly arrange for the meeting.
Thank you. Thank you. Thank you. She's as normal as any other child. Just take good care of her. Okay? It's okay. Don't need to cry.
Ago! Nana and the council request tomorrow morning. Nana wants to meet everybody at the village square. The great Asparrow has spoken. Ago, people of Asempa, Nana and the council request your presence. Nana wants to meet everybody at the village square. The great Asparrow has spoken.
Nana and Council welcome you all to this urgent but very important meeting. Nana want to cut straight to the point. Nana, speak.
People of Asempa, the issue confronting us is a very serious one which needs to be addressed. I'm not an expert myself, but our forebears have told us over the decades the mysteries surrounding deafness in our community. Much as we have tried, we've not been able to find answers to this problem. But today, our son Kojo and daughter Agnes are here with some medical officers from Legon to educate us on the subject of deafness and its possible causes. Dr. Wo and Dr. James, you are most welcome to this community.
Thank you very much, Nana.
Please, you have the floor now.
Thank you very much, Nana.
As part of our work as clinicians and researchers, we investigate what makes each person unique and different. We do this by examining your blood sample. How many of you here have ever given their blood for testing?
Blood. I can't give my family.
Blood.
My family.
No, blood is sacred. We don't give blood. Yes.
It will never happen like that. I can't give my blood. It is very important to give blood to be tested because every information that is used in building our body is packaged as genes, and this can be isolated from our cells in our blood sample. This we inherit from our parents. Please, can you bring the chart? You have to look on this chart. It will help you understand it better. Please. Our bodies are made up of billions of cells. Inside each cell is our genetic material. Chromosomes are the packaging of our genetic material. We usually have 23 pairs of chromosomes, and we inherit one copy from a mother and one from a father. If we unwind the chromosomes, we can identify the genes. Genes are the basic building blocks that makes proteins. Genes are like the blueprints for making a machine, and proteins are the individual components of the machine. Proteins makes us who we are and help us grow and function properly. Our genetic materials is made up of our DNA. Deoxyribonucleic acid is the very long chain of 4 chemical letters: adenine, guanine, thymine, and cytosine that combine to make our genetic code. We all have some variations in our genetic code. Genes variations are like spelling errors, where for example there's a T instead of a C. A variation in the gene that makes it faulty or not working properly is called mutation. Faulty genes are caused commonly simply through chance. They have to occur in one individual for the first time at some stage. They are not related to anything the parents could have caused or prevented in any way.
They occur because the process that replicates and divides their genes so we can pass it on to the next generation is complex, and mistakes occur. For each gene, we have 2 copies. One copy comes from the mother and the other copy from the father. In an autosomal recessive condition, an individual needs both copies of genes to be faulty for a condition to develop. The parents of a child usually carry one faulty gene of the gene pair. They are healthy carriers. It is estimated that every individual is a healthy carrier of several faulty genes, but a recessive condition only occurs when both individuals in a couple happen to carry the same faulty genes. When healthy carriers have children, there are 3 possible outcomes. 1. The child inherits both faulty gene copies and has the condition. 2. The child inherits only one faulty gene copy and is a healthy carrier like his or her parent. 3. The child inherits both working copies of the gene. So each time healthy carriers have children, there is a 1 in 4, 25% chance that their children could inherit both faulty gene copies and develop the condition. When an individual with a recessive condition have children, their children will usually all be healthy carriers of the condition. Their children could develop the condition if their partner happens to be a carrier of the faulty gene, but this could be a rare occurrence. Brothers and sisters of a healthy carrier could also be healthy carriers themselves.
So as you can see, features such as our height, complexion, color, including defects such as sickle cell and deafness, are all inherited from our parents. These traits are controlled by our genes. Have you noticed that every family has a feature that is passed on from generation to generation?
That is why our elders say the crab does not give birth to a bird. But I want to ask a question. Are you saying that deafness is not caused by curses or witches?
My elders and people of Asempa, that was what I believed until I could not find spiritual remedy for my daughter's deafness. That was why I gave in to genetic investigation.
I am glad this has come up. Please, from today, no one should associate deafness to witches or curses. As we have explained, every defective condition is as a result of our genetic inheritance. Although there are environmental factors.
People like Mr. Akoto and his family, and like my daughter, are all normal human beings. Their deafness can be explained through scientific investigation.
No, please, let us put it on record that with the advancement of science and technology, every medical condition can be scientifically examined, including deafness. So please, I will kindly entreat all of you to stop attributing deafness to spiritual causes.
Doctor, please. Some of our neighbors and the entire family are deaf. Does it mean that they spread it to one another? That is why we want Nana to send them out from our community.
No. Please, be rest assured. Deafness is not contagious.
Hey, thank God!
Thank you. It's okay, woman. It's okay, woman. Please sit down. It's okay. Let them finish what they are saying.
Nana, all we need to do is to avail ourselves to be examined. It is possible some can be helped, but the most important thing is getting advice through genetic counseling and also having new babies screened for early detection. That's all we have to do.
Yo, my people, wisdom, they say, is like a baobab tree, and no one person can embrace it all alone. Today, we have been educated that the deaf people in our midst are our brothers and sisters. They are not under any curse. They are humans just like us. There is no curse on them. So we have to show them love and respect. And more importantly, we have to make ourselves available for genetic investigation.
Thank you very much. So the drama is still in progress in perfection. So if you have any concern or any input to finalize on it, you can send it any email address on the website. We can work on it. Thank you very much.
Thank you very much for that. So you have a skill which all other scientists have come to you for. I remember we did genetics in level 200. I don't lead you. I remember, were you in Prof. Booker's class?
Yes.
And we learned about Huntington's chorea. Yeah, and it was implicated in the Salem witch trials in early 17th century. Yeah, because people didn't understand that it was a genetic disease, and they were burnt at stake and accused of witchcraft. So, yes, you are doing just the right thing, Cheriwa and your team. Thank you once again.
Thank you. Thank you to our moderators, Prof. Kwashi and Dr. Ametefe, for handling this session so well. So lunch is on course for 1 p.m. Now we have a Turbo Talk session real quick, and then we'll have some sponsor adverts, which these are from the companies that helped us run to this year's edition of the research conference. So Turbo Talk, please, if you're ready. Let's go. Okay, there's one which is recorded, and then there's one in person. So please, Bill. Bill, if you
Hello,
can.
everyone. I really appreciate the attention you are giving me to share with you the exciting research work that I did during my PhD journey. My name is Eric Wala Wigaba. I'm sharing with you the output of my research work on how pineapple leaf fiber can be used for prosthetic application. Everyone here is just a step away from disability, considering the fact that road traffic accidents have been on an increase in recent times, coupled with diseases such as diabetes. But the issue still remains the same, that how is the space or the sector of disability rehabilitation ready to actually embrace and take care of persons with lower limb amputation. So currently in the field of prosthetic rehabilitation, the materials for designing prostheses are mainly imported, hence making the cost of acquiring a functional prosthesis very high. And in effect, people who are financially constrained are not able to have access to this. So the goal is to find alternative materials that can be used, and in my investigation I considered how pineapple leaf fiber can be used to design prosthetic devices. So having extracted the fiber, I subjected it to various tests such as XRD to confirm the crystal structure of the fiber. I did a tensile test also, and then I also performed some modelings to know how this can be used for the application. So in effect, I was able to identify how pineapple leaf fiber can be processed and used for prosthetic socket reinforcement. Thank you very much.
Bill, you can come now. Thank you.
Good afternoon, everyone. Is it up? Do I control the thing from here to get the slide up? I have a slide. Okay. There we go. Okay, so I'm going to talk about the PEER multigene family. It's transcriptional and protein analysis in a rodent model that we use in the Francis Crick Institute in the UK. This is work done by current and past members of Dr. Jean Langhorne's lab, somebody that a lot of you are probably familiar with. I'm going to spell it out to you basically because I've been told, even in the UK, that a lot of people struggle with my East End Cockney accent, so it's going to be spelled out. So this is a very large family, and it contrasts with the VAR multigene family that's seen in Plasmodium falciparum, which is, as you probably know, very important for parasite virulence and for immune evasion, but it's exclusive to falciparum. The other largest, and by a long way the largest multigene family, is the peers, and it ranges in numbers from about 100 to several hundred in rodent parasites and about 350 in Plasmodium vivax. And the jury's still out on whether the stevors and the riffins can be included in this family in Plasmodium falciparum. But the functions of the peer genes, the peer proteins, is not known. Now, what we've done is we've looked at transcription of this gene family in the limited way that we can in our rodent model, and we can see that it's differentially transcribed throughout the intraerythrocytic developmental cycle. So if you look along the bottom here, these are the various stages through the development in the blood cells, from very early rings through to mature chizons, which we have to culture in order to get them because they sequester. And then along the side here, we've got members of the various subfamilies that are representative of the acute and chronic phases of the infection in mice. And you can see that there's very clear and quite differential transcription of peer genes depending on the different points of the cycle in the blood. So what we've also gone on to do is to look at gametocytes and the expression of these genes in gametocytes. And I know you can't read the detail of this little panel here, but all you really need to do is look at the colors, and you can see quite clearly that there are definitely peer genes, a small number of peer genes that are transcribed in both female gametocytes, male gametocytes, and asexuals, and they're shared between those different forms. Interestingly, there are also peers that are transcribed by males and females, but are not transcribed by males and asexuals, sorry, but are not transcribed in females. And then really interestingly, and absolutely fitting in with Dr. Dinko's work from earlier today, we can see a very clear set of peers that are transcribed only in female gametocytes. We've produced, I say we, I mean I'm just a conduit for the work done by past and current members of Jean's lab. I'm retired. The lab produced antipeptide antibodies with a pan-peer reactivity. And what we see in staining, although I'm not going to show you that today, but you can still see it on the poster if you wish to, is that these antibodies fail to stain peer proteins in live cell imaging. But they do locate the proteins in intracellularly. So if you fix and permeabilize the cell membranes, then the antibodies can get in and they clearly label peer proteins inside the cell. So given this, it seems unlikely that peers are involved in adhesion or of infected red blood cells or in rosetting. Clearly less involved in the pathology of the disease, although mature P. Shabbaudi parasites do sequester. And it's unlikely that if they're not expressed on the cell surface, that there's a need for antigenic variation and immune evasion of these proteins. So the question is what are they good for? And that brings me to some work that Phil Spence and I did, and some other members of the lab at that time, from 10, 12, 13 years ago, where we looked at parasites that were routinely maintained in the lab by serial blood transmission, so mouse to mouse, mouse to mouse every week. And they've been established that way for years, decades even. And recently mosquito-transmitted parasites. And as you can see, there's a clear difference in parasitemia. So along the bottom here, we've got days post-infection, up the side, percentage parasitemia. Serially blood transmitted parasites give a very acute primary peak of parasitemia and then go on to a chronic infection. Whereas in each of these mice, where we've looked at the mosquito transmission, post-mosquito transmission, you can see that the parasitemia is much less acute and much less virulent. And interestingly, when you look at the transcriptome between these two sets of animals, you can see that in serially blood transmitted parasites, sears are transcribed as a relatively minor proportion of the whole transcriptome, whereas when you look at mosquito transmitted parasites, there's a massive increase in sear transmission, in peer transmission. So, given that it's a multi-gene family, it's difficult to get hold of. It's difficult to get hold of VAR in terms of, and PFEMP1 in terms of thinking, well, can we vaccinate against it? Can you direct drugs against it? And that's a relatively small family compared with the peer family. But one way that we might be able to get to it is if we come back to this heat map from the transcriptome in asexual stages, is if you look at this very top line here, this is an ancestral gene, one member of the family that's transcribed right through the asexual developmental cycle. We don't at the moment know whether it's actually transcribed in gametocytes, but certainly it's there present at a relatively high level throughout the asexual cycle. And interestingly, it's highly represented across many parasite species, including all the rodent species and some of the human species. There are some gaps. Cynomolgi is missing, and Nosai is missing, but it is there in Vivax. And that means we've got a handle on it, and that means also that we can investigate it fully in this very tractable model of protein expression in malaria. Thank you.
All right. This is table talks time. So can I see by hand which students are presenting this afternoon? Please, if you are presenting a poster this afternoon, please put your hand up. Bill, you've done yours already. Okay, you stand up so I see you. If you are presenting a poster this afternoon, please stand up. I saw some hands at the back. Where are they? Who's presenting a poster this afternoon? Where are the others? They are not here? Pardon? They are online. Okay. Can you come forward? All those presenting posters this afternoon, come forward. All right, so you know what we call an elevator pitch? Eh? So you know there's a prize for best poster, right? You know that? Okay, there's a prize for best poster. And you need to get people to come and look at your poster and vote for you, right? So I'm giving each of you 1 minute to do your elevator pitch here. Oh, I see more people coming. Ah, okay. I see. Okay. When I talked about price, then people started to stand up. Okay, so you stand in this corner so that they can see. All of you stand in this corner. So each of you have 1 minute. So you just come one at a time. We're going to time you 1 minute. Tell us your name. Are you a master's, PhD student, whatever, and what your poster is about. 1 minute, right? Who's first? Okay, Dora wants to go first. So you come up here. No, you don't need the microphone. You have the podium for 1 minute.
Good afternoon, everyone. My name is Nganyewo Norengochuzi. I'm a third-year PhD student at WACCBIP, and my poster is on variations of Plasmodium falciparum erythrocyte invasion. Sorry, my poster is on variation in erythrocyte, Plasmodium falciparum erythrocyte invasion following short-term adaptation in culture. So we already know the erythrocytic stage of the malarial life cycle is an attractive site for research. My focus was to throw more light on this stage, and I had two questions. Do the invasion profile of P. falciparum change after long-term culture in culture, long-term in culture, and secondly, if you use the same blood group but different donor, is there any difference? And so I used flow cytometry where I got 3 different donors and then got samples from infected patients and use it to set invasion experiments without culturing, and then put it in culture for 2 cycles and 3 cycles to monitor the invasion profile. And then after that, I treated the erythrocyte blood donors with different enzymes to block the invasion wheels. So you can meet me outside for further explanation. Thank you.
Hi, I'm Emmanuel Asante. I'm a final year MPhil student at the Kwame Nkrumah University of Science and Technology. My work basically is on studies on biofilm formation from bacteria that were isolated from cutaneous leishmaniasis wounds. Cutaneous leishmaniasis is a problem in Ghana here, and it's endemic in the Volta region. One of the problems about the disease is the occurrence of secondary bacterial infections. This is because these secondary bacterial co-infections interferes with the treatment and also even in the diagnosis of the disease management. So our group focused on looking at the bacteria that are present in these wounds and how to see their diversity and resistance pattern. After that was done, we wanted to know, relate their resistance to see if the mechanism known as biofilm formation was responsible for that. So we did that, and we categorized and grouped them based on their level of biofilm formation and determined their resistance pattern. So this was what was done. Thank you very much.
Good morning. I'm John Kamzune Alexandra Lindsay, and today I'll give you the privilege to see the first preliminary study looking at DNA methylation profiling of prostate cancer in Africa. So, so we know prostate cancer is declining. The incidence is declining globally. However, in Africa it is rising, and mortality also is rising. The severity is different, so we want to look at the methylation profile. Why the methylation profile? We have different diets, we are exposed to different infections. We have heard about a lot of infections here. We know we have a lot of burden of infections here. How is our genome exposed to these infections affecting our methylation profiling and making us having severe prostate cancer in our communities? I'm waiting for you. Thank you.
Good afternoon, everyone. My name is Fatima, and my poster is on impact of SARS-CoV-2, impact of host genetic diversity on SARS-CoV-2 infection in Ghana. So my study really focused more on the host genetic diversity and how it affects severity or susceptibility to SARS-CoV-2. So my work looked at ACE2 and TMPRSS2, which are 2 receptors that are involved in infectivity or SARS-CoV-2 itself, and how I went on to do characterization of some variants that were found in African population and how they affect infectivity. So if you want to know more about my paper, please stop by at my poster 42. Thank you.
Good afternoon, everyone. My name is Sahid Abdul Karim. I'm an MPhil student at WACCBIP. My work basically is around breast cancer. We know that breast cancer is a highly diagnosed form of cancer, especially in Ghana. Triple negative breast cancer is worst form of breast cancer. And then we know that there are several other factors that are contributing to vasculogenic mimicry in triple negative breast cancer, such as secretion of interleukin-8 among the tumor cell, as well as epithelial to mesenchymal transition. So our work actually look at interaction between interleukin-8 secretion and EMT towards the formation of vasculogenic mimicry, especially in triple negative breast cancer. So when you come to poster 68, I will tell you more about this. Thank you.
Hello, good afternoon, everyone. We all know leishmania is part of, I'm Kwesi Ajenkwamaoli. And we all know leishmania is part of the neglected tropical diseases. And one issue that always arises is mass administration caused drug resistance. And this issue can be tackled from different fronts, but we're looking at it from the point of view of diagnostics. Effective diagnosis, it leads to effective treatment. So with the advent of machine learning in various applications, we are leveraging that approach in order to aid diagnosis of leishmania by training it on microscopy images. And we use two different approaches, the one-class approach and then binary class approach using convolutional neural networks. There were some very interesting insights, and I hope you pass by poster number 40 to know more about it. Thank you very much.
Good morning. I'm Ede Madika, an MPhil student at WACCBIP. So my work is focused on anti-malaria drug tolerance. We know of sensitive and resistant isolate, but we've left that gap, which I'm saying there are tolerant isolates in there. Again, we know host factors affect your response and treatment. And also when you adapt in vitro, there are also changes there. So I'm looking at ex vivo. What is the story? So I've phenotyped some clinical isolates from Ghana, and I've looked at their response, and I found some tolerant isolates, especially to ACT partner drugs. So you can pass by poster number 83 for more insights. Thank you.
Okay, I'm Francis Japon. But then I want to say something. When I came in yesterday morning, I was very early. Dr. Moses said everybody must wear nose masks. So even in our room here, nose mask is mandatory. And then in the emergence of COVID, we all realized that in order to reduce the severe rate on the economies and then reduce transmission, there was a mandatory wearing of nose masks everywhere implemented. But then the prolonged wearing of this nose mask accompanied with its own adverse reactions and side effects. And for that matter, poster number 74 investigated the side effect that comes with the wearing of nose masks. For that matter, I plead with you to come to poster 74 to come and see the various side effects that about 2,000 people, 2,000 respondents to come and see the various and different kinds of reactions that comes with the wearing of the nose mask. Thank you.
Hi, my name is Kwesi Tando, and I'm a PhD MCBI WACCBIP. Now, I'll be manning poster number 71, and I'll be talking about looking for a new druggable space in novel anti-malaria discovery. I have hypothesized that a molecular mechanistic understanding of artemisinin resistance may include a role for extracellular vesicles. And I use a novel data analysis pipeline to show that a model associated with EV's biogenesis is associated with artemisinin resistance. Come check out my poster number 71. I'm looking forward to a good discourse. Thank you.
Hi,
My name is Albert Asewa, a master's student with the Kwashi and Akins lab here at WACCBIP. My poster is sort of a preliminary study of my whole project, which kind of investigates the COVID-19 infection on cancer, which is one of the comorbidities that is related to COVID-19 severity. So visit poster number 73 to have further insight on the expression of the TMPRSS2 gene and the ACE2 gene to see how they interact with cancer cells and COVID-19, SARS-CoV-2. Thank you.
Hello everyone, my name is Amir Prince. I'm a level 400 biochemistry student, and my poster is about preparation of agarose here in Ghana using seaweeds. So we all know agarose is one of the most important reagents used by researchers, and then even though it's very important, it's not produced here in Ghana, and then it's not readily available. Therefore, if you want to use it, you'd have to import it, which comes with problems such as time of delivery and then even the cost involved. So we collected some seaweeds here in Ghana, and then we were able to extract agarose from it, which we ran a gel electrophoresis with and compared to what is commercially on the market. And then we realized that we can produce agarose here in Ghana, and therefore it will make research work very easy. Thank you. My poster number is poster number 24.
Good afternoon, everyone. I'm Issa Samori, and my work is on using random forest and molecular docking techniques to screen FDA-approved drugs that can be repurposed against dengue virus. So dengue virus is the causative organism of dengue hemorrhagic fever and dengue hemorrhagic fever. So these diseases kill millions of people worldwide every year, and there's no potent therapeutic against dengue virus. On the other hand, machine learning has proven to be an effective computational drug discovery tool. So we seek out to apply machine learning to dengue drug discovery. So we built high-performing machine learning models which we use to screen a library of FDA-approved compounds. So we screened 165 compounds, and then with the machine learning, we were able to shortlist 36 compounds. We further used molecular docking to further screen the 36 shortlisted compounds to shortlist 6 final compounds. So we believe these 6 FDA drugs can be repurposed as anti-dengue drugs. And also, the best performing model has been deployed as a web app called Dengue ML, which is openly available for everyone to use. So my poster is poster number 61. I'm looking forward to seeing you there.
Hi, I'm Anissa Abdullahi, a first-year PhD student of the Department of Medical Microbiology. So my work is on insecticide resistance status of Aedes mosquitoes in urban and suburban areas. So we all know Aedes mosquitoes are vectors of arboviral diseases like yellow fever and dengue fever, and they are very abundant in Ghana here. I'm sure we all have been bitten by these black and white mosquitoes. However, not much is done about their control. So my work sought out to investigate their susceptibility to the various insecticides that are used for public health and then the mechanisms that are mediating this resistance among our population. My poster is poster number 58. Please check out my poster. Thank you.
Hi, my name is Klein Owari. I'm a research graduate assistant at WACCBIP here, and my poster is on using machine learning for repurposing drugs against Schistosoma mansoni. So what my team did was to target potential compounds that would inhibit the receptor thioredoxin peroxidase, PRX2, against this known disease. So the issue is praziquantel, which is a known drug to treat schistosomiasis, has been used frequently, and due to mass administration of this drug, there's been reports of resistance. So there is a need for alternative therapeutics for this disease. So my team decided to search it using these data sets, and then we're able to come up with 4 machine learning models to predict these inhibitors. So my poster is number 51. I hope to see you all there. Thank you.
Good afternoon, everyone. I'm Christian Tungiobama. I'm a PhD student at the Oshulam Midvida. And we've all heard so far about different variants of the COVID-19, being it Omicron and so on. And those different variants were actually discovered by those sequencing techniques that all of you have the mastery. But I'm particularly a mathematician. So from my point of view, what I'm interested in is data. How can I use the data that you are producing and eventually help in the discovery of those different variants, at least understanding how they are spreading in the population and so on. So my work is on developing a statistical model that will help estimate the haplotype frequencies as well as their prevalence alongside MY, because here we are looking at the case of multiple infections as well. And our model is kind of targeting malaria, but it's not limited to malaria. So I invite you to poster 76 to look at how we did that. Thank you.
My name is Sandra Sampomasaki, and I'm a research assistant at the Department of Medical Microbiology, University of Ghana Medical School. So my work is on how green tea kombucha how green tea kombucha protects against smearing colitis by averting oxidative damage. So green tea, ulcerative colitis is actually an inflammatory bowel disease which is characterized by oxidative stress and intestinal barrier dysfunction. So generally we use 5-amino salicylates to cure and manage ulcerative colitis. However, they are known to have side effects. So we sought to use natural products such as green tea kombucha. And green tea kombucha is a fermented tea which has significant antioxidant activity. So we assess colon weight to length ratio. We also assess tissue histology. So please pass by poster number 31 for more insights. Thank you.
Good day. I'm Peter Okuchu. I'm a research assistant and a graduate intern here at WACCBIP, and I'm with the Protein Expression Lab. The work we did was to kind of try to see if we can be able to find other vaccine candidates that can be used in efforts at developing a vaccine against Plasmodium malaria. So far, all the efforts in vaccine design has been based on Plasmodium falciparum and Viva. So that was the kind of work we sought to do. And we found an interesting vaccine candidate, that is Plasmodium reticulocide binding protein A1. And in our efforts, the main objective was to try to see if we can find a subclass that is most a subclass that is most released when that vaccine candidate is in contact with blood serum of immunoprevalent patients or people who were exposed to the disease. So please pass by poster number 80 to see the results we had. Thank you.
Good afternoon. My name is Christopher Uswasenso, a PhD student in the Medical Microbiology Department. With the recent outbreaks of yellow fever in Ghana and also recurrent outbreaks of dengue fever and yellow fever within the surrounding countries, my study sought out to look at the bionomics and also the insecticide resistance status of the vectors in the country. So pass by poster number 58 to learn more about the results. Thank you.
Good afternoon. My name is Constance Agbemelochomafu.
I'm a PhD year 4 student in WACCBIP, and my research is on avian malaria parasites in Ghana. So, avian malaria is caused by 3 different genera of parasites, protozoan parasites, and these are Plasmodium, Hemoproteus, and Leukocytosis. And interestingly, Plasmodium parasites in birds are transmitted by all species of mosquito, not only Anopheles. So in my work, we have found out that there is a high prevalence in our poultry birds and then the wild birds. And this is of veterinary importance and of wildlife importance to us. So you want to pass by poster number 64 to get more answers. Thank you.
Hi, my name is Christine Chamo, and I'm presenting on poster number 28, titled Anti-Leishmania Effect, a mode of action of phenolic compounds and glutathione combinations on Leishmania donovani. So the main aim of the study is to be able to identify alternative therapy via combination treatment against the parasites. So please pass by poster number 28 for more info. Thank you.
Hi, I'm Felice James from Department of Molecular Biology and Biotechnology, UCC. Yeah, my research was about aging. You know, aging remains the single largest risk factor for cancer and neurological diseases. So Caenorhabditis elegans was used. It's a human model organism. Its sequence has been completely sequenced. Its genome has been completely sequenced. This organism was used on old gene leaf. So our noble findings could initiate anti-aging drug for treatment of aging-related diseases. Thank you. My poster number is 61.
Good afternoon, everyone. I'm Nesma Adil from Sudan, and I'm doing a PhD in Hochschule Mittweida, Germany, and I'm an applied mathematician. So first, thank you, WACCBIP, for allowing me this opportunity and kind of impose my poster. This is my first time in Ghana. I hope they won't make it the last. So it's a COVID-19 mathematical model, and we just want to answer your curiosity. So we included the age groups, the different vaccines like AstraZeneca, Moderna, Pfizer, Johnson & Johnson. We included the different viral variants, the wild type, the alpha, the delta. So you just come ask your questions and I'll be happy to answer. I'm also asking everyone I had beer with to come see my poster. I'm watching you. And I'm on the second floor, poster 86. I'll be happy to have you. So thank you.
Good afternoon, everyone. My name is Dosu Daniel. I'm working under Dr. Aniwe as research assistant. My work is on evaluating of susceptibility parasites strain against anti-malaria drugs. So the work has already been published by Professor Wandari and his group. And I'm working. My work is to proceed and check gene expression across time. My poster number is poster number 32, so if you need more explanation, you can come to the poster. I explain to you. Thank you.
I'm not sure whether dropping my name is a kind of a way to get ahead, but anyway. So let's give them a round of applause. Thank you. All right, so we want to appreciate all our students for taking the time to make posters under pressure because they were informed about their status just a few days to the conference, and they had to produce posters and get them printed. So We appreciate your effort. That's why we gave you the 1 minute to make your pitch. So good luck to all of you in winning the prize. Thank you.
So before we break for lunch, we have a couple of messages from our sponsors. So Excelsior, we have Ofosu Asante. Come do your presentation. Thanks.
Good afternoon. I'm Ofosu Asante from Excelsior Global. I want to say we are very happy to be part of this research conference this year. And I want to say thank you to WACCBIP for giving us the opportunity to be part. Okay, so Excelsior Global is a global procurement and supply chain management company. Basically, our job is to shop around for your research needs, i.e. your equipment, reagents, or anything you need for your research. So our job is to move around, shop globally for anything you need to make your research successful. Okay, so yes, basically that's what Excelsior Global stands for. We are here to do the work, so you have time to focus on your research. You know, it's kind of tiring moving around looking for equipment and other stuff for research. I know it's very tiring. So we are here to do all the work for you and drop everything at your doorstep. So our services, procurement, logistics. So we take care of sourcing the products from suppliers. What we do is we look for the best suppliers. Yes, we look out for the best suppliers with a high standard. You know, I believe it's the aim of every researcher to improve diagnosis. So what we do is we look out for the best equipment to help you in your work. So we take care of procurement, logistics, strategic solutions, finance solutions, outsource solutions, sustainable sourcing. Yes. So why choose Excelsior Global? Total cost reduction. We take care of the negotiation. We want you to focus on what you do, your research. So we take care of negotiating, getting you a good price. You know, bespoke needs, the standard. We always look out for the high standard. Because you see, if standards are low, it affects research work. So we always take care of getting you the good standard from the very good suppliers globally. The process is very simple, not cumbersome, very, very simple. The lead time is also, I know, we work with WACCBIP sometimes, and the pressure is always on us, you know, so we try to limit the lead time, and daily and ad hoc procurement, exceptional support. You know, when we deliver, anytime you need our support, we make sure we come over, we get the experts to also support you. Actually, we work across different industries. We look at the medical, printing, and the printing industry, energy, agriculture, and mining, and also providing PPEs. So when it comes to medicals, we look at providing sourcing for devices, instruments, test kits, analyzers, PPEs, reagents, you know, anything you need for your research. So we take care of all these things. Yes, these are some of the products we source around the globe for some of our clients. Yes, the PCR analyzers, the diagnostic lab equipment, the clinical chemistry reagents. So anything you need to make your research successful and easier so you focus on your research, we are there to provide. You just contact us. We do that shopping around and we provide them for you. So we are linked with some top brands, Prestige Diagnostics UK. Yes, actually we are the sole distributors in Ghana. Yes, we also linked to Oxygen. So these are the top brands. For us, we don't compromise on standards. So these are some of the equipment we source for the pipettes, the microtubes, the PCR consumables, and other stuff. So anything you need in your research labs, we provide them for you. Yes. These are some of our major clients. We do ECG, provide them with power generation materials, University of Cape Coast, University of Ghana, WACCBIP, and Confanachi Teaching Hospital, and NEDCO too. So for our work, it cuts across industries. But for the purposes of this research conference, we are focusing on providing research centers and researchers with materials to help them improve their research. Yeah, so you can contact us. Our office is at Patrice Lumumba Street, Volta Place, Airport Residential. This is our number, our email address is there, our website. You can also go to our website and also read more about us. So you can also check Prestige. They have a whole catalog there. Everything, diagnosis, anything that helps you in research is there. So you can also check their website. And if you need anything from them, just contact us. We are the sole distributors in Ghana. So you can also check their website. Thank you very much.
It's been a marathon session, right? Thank you so much for staying through. It's lunchtime. As usual, please go upstairs, first and second floor, get seated, and your table will be called for lunch. Now, right after lunch, we have our poster session. Please utilize the time to make connections, go and visit the posters, and interact with the students who have worked so hard to put the information on those posters. Thank you, and we'll see you back here at 3 o'clock sharp.
In the lab, you face consistent pressure to do more with less while trying to keep up with a growing workload. Success means having instruments that you can rely on to overcome diverse challenges. The Agilent 6470B Triple Quadrupole LCMS, the latest version of our trusted workhorse, gives you the robustness and versatility you need to overcome these challenges. The 6470B has a proven reputation for robustness with non-stop operation, backed up by thousands of users with numerous application notes and peer-reviewed articles to reduce downtime and increase efficiency. Enhance versatility with a wide array of ion sources to analyze any application. Ion source options include ESI, the standard choice to broadly ionize analytes of interest, AJS, which enhances the desolvation power of standard ESI for ultra-high sensitivity, APCI, for increased signal-to-noise for very small analytes, and MMI, to carry out ESI and APCI simultaneously for a complementary analysis. With the new WACCShield, you can reduce maintenance time by 92%. WACCShield lets you remove the capillary and run the cleaning cycle in approximately 30 minutes, compared to about 6 hours for maintenance by venting and disassembly. So you can gain back valuable lab time and keep up with high demands. From analyzing environmental contaminants and ensuring food safety to complex life science research, the 6470B meets your need for reliable, accurate results. These accurate results are provided by our Mass Hunter software, including Quant My Way, which enables you to personalize the software with tools that highlight only the capabilities of interest. With comprehensive end-to-end workflow solutions and application services, you can ensure proper sample preparation, expertly developed acquisition and data analysis protocols, and fully customizable reports to get the answers you need as soon as possible. Our solutions are designed to work together to provide efficiency gains that help you get more done and reduce operation costs. The Agilent 6470B Triple Quadrupole LCMS is unbelievably robust to increase your lab efficiency and remarkably versatile to keep up with your evolving demands. Visit agilent.com/chem/6470B for more information. Decades of innovation have brought us into a new era of mass spectrometry with the design of Altivo, Agilent's revolutionary triple quadrupole LCMS. At just 1/3 the size of our previous triple quad LCMS instruments, Altivo integrates seamlessly into the stack of the Agilent Infinity Lab LC System series, saving bench space while offering the same power and accuracy of our larger systems. The footprint of Altivo is 70% smaller than that of similar LC triple quadrupole mass spectrometers, so you can triple your lab's capacity in the same space. The efficiency-optimized Agilent Infinity Lab LC series system can sit directly on top of Altivo, making it the perfect partner for high-throughput mass spectrometry. The innovations that enabled Altivo were specifically designed to maximize quantitative performance, enhance instrument reliability and robustness, and promote greater uptime. The more ions you get into the instrument, the better your MS results. With Agilent's Jetstream ion source, a superheated nitrogen sheathed gas surrounds the electrospray to quickly and efficiently desolvate the spray plume and tightly cluster the resulting ion cloud, maximizing the number of ions that are created and transported into the LCMS. WACCBIP features fast ion injector exchange without the need for system venting and improved diagnostics that guide the operator to appropriate corrective action, leading to less downtime for maintenance and higher throughput for your laboratory. Once the ions enter Altivo, they pass through our innovative precision-manufactured Cyclone Ion Guide. With its twisted and tapered dual hexapole configuration, the Cyclone Ion Guide provides maximum ion capture, compression, and transmission across a large pressure drop region, giving you better, more reproducible results. From the Cyclone Ion Guide, the ions continue to the first quadrupole. Better mass transmission improves analytical results. Each quadrupole assembly includes DC-only, wafer-thin pre- and post-filters, which cancel fringe field regions at the quadrupole entrance and exit to improve ion transmission. This allows for faster switching, enabling more MRMs to be performed. With the analyte selected in Q1, the ions pass through the vortex collision cell. Its innovative twisted and tapered dodecapole hexapole geometry maximizes ion collection, fragmentation, and transmission. The high-efficiency detector offers a wide dynamic range to ensure linear responses for your most challenging analyses. The detector was designed to be easily replaced by anyone in the lab. Working together, the compression and focusing of the Cyclone Ion Guide, the improved ion transmission through Q1 with its pre- and post-filters, the optimized fragmentation in the vortex collision cell, the improved transmission through Q2, and precise data capture of our innovative detector technology give you better MS results. Altivo uses an entirely redesigned electronics architecture, making it the smartest LCMS on the market. New intelligent diagnostics tools enable Altivo to monitor several parameters in real time to ensure that it's operating at peak performance. If it isn't, it lets you know so that you can take appropriate action. For food safety labs, Altivo offers the selectivity and sensitivity needed to confidently monitor for and identify contaminants, even in difficult matrices, helping keep the food supply safe. For environmental analysis, Altivo provides robust and accurate measurements down to sub-part per billion levels and integrates with Mass Hunter software for powerful data collection, processing, and reporting to help you keep up with newly emerging pollutants. Reshape expectations, reinvent capabilities, and redefine what's possible when small meets powerful. The Agilent Altivo Triple Quadrupole Mass Spectrometer.
There is no promise we cannot keep. There is no commitment we cannot honor. There is no delivery we cannot guarantee. There is only one Bethel Logistics Company Limited, a licensed international freight forwarding and clearing agent, a trusted global brand with an extensive network for the movement of air freight and ocean freight worldwide. We are the trusted name for road freight into Western and Northern Africa. We are fully accredited by Ghana Customs and Excise. We cater for all your freight forwarding, customs clearing and forwarding, cross trades, inland transportation, internal cargo tracking systems, customer freight costing and consultancy, cold chain logistics management, logistics and warehousing, quality inspection, and much more. In Accra, Tema, and Takrady, we are always available to ensure easy movement of cargo countrywide. Call us on 0204 528 900 or 0244 707 859. Email info@bethellogistics.com. Locate us in Accra at North Ridge, adjacent Accra Girls High School, or in Tema at Community 4, or in Takrady on the second floor of the GCB building. Website bethellogistics.com. Bethelog, experts in shipping logistics.
Hello, how are you doing today? Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, it's not painful. Blood. Okay.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patients that we need a sample of her blood for the lab.
No. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that.
Oh, good. I hope you are well. Don't worry. We are here to help you. All of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay? For our diagnosis. You see, please, it's okay. Take it easy. Take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. It's okay. You take her back to the ward. You'll be fine, okay? You need a sign language interpreter.
Hey, stop looking at them. Who told you I'm interested? I can see it in your eyes. See. Doris, they're definitely...
If you need a reliable procurement and supply chain partner, look no further. Global procurement company Excelsior Global Limited is here for you. We work across multiple industries and sectors with one goal in mind, ensuring that our clients focus on core businesses while outsourcing procurement. Excelsior Global takes care of purchasing process while reducing all procurement and supply chain costs. We offer a wide variety of procurement solutions connected mainly with medical, COVID-19 antigen test kits, COVID-19 antibody test kits, COVID-19 antigen test kits, virus transport media, specimen collection swab, sample preservation tubes, HIV test strips, hepatitis C test strips, hepatitis B test kits, syphilis test strips, glaucometer test strips, pregnancy test kits, hepatitis C, HCV, examination gloves, syringe, surgical gloves. Excelsior Global makes procurement easy, simple, on time, and most important, the job is in safe hands. We do what others don't. Visit our office at 35 Patrice Lumumba Road, Airport Residential Area, Accra. For more information, call us at 0559-122-092. Mail us at info@excelsiorgh.com or visit www.excelsiorgh.com. If you need a reliable procurement and supply chain partner, look no further. Global procurement company Excelsior Global Limited is here for you. We work across multiple industries and sectors with one goal in mind, ensuring that our clients focus on core businesses while outsourcing procurement. Excelsior Global takes care of purchasing process while reducing all procurement and supply chain costs. The job is in safe hands. We do what others don't. Visit our office at 35 Patrice Lumumba Road, Airport Residential Area, Accra. For more information, call us at 0559-122-092. Mail us at info@excelsiorgh.com or visit www.excelsiorgh.com.
In the lab, you face consistent pressure to do more with less while trying to keep up with a growing workload. Success means having instruments that you can rely on to overcome diverse challenges. The Agilent 6470B Triple Quadrupole LCMS, the latest version of our trusted workhorse, gives you the robustness and versatility you need to overcome these challenges. The 6470B has a proven reputation for robustness with non-stop operation, backed up by thousands of users with numerous application notes and peer-reviewed articles to reduce downtime and increase efficiency. Enhance versatility with a wide array of ion sources to analyze any application. Ion source options include ESI, the standard choice to broadly ionize analytes of interest, AJS, which enhances the desolvation power of standard ESI for ultra-high sensitivity, APCI, for increased signal-to-noise for very small analytes, and MMI, to carry out ESI and APCI simultaneously for a complementary analysis. With the new WACCBIP, you can reduce maintenance time by 92%. WACCBIP lets you remove the capillary and run the cleaning cycle in approximately 30 minutes, compared to about 6 hours for maintenance by venting and disassembly. So you can gain back valuable lab time and keep up with high demands. From analyzing environmental contaminants and ensuring food safety to complex life science research, the 6470B meets your need for reliable, accurate results. These accurate results are provided by our Mass Hunter software, including Quant My Way, which enables you to personalize the software with tools that highlight only the capabilities of interest. With comprehensive end-to-end workflow solutions and application services, you can ensure proper sample preparation, expertly developed acquisition and data analysis protocols, and fully customizable reports to get the answers you need as soon as possible. Our solutions are designed to work together to provide efficiency gains that help you get more done and reduce operation costs. The Agilent 6470B triple quadrupole LCMS is unbelievably robust to increase your lab efficiency and remarkably versatile to keep up with your evolving demands. Visit agilent.com/chem/6470B for more information. Decades of innovation have brought us into a new era of mass spectrometry with the design of Altivo, Agilent's revolutionary triple quadrupole LCMS. At just 1/3 the size of our previous triple quad LCMS instruments, Altivo integrates seamlessly into the stack of the Agilent Infinity Lab LC System Series, saving bench space while offering the same power and accuracy of our larger systems. The footprint of Altivo is 70% smaller than that of similar LC triple quadrupole mass spectrometers, so you can triple your lab's capacity in the same space. The efficiency-optimized Agilent Infinity Lab LC Series system can sit directly on top of Altivo, making it the perfect partner for high-throughput mass spectrometry. The innovations that enabled Altivo were specifically designed to maximize quantitative performance, enhance instrument reliability and robustness, and promote greater uptime. The more ions you get into the instrument, the better your MS results. With Agilent's Jetstream ion source, a superheated nitrogen sheath gas surrounds the electrospray to quickly and efficiently desolvate the spray plume and tightly cluster the resulting ion cloud, maximizing the number of ions that are created and transported into the LCMS. WACCBIP features fast ion injector exchange without the need for system venting and improved diagnostics that guide the operator to appropriate corrective action, leading to less downtime for maintenance and higher throughput for your laboratory. Once the ions enter Altivo, they pass through our innovative precision-manufactured Cyclone Ion Guide. With its twisted and tapered dual hexapole configuration, the Cyclone Ion Guide provides maximum ion capture, compression, and transmission across a large pressure drop region, giving you better, more reproducible results. From the Cyclone Ion Guide, the ions continue to the first quadrupole. Better mass transmission improves analytical results. Each quadrupole assembly includes DC-only, wafer-thin pre- and post-filters, which cancel fringe field regions at the quadrupole entrance and exit to improve ion transmission. This allows for faster switching, enabling more MRMs to be performed. With the analyte selected in Q1, the ions pass through the vortex collision cell. Its innovative twisted and tapered dodecapole hexapole geometry maximizes ion collection, fragmentation, and transmission. The high-efficiency detector offers a wide dynamic range to ensure linear responses for your most challenging analyses. The detector was designed to be easily replaced by anyone in the lab. Working together, the compression and focusing of the cyclone ion guide, the improved ion transmission through Q1 with its pre- and post-filters, the optimized fragmentation in the vortex collision cell, the improved transmission through Q2, and precise data capture of our innovative detector technology give you better MS results. Altivo uses an entirely redesigned electronics architecture, making it the smartest LCMS on the market. New intelligent diagnostics tools enable Altivo to monitor several parameters in real time to ensure that it's operating at peak performance. If it isn't, it lets you know so that you can take appropriate action. For food safety labs, Altivo offers the selectivity and sensitivity needed to confidently monitor for and identify contaminants, even in difficult matrices, helping keep the food supply safe. For environmental analysis, Altivo provides robust and accurate measurements down to sub-part per billion levels and integrates with Mass Hunter software for powerful data collection, processing, and reporting to help you keep up with newly emerging pollutants. Reshape expectations, reinvent capabilities, and redefine what's possible when small meets powerful. The Agilent Altivo Triple Quadrupole Mass Spectrometer.
There is no promise we cannot keep. There is no commitment we cannot honor. There is no delivery we cannot guarantee. There is only one Bethel Logistics Company Limited, a licensed international freight forwarding and clearing agent, a trusted global brand with an extensive network for the movement of air freight and ocean freight worldwide. We are the trusted name for road freight into Western and Northern Africa. We are fully accredited by Ghana Customs and Excise. We cater for all your freight forwarding, customs clearing and forwarding, cross trades, inland transportation, internal cargo tracking systems, customer freight costing and consultancy, cold chain logistics management, logistics and warehousing, quality inspection, and much more. In Accra, Tema, and Takrady, we are always available to ensure easy movement of cargo countrywide. Call us on 0204 528 900 or 0244 707 859. Email info@bethellogistics.com. Locate us in Accra at North Ridge, adjacent Accra Girls High School, or in Tema at Community 4, or in Takrady on the second floor of the GCB building. Website bethellogistics.com. Bethelog, experts in shipping logistics.
Hello, how are you doing today? Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, it's not painful. Blood. Okay.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patients that we need a sample of her blood for the lab.
No. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that. Look, please, we are here.
Oh, good. I hope you are well. Don't worry. We are here to help you. All of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay? For our diagnosis. You see, please, it's okay. Take it easy. Take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. It's okay. You take her back to the ward. You'll be fine, okay? We need a sign language interpreter.
Hey, stop looking at them. Who told you I'm interested? I can see it in your eyes. See. Doris, they are deaf and dumb people. It's true. I hear these people are friends though. How do you even introduce them as your friends?
Every physical condition has its medical cause. It has nothing to do with curses.
Oh, hello, sir. Your money?
Oh, I see. You're not deaf?
No, I'm not deaf.
That is interesting.
Yes.
Aha.
boss.
Oh, okay. I will need your assistance to communicate with a deaf patient.
Oh, okay. Which hospital?
Legon Hospital.
Wow. Actually, I've just been transferred from the Alpha Clinic to the X-ray department.
Really?
Yes.
Wonderful. That makes my work easier.
I think I can help.
Yes.
Okay.
Please.
Okay.
This boy, then we guess it for
Let me see how far.
Hey! What is this?
Masha! Masha!
What is it?
Warn your son! Warn your son! Or else you will see what you don't want to see and hear what you don't want to hear. How many times have I not warned him not to play with my son or come to my house?
Oh, Ejapia! What's wrong? If Kofi plays with your son, are we not from the same clan?
Hey! Don't you know there is a case on you and your husband?
Me and my husband?
Yes!
Me and my husband?
Yes!
Very good. My husband is in.
Did you say curse? All right. We'll see who that curse is on.
Deal with him! Curse him! Deal with him! But shipping, come on. Wow.
Jonathan,
we can't find Amma.
Where is she?
I don't know. We've looked everywhere.
What? Yes. In that critical condition? Oh God. This is unbelievable. We have to find her. We have to find her at all costs. I'll handle it. My friend, thank you very much for coming. I'll call you when she turns up.
I'll shout. What more tests do you need? We know the people who did all the antenatal tests. How can you tell us our child is dead? We will solve our own problem. Let's go.
Excuse me, sir. Is there any problem?
What I say is final. We are not going to any pastor. We are going straight to the village.
I am not ready to take my daughter to the shrine.
No one in my family is deaf. And your family too, there is none. So how can you tell me my daughter is deaf? We must look into it. This is purely spiritual.
What about going to do the test? So the doctor can determine how to help her.
It is my daughter who is deaf. What do they need my blood for?
Our blood. That is what will help her.
Blood is sacred. I am not giving my blood to any doctor. I will call Papa at the village to arrange a meeting with the diviner.
Thank you.
Oh, great diviner, eye of the gods, the great owl whose gaze pierces through the thickest shadow and the darkest night. There is a deity that must be appeased in your family. She has been abandoned and has come with a vengeance of deafness. You must pay the due and purge your child. 3 rams and 3,000 Ghanas.
Your Highness, can we pay in cash?
Oh, the gods can take cash, even mobile money. Chikere, pala wins.
Hey! Something is happening to her.
Don't worry, my daughter. The spirit is leaving her. Mali, Mali, Mali.
He said she was going to get better when we get home. But now, her condition is worsening. Please help us, please.
This is highly demonic. She needs deliverance. I can feel it in me that there's a bad spirit inside this child.
Prophetess, can you deliver her?
That is exactly what I am going to do. Let us bow before the Lord. Jesus, we trample on every serpent and scorpion. According to the word, we rebuke every demonic power of high places, every altar that stands against the authorities of Jehovah. Be brought low. Jesus' mighty name. Jesus' name. Jesus' name. Oh, Father. Jesus. Jesus' name.
Kojo, she's not getting better.
What should we do now?
Let's go back and see the doctor.
Okay.
Let's go.
Doctor, thank you very much for the explanation. We got it all wrong. We now have a better understanding of what genetics is and its implications on deafness.
How can we thank you, doctor? Many people out there are not privy to this information.
Especially the people of our village. Doctor, there is a deaf community in our village. Is it possible for you and your team to come and explain this to the people?
Oh, that's a very good idea. It's an opportunity to reach out to the community and educate them. We will. Why not?
Thank you very much, doctor. Then I'll quickly arrange for the meeting.
Thank you. Thank you. Thank you. She's as normal as any other child. Just take good care of her. Okay? It's okay. Don't need to cry.
Ago! Nana and the council request tomorrow morning. Nana wants to meet everybody at the village square. The great Asparrow has spoken. Ago! People of Asempa, Nana and the council request your presence. Nana wants to meet everybody at the village square. The great Asparrow has spoken.
Nana and Council welcome you all to this urgent but very important meeting. Nana want to cut straight to the point. Nana, speak.
People of Asempa, the issue confronting us is a very serious one which needs to be addressed. I'm not an expert myself, but our forebears have told us over the decades the mysteries surrounding deafness in our community. Much as we have tried, we've not been able to find answers to this problem. But today, our son, Kojo, and daughter, Agnes, are here with some medical officers from Legon to educate us on the subject of deafness and its possible causes. Dr. Wo and Dr. James, you are most welcome to this community.
Thank you very much, Nana.
Please, you have the floor now.
Thank you very much, Nana.
As part of our work as clinicians and researchers, we investigate what makes each person unique and different. We do this by examining your blood sample. How many of you here have ever given their blood for testing?
Blood. I can't give
Not my family.
Blood.
My family.
No, blood is sacred. We don't give blood.
Yes. It will never happen like that.
I can't give my blood.
It is very important to give blood to be tested because every information that is used in building our body is packaged as genes. And this can be isolated from our cells in our blood sample. This we inherit from our parents. Please, can you bring the chart? You have to look on this chart. It will help you understand it better. Please. Our bodies are made up of billions of cells. Inside each cell is our genetic material. Chromosomes are the packaging of our genetic material. We usually have 23 pairs of chromosomes, and we inherit one copy from a mother and one from a father. If we unwind the chromosomes, we can identify the genes. Genes are the basic building blocks that makes proteins. Genes are like the blueprints for making a machine, and proteins are the individual components of the machine. Proteins makes us who we are and help us grow and function properly. Our genetic materials is made up of our DNA. Deoxyribonucleic acid is the very long chain of 4 chemical letters: adenine, guanine, thymine, and cytosine that combine to make our genetic code. We all have some variations in our genetic code. Genes variations are like spelling error, where, for example, there's a T instead of a C. A variation in the gene that makes it faulty or not working properly is called mutation. Faulty genes are caused commonly simply through chance. They have to occur in one individual for the first time at some stage. They are not related to anything a parent could have caused or prevented in any way. They occur because the process that replicates and divides their genes so we can pass it on to the next generation is complex, and mistakes occur. For each gene, we have 2 copies. One copy comes from the mother and the other copy from the father. In an autosomal recessive condition, an individual needs both copies of genes to be faulty for a condition to develop. The parents of a child usually carry one faulty gene of the gene pair. They are healthy carriers. It is estimated that every individual is a healthy carrier of several faulty genes, but a recessive condition only occurs when both individuals in a couple happen to carry the same faulty genes. When healthy carriers have children, there are 3 possible outcomes. 1. The child inherits both faulty gene copies and has the condition. 2. The child inherits only one faulty gene copy and is a healthy carrier like his or her parent. 3. The child inherits both working copies of the gene. So each time healthy carriers have children, there is a 1 in 4, 25% chance that their children could inherit both faulty gene copies and develop the condition. When an individual with a recessive condition have children, their children will usually all be healthy carriers of the condition. Their children could develop the condition if their partner happens to be a carrier of the faulty gene, but this could be a rare occurrence. Brothers and sisters of a healthy carrier could also be healthy carriers themselves.
So as you can see, features such as our height, complexion, color, including defects such as sickle cell and deafness, are all inherited from our parents. These traits are controlled by our genes. Have you noticed that every family has a feature that is passed on from generation to generation?
That is why our elders say the crab does not give birth to a bird. But I want to ask a question. Are you saying that deafness is not caused by curses or witches?
My elders and people of Asempa, that was what I believed until I could not find spiritual remedy for my daughter's deafness. That was why I gave in to genetic investigation.
I am glad this has come up. Please, from today, no one should associate deafness to witches or curses.
as we have explained, every defective condition is as a result of our genetic inheritance. Although there are environmental factors.
People like Mr. Akoto and his family, and like my daughter, are all normal human beings. Their deafness can be explained through scientific investigation.
No, please, let us put it on record that with the advancement of science and technology, every medical condition can be scientifically examined, including deafness. So please, I will kindly entreat all of you to stop attributing deafness to spiritual causes.
Doctor, please. Some of our neighbors and the entire family are deaf. Does it mean that they spread it to one another? That is why we want Nana to send them out from our community.
No. Please, be rest assured. Deafness is not contagious.
Hey, thank God! My children. Oh, my children. Thank you.
It's okay, woman. It's okay, woman. Please sit down. It's okay. Let them finish what they are saying.
Nana, all we need to do is to avail ourselves to be examined. It is possible some can be helped, but the most important thing is getting advice through genetic counseling and also having new babies screened for early detection. That's all we have to do.
Yo, my people, wisdom, they say, is like a baobab tree, and no one person can embrace it all alone. Today, we have been educated that the deaf people in our midst are our brothers and sisters. They are not under any curse. They are humans just like us. There is no curse on them. So we have to show them love and respect. And more importantly, we have to make ourselves available for genetic investigation.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, was established in 2014 as one of the World Bank's Africa Centers of Excellence. WACCBIP is located on the campus of the University of Ghana, Legon.
WACCBIP was established as one of the World Bank African Centers of Excellence. And the inspiration behind it is basically that a number of us who were at the Department of Biochemistry and the Noguchi Memorial Institute for Medical Research, we had trained outside and we did PhDs in various science disciplines outside the country. When we got back and realized that there was not much by way of facilities to train scientists, we saw this as an opportunity to create an environment which would enable many young scientists to get the opportunity to get the quality of training that we got overseas.
This world-class research center started off as an infectious disease center, but along the line, it's added non-communicable diseases to its focus areas.
WACCBIP is providing something to local and regional students that we probably don't have in many places within the country or within the region. Facilities, financial resources, as well as an environment that encourages student learning and research. And that has significantly increased students' output in the Department of Biochemistry, Cell and Molecular Biology.
WACCBIP has a whole range of collaborators at the institutional, national, regional, and international levels. These collaborations include the Noguchi Memorial Institute for Medical Research Ghana, the Department of Biomedical Sciences at the University of Ghana, among other departments, the Ghana Health Service, and the Ministry of Health. The center also has collaborations with institutions within and outside Africa.
Currently, there are about 6 labs in the center, and these labs do research based on the faculty who is in charge of these labs. We have a malaria group that has 3 different units. There's an immunology unit, there's a vaccine development unit, and also a transmission unit in there. We also have a molecular biology lab that has 2 faculty in charge there. One does work with African animal trypanosomes, and the other one, who is myself, works in Purulli ulcer and antimicrobial resistance. We have a huge virology group. They do research, anything that has to do with viruses. So they do HIV research. They even look at some bat viruses and gastroenteric viruses. More recently too, we've added a COVID lab, and this lab does routine testing for COVID positivity and negativity. And we also look for seroprevalence across the population. So people who have been prior exposed to COVID, but maybe have been asymptomatic, so they don't know whether they had COVID or not. And we've actually just recently published a paper on our findings.
The virology laboratory of WACCBIP was set up right from the beginning with one main lab, and they were doing things like rotaviral, researching into rotaviral infections in Ghana. The virology lab at WACCBIP was also involved in the first identification of a dengue case in Ghana, and also doing HIV research, as well as research on viruses which are in bats, but which might be have a risk of causing disease in humans. So these are filoviruses, the same type of viruses that cause Ebola. So that's work that was being done.
The center currently has 6 research laboratories, namely the Cell Biology and Immunology Lab, commonly known as the Malaria Lab, the Molecular Biology Lab, the Virology Lab, the Medicinal and Natural Products Lab, the Next Generation Sequencing Lab, and the Forensic Biochemistry Lab. At WACCBIP, the scientific tools and equipment are always and readily available to students, regardless of the courses being studied at the center.
Really, the driving force was to try to create a world-class research and training environment where young African scientists from across the continent can come here and develop their talent and do high-quality research that can be comparable to research that's done in any part of the world. So basically, that was the vision that we were pursuing.
Throughout my academic journey, I'd always looked for opportunities to come back home. My plan has always been to establish myself, to spend my most productive, my most useful years in Africa and hopefully in Ghana. So even though I studied abroad altogether for 13 years, followed by 6 years as a postdoc also abroad. So altogether I was away from Ghana for about 18 years. I was always looking for opportunities to return. So during my second postdoc, I actually met Professor Wandare, the director of WACCBIP, in London when he came to actually receive an award. And that was the first time I heard about WACCBIP. And so hearing about WACCBIP, hearing what Professor Wandare was trying to do in Ghana, I felt that it was a place that would welcome someone like me, a place that someone like me potentially could thrive. So I began to look for opportunities to return, and I was able to get funding to support my return to Ghana, and so applied for a position and was appointed as a research fellow. It was basically a fulfillment of something I'd always wanted to do, and WACCBIP stood out as a place that, even though it was a young center, was led by somebody who himself was very dynamic, who identified a lot of the ideals that I myself identify with, and so was somebody that I thought I could work for, somebody who could mentor me.
WACCBIP has evolved to become a world-class research center, thriving on initial funding from the World Bank and subsequent support from the Wellcome Trust and the Africa Academy.
We were very fortunate to get the funding through World Bank to establish the Center of Excellence here. And part of that funding gave us a huge chunk of money to buy core equipment that could be used and shared by all faculty in the department and opened up to faculty within the university and within the sub-region. We have a core facility for flow cytometry. We also have a core facility for sequencing of DNA. Just recently, we've even upgraded it to include a NextSeq Illumina sequencer, which is probably one of two in the entire Africa. And that has the capacity to sequence both DNA and RNA, which is world-class. We have a confocal microscope core facility, which can be used to take images of cells and for us to understand actual cell biology as it happens in real time. We also have the protein expression lab that I talked about, and we have state-of-the-art equipment in these laboratories that help us, again, make proteins, express proteins, and take it to the next level of trying to identify what these proteins actually do within cells.
Since I've been in the virology department, I've been doing mostly HIV research, that's my specialty, looking at how the different genotypes we have in Ghana affect the ability of treatment and the progression of the disease. And of course, since the pandemic, we have been right at the forefront of doing research of SARS-CoV-2, that's the virus that causes COVID-19 in Ghana. The HIV research that we've done here at Virology, prior to my coming, there's been some findings that in the Ghanaian population, some of the genes that may be involved in progression are deleted in some of the population. Actually, a few of my colleagues did that work. And since I've been here, we've been looking at the new drug which has been introduced in Ghana, dolutegravir, and our data, which is yet to be published, is showing that it is one of the best drugs to be used, and we are also selecting some mutations for this drug.
My experience in WACCBIP here has been very great. I came here in 2014. So after my undergraduate, I joined the lab in 2015 and I served as a research assistant. When I entered, I didn't know anything about research, but I've got opportunity through mentorship and training. I've had great experience here. Sure, I think there's fairness in the enrollment process. For what I know, I was fortunate after my first year master's, I was enrolled into the PhD program. Then what I have seen so far, I realized before you get admission to the program, you write in exams and they try to balance, look at gender balance and also the international and the local students. So there's fairness in the selection process. So my PhD thesis or project seeks to develop novel diagnoses for detecting Plasmodium species. The Plasmodium species are parasite that causes malaria, and currently we have RDTs and microscopy at the point of care for detecting these species. But unfortunately, these tools are not sensitive or specific enough for detecting these species. So there are more reliable tools, which are the PCR or molecular-based diagnostic tools. And if I can give you an example, that's what we are using to detect COVID now. But the unfortunate thing is these tools are not readily available in terms of the reagent. So my project seeks to develop these reagents here in Ghana so that we can make them readily available and also cost-effective for detecting infectious diseases.
The center runs 3 programs at the master's level, which includes Master of Philosophy in Molecular Cell Biology of Cell and Infectious Diseases, Master of Philosophy in Biochemistry, and Master of Philosophy in Molecular Biology. The PhD level includes PhD in Biochemistry and PhD in Molecular and Cell Biology of Infectious Diseases. The center also engages the community in its activities from time to time.
What was of common interest to all of us was that we wanted to create a world-class research and training environment to do basic science. Now, this was led by me, but with support from, several colleagues here at the Department of Biochemistry and also at the Noguchi Memorial Institute for Medical Research. In terms of the resources we need sustainable funding, predictable funding. If you look at all the countries that have developed, they have predictable funding for research and for science. If you look at the USA, they have something called the NIH, which they pump billions and billions of dollars in every year as part of their national budget. It's locked in. Ghana, we still don't have that. We've been talking about a research and innovation bill for a long time. Still not been enacted. We need to have such things so that we as scientists, there's a fund that we can apply to access funds to do research and to innovate on a sustainable basis. As we say, we compete for funds from overseas, and that is not predictable nor sustainable, because a lot of the funders, they keep asking us, how much is your government contributing? Okay, we've given you 8 million, we've given you 6 million, we've given you 5 million. How much is your government contributing?
Everybody knows that Africa has problems. Our problems are all over the news. We get offended when people point out only the problem. But it's also very clear that the only people who will solve Africa's problems are Africans themselves. We are waiting for malaria vaccines to be developed in Europe and North America for us to use. We are waiting for people, for President Jimmy Carter to come and eradicate guinea worm in Ghana. If Africa and in Ghana is going to achieve what I believe and we all believe we can achieve, we have to take responsibility for our problems and come up with locally relevant solutions to solve those problems. For me, WACCBIP represents a place that is trying to do its bit to contribute towards this. We are trying to train the next generation of scientists, not to come and work at WACCBIP. We are training the next generation of scientists to think of ways to solve problems they see in their own community.
International students are helped through WACCBIP to obtain accommodation. And basically what we do is that we liaise with the hostels and the halls on campus and arrange some of the rooms for them before they actually come in. At WACCBIP, we provide at least two forms of scholarship. One from the ACE World Bank grant and the other from the Delta's program.
Because we are in the tropics, a lot of patients report to the hospitals with high temperature. This is usually attributed to malaria, but we found out that malaria answers, let's say, 40% of those cases. For the other 60%, they are presumptively treated as malaria. And in most cases, the patients keep going back because it's not malaria. So what else is causing it? It's not clearly known. And the diagnostic systems in the hospitals are not advanced enough to take care of a variety of disease conditions. So this project was undertaken by one Nicholas, who looked at a whole panel of pathogens. So this was a discovery project. Okay, so once a child reports with fever and it's not attributed to anything else but infection, and they check for malaria and malaria is not there, they then take those samples and look for all manner of pathogens. Okay, and they were able to discover a whole variety of viruses and bacterial pathogens that explain those other febrile illness. So going forward, that would, instruct a new way of attending to patients in that category, so that we can. What we haven't done is to develop diagnostics that can be used in the hospitals, but that is the next phase that we can look at. The second is diagnostic technologies that have been developed by two PhD students, Francis Krampa and Felix Ansa. So here they are looking for electronic ways of detecting malaria. Okay, so what we have in the system right now is glucometer. You put a drop of blood and it tells you the level of glucose in your blood. We are trying to do the same for malaria so that people can even test at home, right? And not have to go to the hospital when this becomes available on the market. The third one is a project that concerns a new way of developing antibiotics. And what we are trying to do is to target what we call the genome stability of pathogens. This has not been done before, and we are pioneering that from this center for the first time. And we're hoping that over the years we will make progress that will lead to the next generation of antibiotics.
The first output that we had from our sequencing was a manuscript which was published, and that was the first 46 sequences of SARS-CoV-2 in Ghana. We published that last year, and since then we've done almost 1,000 sequences. So we are preparing that also to publish it. And why that is important is we are tracking basically the changes in the virus locally, because there's, if you look at literature or the news, you see how it's changing globally, but you have to know the local outbreak, you have to know the local epidemic. And what we see in Ghana is that sometimes the variants which are increasing are not necessarily what is increasing in the rest of the world. For now, for instance, we have Delta, which is also on the rise in Ghana. But there's another variant which is nobody's talking about, but it's rising almost faster than Delta in Ghana. So that's the sort of things that we do. And when we get the results, we write what we call policy briefs, advising the government that this is what we found. Earlier on in this year, when we did our sequencing of airport samples, and we first found the Alpha variant, that's the UK variant. And then we found it in local circulation, and we wrote it, we communicated to the government, and that was what led to the reinstatement of some of the restrictions, like restricting the funeral size, party size, and things like that. That was as a direct result of the work that we're doing.
The key message that we want to put out there is that Africa has a lot of talent. If you look across the world, there are African scientists doing great things all over the place. Some of these COVID vaccines that we are seeing, African scientists played key roles in developing them so quickly in different countries. If we create the right environment for them, a lot of these people will come back home. We have already brought 7 scientists, African scientists who were in the diaspora. We brought them here. And the reason why they were able to come back, or they were convinced to come back, is because they came and looked around, like you guys have looked around, and they saw that they can do good science here, and that this environment looks like where they've been working in the UK or in the US, and they are convinced that this is world-class enough. If we create that, we can start to bring all our talent back, and Africa can start to develop our own vaccines, can develop our own drugs, we can be more innovative and build new technologies that we can even export to other parts of the world, and not sit as we are now waiting for people to develop vaccines, use for their people. When they've had enough, then they share with us. That's where we are now. Okay? That situation has to change. That means we have to take matters into our own hands. That means the African governments have to start to put some money into science on a consistent basis. Let's have a fund, such as we have National Health Insurance Fund, we have GET Fund, we have all these funds that a percentage of the taxes goes directly into that fund every year. So that fund is available for scientists to access. Let's do that. Let's put some money aside for science, because this type of investment have to be done for us to develop. Otherwise, we keep marking time, we keep just treading water. Because if you keep relying on people for your innovations, that means you are not making progress. I keep telling people that if you think about investment in science, think about it like the way we invested in petroleum exploration. Ever since I was a child, I was hearing about GMPC, spending billions and billions of dollars doing oil exploration. Okay? They didn't find oil until just maybe 10 years ago or less, after spending billions and billions of dollars. Nobody complained that they were spending billions of dollars because they knew that when they found oil, they'll get a lot of money from it. It's the same with science. Don't think about what you'll get tomorrow, you know. Don't say that, oh, I've given you 200,000, so what have you brought after 2 weeks? No, it doesn't work that way. It has to be consistent investment over time. And then when you succeed, it will pay for itself. Okay? If we made a vaccine right now, can you imagine if we had a COVID vaccine and we're supplying to the whole of Africa? Can you imagine how much money Ghana will be making? But if you don't invest, you won't get. You won't get there. So you have to invest, and then eventually you hit the jackpot.
If there is any biomedical research center that promotes innovation and research, or that provides its students with unmatched financial resources and an environment for learning and research, it is the West Africa Center for Cell Biology of Infectious Pathogens, WACCBIP, providing world-class postgraduate training and research into tropical
Welcome to Inqaba Biotech, Africa's genomics company. Established in 2002 in Pretoria, South Africa, Inqaba Biotech is a rare and unique company, as suggested by the name, and we thrive to give reliable and efficient services to all our customers. How it all began. With the concepts developed in the year 2000 to speed up the arrival of primers in molecular labs in the country, Dr. Oliver Pricer, co-founder and executive director, together with a venture team of 5, secured a loan from Swiss government and got right to be the very first company to synthesize primers locally, and thus it all began. With capacity to synthesize over 1,000 primers a day, Inqaba remains the only company that does this in Africa. This is made possible by the Mermaid 192E. Using an ABI 3500 exome, we offer DNA sequencing from various templates, and we offer primer design, PCR amplification, cloning of PCR products, fungal and bacterial identification, and much more. In 2007, we launched Next Generation Sequencing Services, which provides NGS projects design inputs, quality control library preparation, and sequencing workflow using either the MySeq, NextSeq, or the PacBio systems. We have more services that include SNP genotyping and methylation analysis using the Agena Biosciences mass array systems, bioinformatics solutions that complement our NGS services, and qPCR analysis. We also are a distributor of strong international brands, which includes New England Biolabs, Zymo Research, Eppendorf, Glentham, CG, Nolato Treff, Charlab, just to name a few. Inqaba boasts a responsive technical team with expert knowledge in the services we provide. We also have a well-trained engineering staff making sure that the installation and servicing of instruments purchased at Inqaba is well taken care of. A fleet of vehicles commissioned for local trips and deliveries assures we arrive at your lab on time. In the past 18 years, we have started subsidiaries in East and West Africa, and we have offices in Kenya, Nigeria, Ghana, Tanzania, Senegal, and still maintain a strong presence in our neighboring countries. All this is achieved with a highly trained and professional team that is always ready to assist. Welcome to Inqaba Biotech.
If you need a reliable procurement and supply chain partner, look no further.
Global procurement company Excelsior Global Limited is here for you.
We work across multiple industries and sectors with one goal in mind, ensuring that our clients focus on core businesses while outsourcing procurement.
Excelsior Global takes care of purchasing process while reducing all procurement and supply chain costs.
We offer a wide variety of procurement solutions connected mainly with medical, COVID-19 antigen test kits, COVID-19 antibody test kits, COVID-19 antigen test kits, virus transport media, specimen collection swab, sample preservation tubes, HIV test strips, hepatitis C test strips, hepatitis B test kits, syphilis test strips, glaucometer test strips, pregnancy test kits, hepatitis C, HCV, examination gloves, syringe, surgical gloves.
Excelsior Global makes procurement easy, simple, on time, and most important, the job is in safe hands.
We do what others don't.
Visit our office at 35 Patrice Lumumba Road, Airport Residential Area, Accra.
For more information, call us at 0559-122-092.
Mail us at info@excelsiorgh.com
or visit www.excelsiorgh.com.
In the lab, you face consistent pressure to do more with less while trying to keep up with a growing workload. Success means having instruments that you can rely on to overcome diverse challenges. The Agilent 6470B Triple Quadrupole LCMS, the latest version of our trusted workhorse, gives you the robustness and versatility you need to overcome these challenges. The 6470B has a proven reputation for robustness with non-stop operation, backed up by thousands of users with numerous application notes and peer-reviewed articles to reduce downtime and increase efficiency. Enhance versatility with a wide array of ion sources to analyze any application. Ion source options include ESI, the standard choice to broadly ionize analytes of interest, AJS, which enhances the desolvation power of standard ESI for ultra-high sensitivity, APCI, for increased signal-to-noise for very small analytes, and MMI, to carry out ESI and APCI simultaneously for a complementary analysis. With the new WACCBIP, you can reduce maintenance time by 92%. WACCBIP lets you remove the capillary and run the cleaning cycle in approximately 30 minutes, compared to about 6 hours for maintenance by venting and disassembly. So you can gain back valuable lab time and keep up with high demands. From analyzing environmental contaminants and ensuring food safety to complex life science research, the 6470B meets your need for reliable, accurate results. These accurate results are provided by our Mass Hunter software, including Quant My Way, which enables you to personalize the software with tools that highlight only the capabilities of interest. With comprehensive end-to-end workflow solutions and application services, you can ensure proper sample preparation, expertly developed acquisition and data analysis protocols, and fully customizable reports to get the answers you need as soon as possible. Our solutions are designed to work together to provide efficiency gains that help you get more done and reduce operation costs. The Agilent 6470B Triple Quadrupole LCMS is unbelievably robust to increase your lab efficiency and remarkably versatile to keep up with your evolving demands. Visit agilent.com/chem/6470B for more information. Decades of innovation have brought us into a new era of mass spectrometry with the design of Altivo, Agilent's revolutionary triple quadrupole LCMS. At just 1/3 the size of our previous triple quad LCMS instruments, Altivo integrates seamlessly into the stack of the Agilent Infinity Lab LC System series, saving bench space while offering the same power and accuracy of our larger systems. The footprint of Altivo is 70% smaller than that of similar LC triple quadrupole mass spectrometers, so you can triple your lab's capacity in the same space. The efficiency-optimized Agilent InfinityLab LC Series system can sit directly on top of Altivo, making it the perfect partner for high-throughput mass spectrometry. The innovations that enabled Altivo were specifically designed to maximize quantitative performance, enhance instrument reliability and robustness, and promote greater uptime. The more ions you get into the instrument, the better your MS results. With Agilent's Jetstream ion source, a superheated nitrogen sheath gas surrounds the electrospray to quickly and efficiently desolvate the spray plume and tightly cluster the resulting ion cloud, maximizing the number of ions that are created and transported into the LC-MS. VacShield features fast ion injector exchange without the need for system venting and improved diagnostics that guide the operator to appropriate corrective action, leading to less downtime for maintenance and higher throughput for your laboratory. Once the ions enter Altivo, they pass through our innovative precision-manufactured Cyclone Ion Guide. With its twisted and tapered dual hexapole configuration, the Cyclone Ion Guide provides maximum ion capture, compression, and transmission across a large pressure drop region, giving you better, more reproducible results. From the Cyclone Ion Guide, the ions continue to the first quadrupole. Better mass transmission improves analytical results. Each quadrupole assembly includes DC-only, wafer-thin pre- and post-filters, which cancel fringe field regions at the quadrupole entrance and exit to improve ion transmission. This allows for faster switching, enabling more MRMs to be performed. With the analyte selected in Q1, the ions pass through the vortex collision cell. Its innovative twisted and tapered dodecapole hexapole geometry maximizes ion collection, fragmentation, and transmission. The high-efficiency detector offers a wide dynamic range to ensure linear responses for your most challenging analyses. The detector was designed to be easily replaced by anyone in the lab. Working together, the compression and focusing of the Cyclone Ion Guide, the improved ion transmission through Q1 with its pre- and post-filters, the optimized fragmentation in the vortex collision cell, the improved transmission through Q2, and precise data capture of our innovative detector technology give you better MS results. Altivo uses an entirely redesigned electronics architecture, making it the smartest LC-MS on the market. New intelligent diagnostics tools enable Altivo to monitor several parameters in real time to ensure that it's operating at peak performance. If it isn't, it lets you know so that you can take appropriate action. For food safety labs, Altivo offers the selectivity and sensitivity needed to confidently monitor for and identify contaminants, even in difficult matrices, helping keep the food supply safe. For environmental analysis, Altivo provides robust and accurate measurements down to sub-part per billion levels and integrates with Mass Hunter software for powerful data collection, processing, and reporting to help you keep up with newly emerging pollutants. Reshape expectations, reinvent capabilities, and redefine what's possible when small meets powerful. The Agilent Altivo Triple Quadrupole Mass Spectrometer.
There is no promise we cannot keep. There is no commitment we cannot honor. There is no delivery we cannot guarantee. There is only one Bethel Logistics Company Limited, a licensed international freight forwarding and clearing agent, a trusted global brand with an extensive network for the movement of air freight and ocean freight worldwide. We are the trusted name for road freight into Western and Northern Africa. We are fully accredited by Ghana Customs and Excise. We cater for all your freight forwarding, customs clearing and forwarding, cross trades, inland transportation, internal cargo tracking systems, customer freight costing and consultancy, cold chain logistics management, logistics and warehousing, quality inspection, and much more. In Accra, Tema, and Takrady, we are always available to ensure easy movement of cargo countrywide. Call us on 0204 528 900 or 0244 707 859. Email info@bethellogistics.com. Locate us in Accra at North Ridge, adjacent Accra Girls High School, or in Tema at Community 4, or in Takrady on the second floor of the GCB building. Website bethellogistics.com. Bethelog, experts in shipping logistics.
Preventing the interaction between ILA and CACR. I checked on the ICT target about 15 nanomolar. After that, I did what we call 2 formation assays using treatment resistance cell lines. I did them for the critical is enabling the tube to form. So this my results. The samples that I used untreated is M2 light water. But when this drug was applied in a dosage dependent manner, the number of tubes were coming down. This shows that if I inhibit the interaction between the different ACL2, reduce tube formation, thereby reducing so this month I went ahead to do overall active cell. I extracted the iron and I put it to PCR. So of course this is the result and I took out the markers. These are the results. When I did that, I realized that it was a kind of they went down a bit, but the marker goes up. It means that if I inhibit this interaction, it actually reduces or slows down the rate at then finally, I did go from gene knockdown. I knocked down CXCR2 using SIR that targeted CXCR2. So I did this CXCR2 formula with my knockdown process. After that, I went ahead to run CXCR2. My next month to decline as I said. If I then want to decline, if I then went up. All this results confirm that there is an interaction between either the CXCR2 and EX. That give me reason to conclude that there is interaction between this HCR2 and the inverted gamma. And then that if there is a way of inhibiting interaction, it will reduce the function of vascularity in different that is what I was about. And then of course there are other ways because I tend to people who
It's okay. Take it easy. Take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. It's okay. You take her back to the ward. You'll be fine, okay? You need a sign language interpreter.
Hey, stop looking at them. Who told you I'm interested? I can see it in your eyes. See? Doris, they are deaf and dumb people. It's true. Ah, I hear these people are cursed though. How do you even introduce them as your friends?
You know, every physical condition has its medical cause. It has nothing to do with curses.
Oh, hello, sir. Your money?
Oh, I see. You are not deaf?
No, I'm not deaf.
That is interesting.
Yes.
Aha. Oh, okay. I will need your assistance to communicate with a deaf patient.
Oh, okay. Which hospital?
Legon Hospital.
Wow. Actually, I've just been transferred from the Alpha Clinic to the X-ray department.
Really?
Yes.
Wonderful. That makes my work easier.
Yes. I think I can help.
Yes.
Okay.
Please.
Okay.
This boy, then we catch it for this.
Let me see how far.
I'll show you.
Hey! What is this?
Mansa!
What is it?
Warn your son! Warn your son! Or else you will see what you don't want to see and hear what you don't want to hear. How many times have I not warned him not to play with my son or come to my house?
Oh, Ejapia! What's wrong? If Kofi plays with your son, are we not from the same clan?
Hey! Don't you know there is a case on you and your husband?
Me and my husband?
Yes!
Me and my husband?
Yes!
Very good. My husband is in.
Did you say curse? All right. We'll see who that curse is on.
Deal with him! Curse him! Deal with him! But shipping, come on. What?
Jonathan, we can't find Amma.
Where is she?
I don't know. We've looked everywhere.
What? Yes. In that critical condition? Oh God. This is unbelievable. We have to find her. We have to find her at all costs. I'll handle it.
Thank you.
My friend, thank you very much for coming. I'll call you when she turns up.
I'll shout. What more tests do you need? We know the people who did all the antenatal tests. How can you tell us our child is deaf? We will solve our own problem. Let's go.
Excuse me, sir. Is there any problem?
what I say is final. We are not going to any pastor. We are going straight to the village.
I am not ready to take my daughter to the shrine.
No one in my family is deaf. And your family too, there is none. So how can you tell me my daughter is deaf? We must look into it. This is purely spiritual.
What about going to do the test? So the doctor can determine how to help her.
It is my daughter who is deaf. What do they need my blood for?
Our blood. I think that is what will help her.
Blood is sacred. I am not giving my blood to any doctor. I will call Papa at the village to arrange a meeting with the diviner.
Thank you.
Oh, great diviner, eye of the gods, the great owl whose gaze pierces through the thickest shadow and the darkest night. There is a deity that must be appeased in your family. She has been abandoned and has come with a vengeance of deafness. You must pay the due and purge your child. 3 rams and 3,000 Ghana cedis.
Your Highness, can we pay in cash?
Oh, the gods can take cash, even mobile money. Chikere, pala wensu. Pala.
Hey! Something is happening to her.
Don't worry, my daughter. The spirit is leaving her. Mali!
He said she was going to get better when we get home. But now, her condition is worsening. Please help us, please. This is highly demonic. She needs deliverance. I can feel it in me that there's a bad spirit inside this child.
Prophetess, can you deliver her?
That is exactly what I'm going to do. Let us bow before the Lord. Jesus, we trample on every serpent and scorpion. According to the word, we rebuke every demonic power of high places, every altar that stands against the authorities of Jehovah. Be brought low. Jesus' mighty name. Jesus' name. Jesus' name. Oh, Father. Jesus. Jesus' name. Koyo, she's not getting better.
What should we do now?
Let's go back and see the doctor.
Okay.
Let's go.
Doctor, thank you very much for the explanation. We got it all wrong.
See?
We now have a better understanding of what genetics is and its implications on deafness.
How can we find doctor? Many people out there are not privy to this information.
Especially the people of our village. Doctor, there is a deaf community in our village. Is it possible for you and your team to come and explain this to the people?
Oh, that's a very good idea. It's an opportunity to reach out to the community and educate them. We will. Why not?
Thank you very much, Doctor. Then I'll quickly arrange for the meeting.
Thank you. Thank you. Thank you. She's as normal as any other child. Just take good care of her. Okay? It's okay. Don't need to cry.
Ago! Nana and the council request tomorrow morning. Nana wants to meet everybody at the village square. The great Asparrow has spoken. Ago, people of Asempa, Nana and the council request your presence. Nana wants to meet everybody at the village square. The great Asparrow has spoken.
Nana and Council welcome you all to this urgent but very important meeting. Nana want to cut straight to the point. Nana, speak.
People of Asempa, the issue confronting us is a very serious one which needs to be addressed. I'm not an expert myself, but our forebears have told us over the decades the mysteries surrounding deafness in our community. Much as we have tried, we've not been able to find answers to this problem. But today, our son Kojo and daughter Agnes are here with some medical officers from Legon to educate us on the subject of deafness and its possible causes. Dr. Wo and Dr. James, you are most welcome to this community.
Thank you very much, Nana.
Please, you have the floor now.
Thank you very much, Nana.
As part of our work as clinicians and researchers, we investigate what makes each person unique and different. We do this by examining your blood sample. How many of you here have ever given their blood for testing?
Blood. I can't give my family.
Blood.
My family. No. Blood is sacred. We don't give blood. Yes. It will never happen like that. I can't give my blood.
It is very important to give blood to be tested because every information that is used in building our body is packaged as genes, and this can be isolated from our cells in our blood sample. This we inherit from our parents. Please, can you bring the chart? You have to look on this chart. It will help you understand it better. Please. Our bodies are made up of billions of cells. Inside each cell is our genetic material. Chromosomes are the packaging of our genetic material. We usually have 23 pairs of chromosomes, and we inherit one copy from a mother and one from a father. If we unwind the chromosomes, we can identify the genes. Genes are the basic building blocks that makes proteins. Genes are like the blueprints for making a machine, and proteins are the individual components of the machine. Proteins makes us who we are and help us grow and function properly. Our genetic materials is made up of our DNA. Deoxyribose nucleic acid is the very long chain of 4 chemical letters: adenine, guanine, thymine, and cytosine that combine to make our genetic code. We all have some variations in our genetic code.
Genes variations are like spelling errors, where for example there's a T instead of a C. A variation in the gene that makes it faulty or not working properly is called mutation. Faulty genes are caused commonly simply through chance. They have to occur in one individual for the first time at some stage. They are not related to anything the parents could have caused or prevented in any way. They occur because the process that replicates and divides their genes so we can pass it on to the next generation is complex, and mistakes occur. For each gene, we have 2 copies. One copy comes from the mother and the other copy from the father. In an autosomal recessive condition, an individual needs both copies of genes to be faulty for a condition to develop. The parents of a child usually carry one faulty gene of the gene pair. They are healthy carriers. It is estimated that every individual is a healthy carrier of several faulty genes, but a recessive condition only occurs when both individuals in a couple happen to carry the same faulty genes. When healthy carriers have children, there are 3 possible outcomes. 1. The child inherits both faulty gene copies and has the condition. 2. The child inherits only one faulty gene copy and is a healthy carrier like his or her parent. 3. The child inherits both working copies of the gene. So each time healthy carriers have children, there is a 1 in 4, 25% chance that their children could inherit both faulty gene copies and develop the condition. When an individual with a recessive condition have children, their children will usually all be healthy carriers of the condition. Their children could develop the condition if their partner happens to be a carrier of the faulty gene, but this could be a rare occurrence. Brothers and sisters of a healthy carrier could also be healthy carriers themselves.
So as you can see, features such as our height, complexion, color, including defects such as sickle cell and deafness, are all inherited from our parents. These traits are controlled by our genes. Have you noticed that every family has a feature that is passed on from generation to generation?
Yes. That is why our elders say the crab does not give birth to a bird. Yes. But I want to ask a question. Are you saying that deafness is not caused by curses or witches?
My elders and people of Asempa, that was what I believed until I could not find spiritual remedy for my daughter's deafness. That was why I gave in to genetic investigation.
I am glad this has come up. Please, from today, no one should associate deafness
for everyone
with witches
who has
or curses.
made the effort to
As
come
we have explained,
on time. We'll give a few, maybe 2 more minutes to see if the room fills up a little bit, and then we can start. But in the meantime, I'll introduce our co-chairs for this last but not least session. And that's Dr. Samuel Kusi from Noguchi and Dr. Teresa Manfujira from BCMB. So I think in 2 minutes or 3 minutes, at your discretion, you can take it away.
Defective condition is as a result of our genetic inheritance. Although there are environmental factors,
people like Mr. Akoto and his family, and like my daughter, are all normal human beings. Their deafness can be explained through scientific investigation.
No, please, let us put it on record that with the advancement of science and technology, every medical condition can be scientifically examined, including deafness. So please, I will kindly entreat all of you to stop attributing deafness to spiritual causes.
Doctor, please. Some of our neighbors and the entire family are deaf. Does it mean that they spread it to one another? That is why we want Nana to send them out from our community.
No. Please, be rest assured. Deafness is not contagious.
Hey, thank God!
Thank you. It's okay, woman. It's okay, woman. Please sit down. It's okay. Let them finish what they are saying.
Nana, all we need to do is to avail ourselves to be examined. It is possible some can be helped, but the most important thing is getting advice through genetic counseling and also having new babies screened for early detection. That's all we have to do.
Yo, my people, wisdom, they say, is like a baobab tree, and no one person can embrace it all alone. Today, we have been educated that the deaf people in our midst are brothers and sisters. They are not under any curse. They are humans just like us. There is no curse on them.
Welcome
so we have to show them
the last session for the day, and unfortunately, the last set of talks for the conference. We will start with a talk by Felix Ansah, and he will talk on towards the development of a cost-effective nucleic acid-based point-of-care diagnosis. Please, you have 15 minutes.
Hello. Okay. Good afternoon. Okay, all right. So before I start my presentation, I just want to give a brief background of what I'm doing. So in the opening ceremony, we heard about the need to establish local diagnostic tools for detecting infectious diseases. And yesterday we heard about some biotechs and what they are doing. So this is just a follow-up of what we've heard. Okay. Right, so before, because some of my colleagues, they've already introduced these methods, microscopy, culturing, and audits. I wouldn't talk about them. Right? So I'll go straight to my talk, which is nucleic acid-based amplification test for detecting infectious pathogens and also biosensors. Okay, right, so the focus of this project is to develop these tools for point-of-care application. They focus on looking at low and middle income countries. So once we talk about that, we have to think of affordable tools. We're also thinking of making it readily available without compromising the efficiency of our assays. So I also want to highlight that these assays are readily, they're already available, but the challenge is that they are not affordable and also not readily available. Okay, so to do that, what we are going to do is to, or what we did is to express the enzymes that we use for the nucleic acid-based amplification assays. Then with the enzymes that we express, we use it to prepare reaction mix. Then with the reaction mix, we prepare species-specific assays for detecting our target. So for PCR, we selected PFU polymerase, and also for LAMP, BST polymerase, and reverse transcriptase for RNA-based assays like COVID. Then the last thing is to also talk about some biosensors that we developed for detecting infectious pathogens. All right, so so the plasmid for expressing these enzymes are commercially available. So we obtain them from AdGene, then transfer them into BL21 expression cells and express the target enzyme. So these are some of the gels showing this is for PFU, which is for PCR, then this is for BST for LAMP, then also reverse transcriptase for RNA-based assays. For what we are trying to do here, like I said, these assays are already, they're already available, but the rationale here is to find the simplest method to use to purify this enzyme to reduce cost and make it readily available. So that's what we've done. I wouldn't talk about it. I wouldn't talk about the details of how we did that because of time. So we're using the purified enzyme. We prepared in-house reaction mix. So I'll refer to the master mix that we prepared with these enzymes as in-house reaction mix in my subsequent talk. Then with the master mix, we prepared and with the master mix, we use for detecting Plasmodium species, SARS-CoV-2, and also ISKN virus, which our sister presented on last 2 days, which is the virus that infects tilapia. So first one is why we chose PFU. So PFU is a high fidelity enzyme, and the other thing is that it also has a high thermostability and high proliferating activity, and it's also better for amplifying long reads compared to TAC polymerase. That's why we selected this enzyme. So with the enzymes that we expressed, what we did was to prepare reaction mix, and with the reaction mix, this for the red one signifies the reaction mix for the in-house. This is the amplification plots for the in-house reaction mix. Then this is the commercial one. So we compared the two. Then we realized when you put the plots together, you realize that they are comparable. And when you look at the melt curve, which is used for analyzing the specificity of our product, you realize the two reagents are comparable. Then when you separate the resulting products on the gel, this is for the commercial, then the in-house, we paired two different master mix with different concentration of our enzyme, and we got comparable results. Then after that, I tested it on some few clinical isolates, and we got our target amplified with good background. So what this means is that the enzyme that we produce has comparable activity to the commercial reagent. So using this reaction mix, what we intend to do is to prepare or develop species-specific assays for detecting as many pathogens as possible. So we will develop cyber green-based mix and also probe-based mix, and using the assays, we will perform routine surveillance and also real-time tracking of our target pathogen. Okay, right, so I'll start with the LAMP assay, because that's the focus, because the LAMP assay is for point-of-care application. So these are some of the methods. So we already heard about some LAMP assays that were developed, and also the first presenter, that's Kenny, she also talked about the LAMP assay that she developed. So I didn't go into detail. But these are some of the methods for detecting the LAMP product. So LAMP is similar to PCR. You amplify your target. Then these are the various methods, some of the methods that you can use to detect your target products. You can use a real-time qPCR machine, or you can also use colorimetric assays. You can also use pH indicators, agarose gel, lateral flow, and CRISPR-based method, and also biosensors. So I just want to highlight that yesterday the biotech personnel, Lord said he's using the lateral flow system. Then he intends using the CRISPR-based. So which method are you trying to use? We are using the colorimetric assays, and the reason for choosing this is that for all these methods, you can use PCR. Apart from the colorimetric method, PCR product, you can use all of them to detect. So only the colorimetric that you can use for detecting PCR based. And it's a bit, it's simple, but then it comes with its own challenges. So if you can solve those challenges, then it makes it relatively easier. So all the other ones comes at a cost. So for instance, when Kenny presented on Monday, she said, they are using a biosensor platform to detect their product, and she told us about the cost, which is about 50 pounds to 100 pounds, so that's extremely expensive. Then Lord also said using the lateral flow with the CRISPR, and it also comes with a challenge, because with the lateral flow, you would need to pipette your resulting product and load it into the flow pad, and that could cause contamination. So especially when you're dealing with them, you don't want to open your reagent. Then also with the real-time analysis, because of the bulky nature of the instrument, it's not easy for doing a field-based application. So the color-based assay seems to be the most preferred method for detecting the resulting product. Okay, so how does it work? So like I said, it's preferred for point-of-care application. It's field adaptable. It's based on color chain. Then the color is based on the amount of free magnesium ion in your reaction mix. So what happens is that, so the dye that we use is called hydroxynaphtha blue. So when you add it at the beginning of a reaction, you have purple color, or you can have purple or violet color. Then with the color, what happens is that in the course of the reaction amplification, we have PPI, which is pyrophosphate released from the DNTP incorporation during the growing strand. Then the PPI has affinity for binding magnesium ion. So once it binds and removes magnesium from your, or depletes magnesium from your reaction mix, then the color gradually changes from purple to sky blue. And that's how you're able to detect your positive sample. So there's an optional that you can further process your the resulting image if it's not too clear for you. So there's a software that's freely available. What the software does is to rotate the results, the color that you have through the color spectrum and give you, so it moves it from this particular range and brings it to this range from blue, from green or yellow to red. So this is one of the resulting assays that we did, and this is the original image. And after processing, these are some of the colors that the software retains. So sometimes it's not visible enough, so you want to use the software to process it. Okay, all right, so using this method, we prepared reaction mix for detecting Plasmodium species, SARS-CoV-2, and also the tilapia virus. Then we tested it on purified DNA samples and unpurified DNA samples. Like I said, the essence is to do point of application. So you want to look at unpurified sample and how it compares to purified DNA samples. So this is one of the resulting images. So this is for Plasmodium species, for purified DNA samples, and for this for SARS-CoV-2, then also this is the tilapia virus. Then all the positive targets. So these are samples that we've already determined, and we've run them with qPCR, so we knew that they were positive. So all the positive samples were detected. Then with the unpurified DNA, the same thing, all the positive samples were also detected. We ran negative as well, but I just decided to show these particular images. So the purified, so the assay shows that both the purified and unpurified DNA can detect our target with high sensitivity and specificity. So I also want to highlight that here, with the unpurified, with the Plasmodium, we use raw blood. So this is a blood you pick directly from the patient, or you take your sample and you add it to your reaction mix. With the sputum, so you take your sputum, you lyse, then you add it to your reaction mix. Then this is the fish tissue, or you pick the fish tissue, you lyse, then you add it to your reaction mix. So these are raw samples without purification. They work very well with our reaction mix. So the next thing we did was to compare the performance of the in-house BST to commercial BST, see how it compares. So these are different by the blue line. The blue plots are different badges for our enzymes. Then the red one is the commercial one. Then you can see that with the commercial one, it has a high, smaller time to positive, so it amplifies faster. What this means is that when you are, so when you look at different badges, here is it. So this is time to positive. So how many minutes it takes to get a positive result. So with our in-house master mix, you can see in less than about 5 minutes, you're able to tell whether it's positive or negative. That's if you're using if you're using real-time machine to measure. But what it means that from here, since the reaction ends here, so in less than 10 minutes, if you're using a calorimetric assay, you can determine whether your assay is positive or negative. So for both the purified and the unpurified, or the whole blood sample, so this particular one for Plasmodium species, so we use the raw blood and we're able to get them amplified with higher sensitivity and specificity, better than the commercial ones. Yeah. So what this means is that the in-house BST mix would have relatively less therapeutic turnaround time to get your results. So finally, what we did was to also compare the LAMP assay to our qPCRs. Well, this is also a published method. So this one, the qPCR, we use commercial reagent. So, and what we did was to take the fish virus, that's the tilapia virus, the tilapia sample, that one that was positive, then we diluted in 10-fold dilution. So from, okay. Yeah, so with this 10-fold dilution, so one sample that was positive, then we diluted further down in 7 steps. So you can say, assuming the starting concentration, though using nanodrop, the starting concentration was 2 nanogram. We diluted it to 0.2 femtogram. That's very small. So assuming the starting concentration is about 1 million per microliter, we diluted further down to about less than 1 copy per microliter. So how does it compare? So with the PCL, when you use the commercial reagent, the PCL, it's out of the 7, it's amplified for the first 5. So then when you compare to the lamp assay, when you compare the lamp assay, the lamp was able to amplify all the 7 samples. So it shows that the lamp assay has about 100-fold sensitivity for this particular target, or for this particular assay. So this work was done by Hannah Sewefia. Okay, all right, so what are they? So this, what this shows is that our in-house reagent works very well with our targets. And so what are they, towards developing this point of care tools, what are the key items that we need? So we need our reagents that we've produced, and few consumables, and also our heat source. So these are the few things that we need to perform these assays at the point of care or wherever it's needed. So during the COVID time, that's where we try to see whether we can move the assays from the lab to a place, maybe our home. So this particular assay, so these are the items that we packed. So we packed, this is the heat source. So this is a mini PCR machine, a handheld PCR machine. There are consumables, some gloves, then this is with our pipettes, then the reagents here. Then we were able to do some tests, and we got it working outside the lab. So that shows that the reagents that we are producing in-house, it can perform at point of care, like we are trying to do. So what we are looking forward to do is to lyophilize this reagent for easy transportation or movement. Okay, so the final bit is the development of the biosensor. This is the final slide. So this is how the electrode looks like. It has an electrical section for connecting to our instrument, then also a dielectric material for insulation, also defining the working area of the gold electrode. So this is a gold electrode. So what we are doing is to just we modify, so this electrode we modify with species-specific detection probes, which are nucleic acid based. Then with the probe, what we do is we add our sample, and if our target is present, we're able to read. So this one, we've already done a lot of work on it. We just add our sample, and we're able to detect whether it's positive or negative. So because it's nucleic acid based, it's easy to do for any target of interest. Yeah. So at the end of the day, what we hope to do is to be able to package this. Like these are some examples of biosensors, like the glucometer for field-based application. Then using that, we're able to detect your target of interest. Yeah, so thank you. I'd like to thank my supervisors, Professor Gordon Awanda, Dr. Yawaniwe, and Dr. Prosper Keyong for their support and contribution to the study. They are leading the diagnostic team. Then also everyone in the protein expression lab, and also Dr. Polly's research group for helping with the expression work. Then WACCBIP, and then NHS for sponsoring the work through the TIBA project. Thank you very much.
Questions, please.
Thank you very much. I think I'll open the floor. So the result you showed about your test taking 5 minutes, and the commercial one you're comparing to taking longer. Is there a difference in terms of the concentration of the enzyme that you are using, and could that affect your outcome? And then let me just follow it with my second question, which is there, I mean, when you are finished with developing the final product, is there a pathway to multiplexing where you can use the same device to detect multiple pathogens? Do you foresee any possibility like that?
So with the with the first question, yes. So the concentration of the enzyme, commercial one, they wouldn't. Yes, they stated the concentration, but ours, the badges that I show, we have some slight differences in the concentration. So there was differences in ours, the 3 different concentrations. So the commercial one, yes, I'm not sure they've stated, but it's not the same as what we are using. Yeah. So this is based on the fact that we have optimized our system. So perhaps ours might have more concentration, less concentration, but the rationale is to get our assay as fast as possible. So we've already optimized we've done serial dilution of our enzyme concentration. We know at which point will give us the optimum assay as fast as possible. Yeah, without compromising on the specificity of the assay. So it's likely ours has more enzyme than theirs, but we have done our in-house optimization. Then with the second question, multiplexing. Yes, so with the lamp assay, we can't multiplex in a single reaction because we are doing the color-based change. So, but what we can do, since when you pick like how a PCR strip look like, so it has 8 different tubes. So in each tube, we can put a particular target in there. So in one run, we can run as many targets, depending on the number of tubes we have. But each tube will take a particular one target. But with the biosensor platform, it depends on the number of electrodes we have on the number of working electrodes we have on a particular electrode. So this one, what I show, we have only one electrode, so we can detect only one. But we have some electrodes in the lab that has 5 different electrodes. So that one means it can take 5 different targets at the same time. So yeah, we can multiplex.
Okay, do we have more questions? So there are no more questions online. Okay, Dr. Kwashi.
Just to follow up on that, so is it not possible, sort of like almost like a litmus paper, to have, if you have different, I'm just asking you to think about it, if you have different reactions, each one gives different color, and you can sort of have a gradation of color, and then you would have to design, of course, something to be able to analyze that. You know what I mean?
So pH base.
No, I'm giving that as an example.
Yeah.
Where you have different things. So then let's say you have red and blue is going to show up as green and something like that.
Yeah.
I'm just thinking what do you think?
Okay, if I understand your question, you're looking at the color, maybe a color chart to
exactly
help. Yes.
Exactly. If you wanted to combine everything in one tube.
Okay, like different targets in one tube.
Exactly.
No, so the
because
color
of
has
the different chemistries.
No, yes, no. So the color has to do with magnesium ion, which is non-specific. So it's just for the reaction. So this particular one we can't multiplex. Yeah. So just the magnesium ion is responsible for the color chain. So one target per tube.
Okay, thank you very much. There's somebody's hand up at the back.
Felix, hi. So my question is, I know the goal of a biosensor is to, the goal when you develop biosensors is to try and develop something that's low cost. And I know the electrode you used was gold. So I wanted to know, wouldn't that influence when the final product is made, the price? And there are other alternative electrodes that you can use that might be as effective?
Okay, right. So you mean the electrode I presented here? Hello? The electrode I presented here? Yes, that's good electrode. So the other material like carbon or graphene, carbon that you can use, but once you use that one to remember the sensitivity, yeah, so we prefer gold because of sensitivity. You want to pick, we did this work because we are looking at low density infection. So you want something that's very sensitive, and gold has least electron transfer inhibition, and that's why we use the gold. So you can use other material, then but you need to, what you can do is to do good deposit, use good nanoparticle to deposit on those material to increase the sensitivity. So it's just a trade-off. If you want to use that one, you compromise sensitivity, and go to comes to explain. But then if you're using the gold, what we can do is the mass production. The mass production will reduce the cost. Currently, we are just buying few electrodes, but it comes in batch, and each batch they charge at a specific cost because they produce in batches. So even if you are buying 20, they will charge you that cost. If you are buying 10, they will charge you that cost. So it's just mass production. So if you're able to optimize this, once we do mass production, it's going to significantly reduce the cost of the assay.
Right, thank you.
Okay, so I think we should move on. Thank you, Felix, for a very exciting talk. So we'll continue with a talk from Charles Owal, network analysis revealing a model of HIV HPV synergy. Please, you have 12 minutes.
Good afternoon. All right, so I'm Charles. I'll be presenting part of my PhD work titled Network Analysis Reveals Model of HIV-HPV Synergy in Driving Cervical Cancer Progression. So cervical cancer still remains a major problem in most countries, particularly in sub-Saharan Africa. It's causing over 300,000 deaths annually. If you look at this map here, which shows different cancer mortalities among females, then you realize that the cervical cancer burden is concentrated in sub-Saharan Africa. And coincidentally, this is also the region that is highly burdened with HIV. So there's a clear correlation between HIV and cervical cancer burden. So here are just a few facts that I want to bring to you. So most cancers, cervical cancer is usually associated with HPV infection, persistent high-risk HPV infection. However, what has been noticed is that only few women progress to cervical cancer with HPV infection. So that means that HPV alone is not often sufficient to drive cervical cancer progression. However, when there is HIV co-infection, then it tends to drive cervical cancer progression faster, usually estimated to be 6 to 8 times higher. So here is a plot showing cervical cancer burden before introduction of antiretroviral therapy and after introduction of therapy. So if you look at the Kaposi sarcoma, you realize that there is a very drastic drop after the introduction of ART. The same applies to non-Hodgkin's lymphoma. However, when you look at cervical cancer, you realize that there is really no drop. We even see a slight increase in the ART era. So this really points to the fact that immune suppression that was thought to be driving cervical cancer progression among HIV-infected women may not be the key mechanism here. So what then is the problem? What then is causing this faster progression? Now, if we take this example, HPV infection, you need another factor, X, and that would lead to cervical cancer progression. So what is really X? Now, in a case where you have HIV and infected women, then X is likely to be mutations, just like any other cancers, maybe in the tumor suppressors or other genes. How about a case where you have HPV co-infection? Then what would be X? So this is what we are really trying to understand. So to really address this, we have proposed two hypotheses. In the first one, we are saying that HIV-infected women tend to overexpress certain inflammatory factors, which synergize with HPV to drive cervical cancer progression. Then in the second hypothesis, we are thinking of a direct mechanism where HIV or HIV proteins impinge on cancer pathways to drive cervical cancer progression. So to do this, we are harnessing a number of techniques like network modeling, statistical and bioinformatics analysis. And so we are trying to combine different sets of data sets here to answer these questions. So the first set of data is HPV and HIV protein interactions that were obtained through affinity purification mass spec in previous studies. And then we're also looking at inflammatory factors that we've gotten from literature, which are persistently expressed by HIV women, regardless of the viral load. So, but we think that the X could actually be overlapping with mutation data. So let's see how that happens. So here is an overview of the method or the pipeline that we are using for the analysis. So for hypothesis 1, we are taking inflammatory factors from literature and then HPV PPIs on one hand, and then for hypothesis 2, we are looking at HPV PPIs and HIV PPIs, and then we are integrating them using network modeling. In this case, we are using what we call network propagation. I will explain that in a short while. And then we integrate cervical cancer genomics data, and then we do a lot of bioinformatics and statistical analysis to be able to propose a model that explain what we think could be happening. So basically, when we talk of network propagation, then this is just a technique that enables you to, bring two data sets together, combine them, so that you get pathways that, converge between them. And so for you to do network propagation, you need a base network. And what we are using as our base network is pathway commons. Now this one is a very big network that has been obtained over decades. And the beauty of this network is that it has both physical and functional interactions, and it spans almost the entire genome. So it's very massive. So for network propagation, basically I'll not go into the details, but basically you seed your genes of interest into the base network, and then you let them spread, and then you get the ones that tend to associate more often with your genes of interest. So they are scored, and then you can then integrate them either by multiplying the data sets gene-wise or by just looking at whatever is significant in both of them. So for hypothesis 1, this is what we did. So we just took the inflammatory factors, HPV PPIs, performed network propagation, and then we ended up with 19 subnetwork clusters, and then subjected them to gene set enrichment analysis to identify the pathways that they participate in. And we got this enrichment map. But then at this point, we were not sure which pathways to select because there are a number of pathways. So to be able to identify pathways that could be linked to, are likely to be involved in cervical cancer, we then plotted alteration frequencies from TCGA for each of the nodes from, for all the nodes per clusters. And then we selected the top 5 nodes, the top 5 clusters, and then went back to our map to identify the pathways that, are associated with them. And we saw a number of pathways like PIK3, DNA damage response, and that. Then just to add another layer of confidence that we selected the most important genes, pathways, we did this plot where we picked different cancers, leading cancers among females, and then looked at the genes that are associated with them, and then the pathways that we have identified from the map. And as you can see, the purple showing that cervical cancer is actually more often, these pathways are more often altered in cervical cancer than other cancers. So we then proceeded to look at what could be happening in human, samples. So we went to TCGA to get the real human samples, human data sets, and then plotted our mutation frequencies and copy number alterations for across different genes, grades and stages, and then selected the top 3 per heat map, and then overlaid them onto our network, propagated network, and did some enrichment for the clusters that were associated with those inflammatory markers, factors. And this, as you can see, we ended up with the same pathways that we identified from our propagation. So then, based on literature and of our observations, we are proposing an indirect model here where we think that HIV induces inflammatory factors like interleukin-6, and then this one sort of downregulates P10, and then subsequently overactivating other nodes within the PI3K pathway. And then eventually this would lead to increased cell proliferation and migration, and then advanced cancer. So this model is corroborated by a piece of network that we prepared from string database, which shows that there's a lot of interaction between our inflammatory markers and the nodes within the PI3K pathway. So we moved on to the second hypothesis, but the analysis method is the same as what I've shown you for hypothesis 1. So I'll not go into details, but in brief, we simply did now here propagation using HIV PPIs and HPV PPIs, and then here we ended up with 33 clusters, which were then subjected to gene-set enrichment analysis. And all the other analyses that I've shown you in TCGA and the rest. And this pointed us to three pathways, mRNA splicing, nidylation, and cell cycle progression. And so based on this, we proposed our second model, where we are suggesting, or we think that the E3 ligases, which were actually the leading, the common HIV interactors that we observed, are nidylated, and then this activates them, and then eventually we end up with a lot of aberrant ubiquitination, and this could affect the mRNA splicing proteins, leading to either depletion of maybe tumor suppressors, or different variant forms of proteins could result. And this eventually leads to aberrant cell cycle or immune evasion. And of course, we end up with advanced cancer. So in summary, we have tested 2 hypotheses to be able to understand how HIV contributes to cervical cancer progression. And we have seen that for HPV and inflammatory factors, they tend to converge on PIK3 AKT pathway. And then the HPV and HIV proteins tend to co-hijack mRNA splicing and cell cycle. And so overall, we think that HIV directly or indirectly synergizes with HPV to target, pathways that are associated with proliferation, migration, or immune evasion, and this could lead to advanced cancer. So we are currently planning on validating some of these findings, and we hope to update you in subsequent conferences. So I wish to thank our funders, WACCBIP, RAP, UCSF, and all my supervisors and the other teams for ensuring that we have this collaboration going on. Thank you.
Thank you very much. So we have time for some questions. Yes, Dominic.
That's a nice talk. I'm really interested in your project because you are looking at two viruses that are of interest to me, even though I'm in human genetics. So the findings that you have, do you have any idea of how we can implement this in terms of therapeutics against these pathogens? Such that, do you think we can have one particular compound that may be able to handle these two pathogens at the same time? Yeah. Is it possible to design something like that?
Yes, it's possible. So that's why we are trying to look for, pathways that where they converge, the HIV and HPV. And so we believe that after validation, we can identify certain targets that would, just be able to target both of the viruses. But I believe with further validation, that should be very possible. That's what we aim for at the end, yeah.
So the first question is from Mehdi Bohado. How could you translate your insights into clinical actions or therapeutics?
Okay. So that's more or less what I've explained, that we are looking at identifying pathways that are common between HIV and HPV, and that could lead to progression to cervical cancer. So we can then identify key molecules that, key proteins that are of interest that we can target to be able to either, develop therapeutics or detection methods for this cancer at early stages.
Okay.
Yeah.
So the next question is from Nathaniel Lati. Nice work. How do you think CDC 42 may be driving cervical cancer progression?
Okay, so CDC 42 is a raw GTPase. So that's one wing of the pathway that, the model that I didn't show. So this is a raw GTPase, and we know that raw GTPases are, involved in cell migration and proliferation. And so if they are altered, if interleukin 6, for instance, alters the expression of CDC42, then we expect that would impact cell migration and proliferation, and consequently that should lead to faster progression to cervical cancer, advanced cervical cancer.
Okay. Do we have more questions? Okay. Okay.
So maybe I missed this. Why, if this synergism between the two viruses, why does heart treatment not make not help?
Yeah, so that's what is really a major puzzle, because initially we thought that this is an immune, it's driven by immune suppression that's induced by HIV, right? The depletion of CD4 T cells. But then compared to other cancers that are also associated with HIV, you realize that for this one, we don't see a reduction after introduction of ART. So we don't know really what happens, but what we can clearly say is that immune suppression does not seem to be the main mechanism that is, responsible for for increased cervical cancer progression in these patients. So it's something that we are still investigating. We don't have a clear answer.
Go ahead, Philip.
Okay, thank you, Charles, for the presentation. My question is, does your analysis cater for other infections, or you have these two viruses in isolation? I don't know if...
Okay, so this is a computational work, so we don't have a challenge with co-infection with other things at this stage. Maybe when we get to human phase, where we are looking at human samples, then we can begin to worry about that.
Okay, I think we should move on. Thank you very much, Charles, for a very interesting talk. So a small announcement. So for those who want coffee, there's coffee in the WACCBIP conference room. So you can get coffee if you want coffee. All right, we'll continue.
Teresa. So we continue and we switch gears, going to Mycobacterium now. If Judah Kofi is around. Yes. So Judah will be telling us about microbiological assessment of Mycobacterium smegmatis strains with mutations in genes required for genome stability, effect of temperature on antibiotic susceptibility profiles of Mycobacterium smegmatis strains. And while I was reading, I was hoping I wouldn't make a mistake. That's a very long one. All right, over to you. You have 15 minutes. Oh, 12, sorry. 12 minutes.
Okay. Yeah, my name is Judah Kafui Kofi, and I'm excited and nervous also to be here presenting my work. So, as part of a bigger project where we seek to find extracts that are able to cause double-strand break, or and also some extracts that are able to inhibit the repair of double-strand break, we came up with this mini project out of it. And so I'll be taking you through parts of the findings that we've made so far from this project, which is the microbiological assessment of Mycobacterium smegmatis strains with mutations in genes required for genome stability. So to begin, the progression of science has been really great over the years, and especially for biological science, we've seen the use of model organisms. And such one of these model organisms is the Mycobacterium smegmatis. So as a model organism, it is non-pathogenic. It's a non-pathogenic environmental strain. And then also it shares over 2,000 homologous genes with other Mycobacterium strains. And then also, you know that it has this unique cell wall structure, which is similar to the other strains as well. As such, it is good for studying molecular and physiological and drug resistance mechanisms in mycobacterial diseases. Moving forward, as a model for studying genome stability, we know that genome stability contributes to the transmittance of genetic material from organism to organism, from one generation to the other. And in order to maintain the fidelity of this process, two processes are involved, which is homologous recombination and then non-homologous end joining. These ensure the fidelity of genetic stability. Moving forward, over the years, we have been faced with problems of antimicrobial resistance. And so in order to fight against this, now we have research that targets antibiotics to the genome. And one of such publications was by Dr. Vincent Tama and Dr. Patrick Arthur, which says that which indicates that DNA double-strand break formation and repair as target points for the discovery of novel antibiotics. So for my work, I used 4 different strains, and these are mutant strains. These strains have, so these strains have specific gene mutations that affect specific proteins that are needed in the 2 pathways that ensure genome stability, which is the homologous recombination and non-homologous end joining. So we have one of them being the strain has been nicknamed MS, that's Mycobacterium rec-A. And then we also have, that's another one called the rec-BCD. And then we have the LIC-D and then the MSKU. So these are, for MS rec-A and rec-BCD, they both are involved in homologous recombination. And then the LIC-D and the KU strains are involved in non-homologous end joining. So because there is currently work that's going on using these strains, and yet not much is known about their microbiological properties, I found it appropriate to embark on this study to be able to present data on that will be able to help us with the bigger project that is ongoing. And so the aim was to assess some microbiological characteristics of the mutant strains of the Mycobacterium. And the first objective, which we have accomplished successfully, was to determine the antibiotic susceptibility profiles of the mutant strains compared to the wild type. So we have some selected assay plates to highlight some differences. So the green shows the points of susceptibility of the cell to the antibiotics, to some antibiotics. So for MS wild type, we can see over here that the cells are very susceptible from the zone of inhibition that was formed at this point and then at this point. But looking at it at the same positions for the different strains also, we can see that there is no zone of inhibition formed, which means that these strains are resistant to the same antibiotics. Now, this wasn't only found in one strain, but the same points of resistance was located in all 4 mutant strains. This was done at 37 degrees. We profiled the same strains at 24 degrees Celsius. And we noticed some similarities as well as some differences also. So this also highlights some of the differences between the wild type and the mutant strain. So we can see here that at the point where there is resistance, there is susceptibility for the mutant strains in all 4. Here, let me see, so yeah. In this graph, we can, so to point out first, we have this panel of antibiotics, which is for cell wall disrupting, which are antibiotics that disrupt cell wall. And we can see that comparing the rec-A strain to the wild type is ultra-sensitive to antibiotics that target cell wall and also antibiotics that target, that inhibit ribosomes. Ribosome inhibiting antimicrobacteriums, yes. A similar pattern is observed even in the second mutant strain. As you can see, the same for the LIC-D, and the same can be seen in the KU. When we go to 24, we observe we observe a similar pattern also for all the strains. That's so now at 2 different temperatures, we are observing a similar pattern of sensitivity to the antibiotics. Again, when we go to at 37 degrees, we can see partial resistance or a decrease in sensitivity to penicillin-like or the penicillin type of antibiotics, which are here in front, the first 3, ampicillin, amoxicillin, and vancomycin, which the same effect is seen in Rec A, Rec B, C, D, Lec D, and MSKU, and in MSKU. But what's surprising is the same panel of, sorry about that. What's surprising about it is that the same panel of antibiotics that had shown that the mutant strains had shown some slight susceptibility to at 37 degrees, we could see total resistance for the same panel of antibiotics at 24 degrees Celsius, which we were quite not expecting. Also, there's a resistance that ran across all the strains of the mutants, which is for Ciza and Met, that's cycloserine and metronidazole. We can see resistance for Rec A and Rec B, C, D at 37. We can see the same resistance for Lec D and KU at the same 37 degrees. But when we go to 24 as well, we see that the same resistance was seen in the same type of resistance was seen in those antibiotics at even 24 degrees Celsius. Also, at 24 degrees Celsius only, we can see some form of sensitivity, just a little bit, to rifampicin between Rec A and then Rec BCD. But when we come to for the strains of Lec D and KU, which are involved in non-homologous end joining, they have complete resistance to that rifampicin, to the rifampicin antibiotics. And so here's a summary of the results that I just went over. So we have a partial resistance to the penicillin type antibiotics at the 37, but complete resistance to them at 24 degrees. And then all the strains, all the wild type strains, all the mutant strains compared to the wild type were all sensitive to the antimicrobials at both temperatures. And then also, all of them were still sensitive to the ribosome inhibitors. And then also, all show resistance to cycloserine and metronidazole, whereas only Lec D and MSKU at 24 show resistance to rifampicin. So for MS-Rec BCD, it was the only one that showed resistance to 5-FU at 37 degrees Celsius. So currently, we are determining the MIC of these antibiotics at 37 degrees with 1/4 the MIC of moxifloxacin. Moxifloxacin because it is an antibiotic that causes double-strand break. And so we want to see how it is affecting the cells that are mutant to the proteins that are defective in the repair and in the repair system for double-strand break. And then also, like I said earlier, the screening is ongoing for the double-strand break inducers and then the repair inhibitors. And then assessing some regulators of these inhibitors and inducers. Also, some gene expression analysis will be going on. Yes, and then we have some other physical, some physical assessments such as adhesion and biofilm, and then growth curve, and then some morphological assessments of the cell itself. So this is the first time such an analysis has been done on the strains. And then this is obviously going to be providing a platform for the study and the discovery of the compounds that target genome stability. And then also by providing additional information about these strains. So I'd like to acknowledge my supervisors, the entire team of PACALAB and WACCBIP for the funding to do this work. Thank you.
Okay, so thank you very much. Right on time. Time for questions, if there are any. Yes. Who has the microphone? Yes.
Hi, thank you for your presentation. Please, on your slide 7, when you were categorizing or determining the resistance pattern, I saw you use the zones and you determine susceptible and resistance. I was wondering, did you use any guideline, maybe UCAST or CSI, to guide you know that these organisms, because you can get zones of inhibition, but when you compare them with the guidelines, sometimes there may be resistance, although you have seen clear zones. So I wanted to know whether you use guidelines to determine the resistance profiles of the bacteria that you were indicating.
We didn't use guidelines to determine the resistance profiles, because these are just zones of inhibitions that were seen on the plate for the assay that we performed.
Then we can say that you saw some level of activity, but you cannot say the organisms are susceptible or they are sensitive, because there are no guideline states that's what I was trying to bring, because before you can say that an organism is susceptible to this type of antibiotic, there should be a standard guideline to
okay, so the antibiotics that we used were standard antibiotics.
Yes, but before you can, let's go on, let's move on to that.
Yeah, I think that's something you can look at in addition.
Yeah, sure.
Yeah.
And also, you talked about, you said you're using antibiotics that inhibit cell wall synthesis and ribosome synthesis, but you didn't give us the class of antibiotics that you used, because we can have 1 or 2 classes of antibiotics that do the same thing, but their susceptibility will be different, or their sensitivity will be different. So I think you should look at that one to know how you compare.
Right.
Thank you.
Thank you.
Thank you very much.
Hello, Judah. Great presentation. Well done. I just want a quick insight why the different temperatures for which you did the ASTs, 3724.
So, because Mycobacterium smegmatis is an environmental strain, there can be several fluctuations in the temperature of the environment. And so we decided to use the lowest point that we saw in literature, which was the 24, and then the highest point within which they usually grow, which was 37. So yes, that's why we use those two.
Okay, any other questions? Otherwise, I would say thank you very much, Judah. That was a very nice talk. So that is the end for the fellows sessions. We'll move on to the next, which is preliminary talk 9. And for this talk, we have Professor Gideon Hellegbe to talk to us about the glucose 6-phosphate dehydrogenase deficiency landscape in Ghana, the story of northern Ghana.
Good evening. Yeah, it seems you're tired. I think it's been a long day and then a long event. And I'm also excited to be here and also to get this presentation for you guys. Story that you've been doing all these years on the unique condition that have been neglected for some time among these vulnerable group, the pregnant women. And I also want to take the opportunity to also congratulate the team for what they are doing. I think fantastic work. and also excellent presentation by the students and all. It's amazing. And every year that I come here, I can get excited new things, and thumbs up to the group. So back to the presentation, glucose 6-phosphate dehydrogenase deficiency landscape in Ghana, the story of northern Ghana. Yeah, so do the outline of the presentation, look at the background and also why they study all these years, and then discuss the results, give some conclusions and recommendations, and then what the future look like. Now, by way of background, we want to look at the essentiality of the enzyme, GCPD, for the survival of the cell. Now, if you look at this scheme of work, rather in a cell, a lot of things can go on. Such as the respiration within the mitochondrion, and then the cell can be exposed to certain agents, such as ionizing radiation, some sulfur drugs, herbicides, anti-malaria, divisine, basically some compound found in fava beans. And when this happens, this oxygen molecule can be converted to superoxide radical and then fed down to the hydrogen peroxide. Now, with this enzyme, glutathione peroxidase, this can be converted to water, and however, this can go on to form the hydroxyl free radical, and this is where the damage can come in. And then where there will be oxidative damage to these biomolecules, i.e., such as lipids, proteins, and also the DNA. Now, this is where the enzyme comes in handy. If this enzyme, GCPD, is efficient and working, and it helps in the presence of this NADPH when we have glucose 6-phosphate being converted to 6-phosphogluconate, glucolactone. Now, the importance of this activity that it produces this enzyme, this NADPH, that to ensure the reduction of glutathione, and to produce this reduced glutathione, and then ensure that these hydroxyl free radical will not damage this DNA and also these other biomolecules, and also ensure that the peroxide is converted to water. So moving on, we noticed that to look at the essentiality, rather, when the GCPD activity is reduced, cell growth is inhibited, and when increased as well, of course, it stimulates the growth, and also it prevents the ROS mediated cell death, and also prevent the starvation of the serum. So individuals that are lacking this enzyme, of course, you realize that this molecule will not be available, hence these hydroxyl will be just available, causing damage. So there are individuals where this enzyme is deficient, and then data shows or research shows that there are about 400 million individuals globally, and then about 33% within Sub-Saharan Africa region. And in Ghana, almost 20% individuals have this condition. And this is very important for us to look at. Now, I brought this chart for you to see that studies are going on, and they want to look at the pattern. And then the focus of this is to look at the location where these studies are done, and also their conclusion that most of these, they were able to show that there are some variants of these that are resistant to certain species of the Plasmodium, and also for the fact that also during blood transfusion, they need to take note of the status of the individual for the blood that we donated. And then also, it's also showing here that there's some co-occurrence between the sickle cell trait and also the deficiency of this enzyme, GCPD. And then this other study showed that there was no significant difference between the enzyme, normal and deficient women in proportion when they were given this intermittent preventive treatment of the sulfur-based anti-malaria. Now, looking at this, it tells us with the location that in terms of northern Ghana, not much is being looked at. It's as though this condition is neglected in them. And one other issue that I also want to look at is that among a vulnerable group such as what? The pregnant women. And why Northern Ghana? I mean, you bear with me that in this is a regional disadvantage. When you look at the Northern Ghana compared to the southern part, it's a regional disadvantage in relation to healthcare. And also, the pregnant women have been a vulnerable group in that regard. And also, to look at SDG 3 to ensure healthy lives in this individual. Now, to know the story and to see how this can be really addressed or made a case, we need to know the baseline, because data is not available in this part of Northern Ghana with respect to the deficiency in this enzyme. So we need to find a baseline data, and that's very crucial in that regard. And also, it's observed that there are certain implications when individuals are deficient in that enzyme. One of them is on the liver, pressure on them. This is the accelerated degradation of or breakdown of the hemoglobin in that regard. And also looking at this co-occurrence, I'll discuss that later, how this is a very important in clinical practice. Then the method that are being utilized in assessing this in the hospitals. We want to look at the challenges and the prospects in certain new ones that can be utilized, and also what are the knowledge level of this disease among those that are being looked at, i.e. the pregnant women for that matter. Now, so this is the study area. Northern Ghana is made up of Savannah region, northern region, northeast, upper east, and upper west. And the study areas were done in the colored areas, that's the northern region, upper west, and upper east. And for the reason that these regions have these referral hospitals, in the Tamale, we have the Tamale Teaching Hospital, in upper east, Boga Regional Hospital, and also upper west, Wa Regional Hospital. And so that easy to get access with this patient, or not patient per se, but the pregnant women, so that we can use for the study. And then ethical clearance will also obtain so that we can have their consent. So in that period, we were able to screen over 8,000, almost 8,500 pregnant women using the test available in the hospitals, made hemoglobin reduction test. And then after that, we read that these were the prevalence 8.6, and then which is quite important for our observation. Now, we try to look at this infection among the pregnant women in relation to this condition, the GCSPD deficiency. You know, one of the triggers that have been noticed or mentioned is the fact that is exposure or into these infections of bacterial and viral origin. But there's no local data as to whether which kind of infection that should be or that is triggering this. So we look at these infection, syphilis, that's bacterial, and the HIV and hepatitis B out of viral. And so looking at plasmodium being a common disease condition in this part of the country, and Ghana in particular. It's just interesting to know that none of these were not signal associated in relation to the deficiency in that enzyme. We are not sure why that is so. As to whether it's because they're instrumentic, we are not clear as to that, and it does need to be proved further. Now, the other thing that also interesting also regard the anemia condition. We're expecting this to be significant between the deficient group and the normal. But what we need to know is that pregnant women already are having low hemoglobin. So it may be the case that those normal also have low HB, and the deficient one, therefore they are comparable in their HBs. Now, what is very interesting regarding public health interest is they associate with sickling, and that's significantly significant. I mean, yeah, associated. Okay, yeah, it means you are alive. Thank you very much. Now, and then this has clinical importance. And then what is that? The implication inside that, when you have these co-occurrence of DSPD deficiency and also the sickle cell disease, one issue of major concern is the fact that that individual can be misdiagnosed. The fact that with the sickle cell disease or the trait, what is happening is that there is isolated destruction of these red blood cells during their crisis. And when that happens, the body responds by producing new red blood cells, the reticulocytes. And for them, the enzyme level is high in them. So when you screen for them, you'll be measuring as though it's normal. And therefore, the value you get in terms of the enzyme activity will be in the reference range. So if that individual is deficient in that enzyme, and because it's also having a sickle cell disease situation, then you saw that individual is normal. But what you are measuring is that is the enzyme level within the young red blood, the reticulocytes. So this has implication in the sense that you have some false positives. And those individuals, if normally in the antenatal, what they do is that if they screen and they realize that you have a malaria, they'll give you the sulfur-based anti-malaria. And these individuals can be given that because they think they're in the reference range. And when they are given that, then what will happen is that there will be acute hemolysis. And if care is not taken, that individual or the pregnant woman can die. And there's situations where that has happened. And some of my students have been reporting that to me in the sense that we need to take note of. So this is an issue that needs to be looked at. The co-occurrence is not really looked at. If you are deficient, then they'll try to, but if you are co-occurrent, they don't look at that. So those people are falling in that category whereby they are giving such anti-malaria in that regard, that quinine-based and also chloroquine and then sulfur-based. Now, a study was shown and realized that this enzyme alkaline phosphate was elevated in individuals that were having the acute hemolysis in the GCPD deficient individual, and suggestive of the compromised liver situation. And therefore, we try to look at that. But it's been shown that alkaline phosphate elevation also can be associated with certain tissues, such as bone, and also in the last trimester, and other infections as well. So when we try to look at by means of liver function tests, and then we'll try to look at the function of the liver, measuring these enzymes, okay, it was interesting that we rather saw a low level of alkaline phosphatase within the deficient individual. Now, this was shown to us, and reading through the literature, it was observed that this also has certain implications in the sense that it's as though these individuals were, their nutrition was a problem, and also lacking certain vitamins or minerals, magnesium and zinc. Now, this, if that's the case, it relates to a study that we did with some individuals in that area, and whereby we were told that pregnant women are not to be given egg and meat. And when we found out why, we were told that it will make the unborn child to become a thief. Yes. These are some of the stories that goes on. Okay, and therefore, it's not, if that's the case related to nutrition, then that could be an issue. So we always try to recommend to them that they should come to the antenatal with their husband, because the husband are looked at very strong and powerful, and therefore when they come and they listen, they go back, they will to implement that to them. So that's something that we also have to look at. And then we realize that The increased hemolysis in these patients can also result into jaundice because of those hemoglobin, the bilirubin level. And it's something that the pregnant woman should be looked out for and also treated or monitored for that matter. Now, the other thing that we also want to look at is that the method that have been used, especially the MATE hemoglobin reduction test, will have certain challenges in terms of the amount of blood that is used, the time, 3 hours, and also it needs power. Okay, and therefore these normally are not in the rural setting. The individual will have to travel some distance before, because there's no power. Touch machine equipment cannot be implemented or set up there. So what we came across was that this new method that can be the RDT, but it's just like the cassette that we have for the malaria RDT, and also the care start by, so they're just like a glucometer. You put a strip in there, take this amount of blood, 5 microliters compared to 5 mils. Already the pregnant women have low hemoglobin, so it takes such amount of blood. So we were thinking that this should be something that we should look at. So this RDT has already been evaluated by this author in the Birmingham, and so it's to be very good. And I know some facilities are using them, but at the government hospitals, it's something that is not well utilized. But this one is not yet used, so we said let's evaluate and see how we can recommend so that it can be. So having done the evaluation with the biosensor with the MATE hemoglobin test, you realize that it had an AU of 0.3. Even though it was significant, it was far below the WHO recommended figure. That's what WHO says to be more than 7 for the 30% cutoff. Now, we're not sure why this is the case, but sometimes the time that we did the studies in those seasons, they're quite high, the temperature. So we're not sure if that could be a case, but we need to repeat it again and see what we can get.
Okay, Prof, you can round up in about 2 minutes for us.
Okay. Yes. Yeah, so the other thing is the knowledge level. We realized that most of them had low knowledge regarding the disease condition. Okay, so it's not surprising that, and the knowledge that we tried to ask them was relating to whether they know the disease, the reason why they should go for the test, and what are the triggers for that, and what are the symptoms if there's a trigger comes on. They didn't know much about that. And we can't blame them much, because when we had personal communication with the health with the midwives, they were saying that, well, India, they just were told they just have to do the test. But why they do the test, that's something. So they're unable to translate that to the midwife, so that even if some facility is not in that environment, they can encourage them. So if it's not there, they also not do it. When the woman comes with malaria, they give the sulfur-based or the quinine-based anti-malaria, and that ends it. And then we also recommend that there was no local word for the disease. So if you want to explain to them, how do you go about it? And I had a personal experience when I was teaching them, but that would be for dinner to discuss. Now, so the conclusion that we want to give is that the prevalence of this deficiency in enzyme is relatively high, and with this co-occurrence in northern Ghana, we'll have to influence policy guidelines on the routine DCPD deficiency assessment, and taking note of what? The sickle cell disease in combination. And what we want to recommend is that low HB should be monitored within these co-occurrent individuals. I mean, with the GCPD deficiency plus the sickle cell disease. And the full blood count should also be included in the antenatal. Yes, they do, but they only extract the HB level, that's it. But if they could look at the reticulocyte count, that would suggest that, well, this, if it's high, then you have to take note of that and see how, and avoid giving them this quinine-based therapy or the sulfur-based. And there should be increased education on the relevance of this deficiency among the health workers, the midwives, and also the pregnant women. And what does the future look like? It looked promising, and they want to replicate the study in other parts of the country, in the regional hospitals, but taking note of these co-occurrence, what the pattern like, so that we can look at that further. And also to do genetic studies to see which allele is associated with these co-occurrence, and that further make us understand the mechanism by which this situation is taking place.
Okay, Prof, I think you can leave your acknowledgement slide there,
okay,
so that we round up.
Yeah.
In Ghana, when you are the last born, you also get extra meat. And since he was the last, he got a little bit of extra time. So apologies for that.
Yeah.
Thank you very much, Prof. I
Thank
think
you.
that was a very good presentation. So it's time for a few questions, if there are. If we have any burning questions. Yes.
Thank you for the nice talk. I'm very happy you gave this talk here. This is because we did some small work with pregnant women at the police hospital, and we noticed that most of the methylene blue test, which is the gold standard, actually is not able to detect individuals who are having enzyme inactivity or those who are GCSPD deficient. So I'm very happy that you've been able to come up with this data. My question is, going forward, how can we put this together into a policy draft that probably could be implemented by the Ghana Health Service across board, so that we can probably contribute towards reducing maternal mortality? Thank you.
Yeah, thank you very much. You know, the mid hemoglobin reduction test is actually not the standard. It's because that's what we are doing. Most people conceive the idea that is the standard. The other thing that have to be done. And also, this qualitative. And that's where the problem can be, whereby subjective. More so for the partial defect, because this enzyme is on the X chromosome. And for the pregnant woman, because XX. So if it's on one X chromosome, it's partial, then those people can be missed if you use the test that is being used now. And therefore, those can have challenges because they can be given unknowingly. If the person, I showed the data earlier, if you use the tutor that they look at this, instead of using the spectrophometry, sometimes you don't do it. Now, going forward, with regard to policy, I think what we have to do is that it was interesting when Professor Nadia made her presentation, and the fact that we need to look at how we can do some engagement with before we do the work. Let's engage the stakeholders. I think that's what we are learning. I learned from her talk, and also going for that. The replication that I want to do, I think we have some colleagues we had a discussion earlier. We need to engage the stakeholders, the Ghana Health Service, the Minister of Health. Now, this is what we want to do. And this, in our prime data, this one is found over this number of years, over almost 9,000 subjects. Okay, and therefore, let's see together. If we involve them from the beginning, and we let them see the data speak for itself, then I believe they will take it up. But if I wait, and after the study is done, they'll just say, okay, thank you very much. And then the thing will lie on their shelf. So I think engaging them from the onset will help us a lot, so that we can be put in kind of a policy. And we're doing this dissemination and also inviting the media along the way or before, it will help. I think that's what I can address. Thank you.
Okay, there's another hand.
All right, thank you, Prof. Two quick ones. So for the co-occurrence, I'm interested the RBCs. So for sickle cell, how young? You said when the cells are, because they reproduce cells all the time, so you have a lot of the GCSPD deficiency, right? So the enzyme, how young should the cells be? Have they done any work to find out whether as early as so many days or something of the cells? That's when you observe that. And then secondly, which of the variants of the sickle cell, is it SS, SC, or which of them? I mean, the sickle cell, which variant?
Okay, regard to your first question, how young? As long as the Rebbe said, what we know is that as you are losing the blood, new ones are being produced. Okay, and the fact that the new ones are there, their enzyme activity is obviously high, and that can mask the deficiency that was observed as a result of what the aged one. As to how long, I'm not sure of the data on that yet, but all I know is that they are produced frequently, and therefore, if they are there in the system, and as you measure them, you'll be measuring that of the new ones, and then that will have a challenge in terms of the, let's say, the false positives, because you have the value within the reference range. Yeah.
Okay, so let's take the last question from
The second one.
Oh,
it was two
Jonathan?
questions. I was talking of the sickle cell, which variant, whether SC, SS, doesn't matter which variant of the sickle cell.
To be with the co-occurrence.
Exactly.
Oh, in this data, we didn't focus on that. As long as you have the trait and the disease, I mean, that's where the co-occurrence came in, yeah. But
Okay,
it can be
thank
looked
you.
at further.
All right.
Thank you.
All right, so last question here. Then we round the session up.
Yeah, thank you. I think it is related to the last question.
Okay.
Because the S gene we know occurs in our population, give or take 25%.
Yeah.
But the disease occurs in 2% of newborns. So ordinarily, you would expect that if you screen 8,000, we know that of 2% with the disease who are born, majority will die before they reach childbearing age. It's improving, but that's the situation. So I think when you tie that in with the policy implications, the question is, are you measuring the trait or the disease? Because that's when you can begin talking to the policymakers. Because if 20% of the population carry the S trait, then technically, in the 1970s, a lot of advocacy had gone forward not to make those who carry the S trait be considered as diseased, because there's a lot of discrimination against them and they've won that battle. So it is an important question from a public health perspective, whether we are talking about the trait or the disease. And I think that question, maybe if you look into your data, it should be reflected upon in that respect.
Okay.
Thank you.
Yeah, thank you for the comment.
Okay. So we will end this here. I just want to say thank you very much for being engaging. It has been a session that has gone rapidly. So thank you very much, Prof. Next, we will invite the Caramo rep, if he's around, to give us a short presentation. So in doing so, we will also quietly vacate our seats since the mandate given to us has come to an end. Thank you very much.
Thank you very much for your time and good afternoon. My name is Gershon from Caramo International, and we are here to introduce ourselves to you once again. Caramo has been existing for some time now. In Ghana, we are partnering with MES Equipment Limited, which MES is in the science research for more than 45 years now, supplying equipment and chemicals to most researchers. And Caramo is in the UK. That's the head office now, but we operate in more than 50 countries all over the world, and we are always there to support science. So this is what we do. We source science items all over the world, and we supply to researchers and for research projects everywhere. And this is why we stand out. we are not just focused on the equipment and the consumables. That is our basic function. But we help with the cold chain as well, the dry ice items. And also, these are some of the things that we do. We don't just buy for you, but in the process, we help you from the onset. We help with the paperwork. So if you need export licensing or you need to do tax exemption for your products coming to Ghana or most parts of the world, we are there to assist. And also, we help with the clearance. So you make your order, we do the clearing for you, and we deliver to your lab to enable you have the research. And also, we help with advice. We advise you on things that are available and what the options are available for you in case you need a particular chemical or standard for research and it's not available at the time, and how we do the shipment to you. Because mostly some of the things comes in batches. So we advise you on how it will come so that also help you to plan your work. And also we do the export packaging. So we package the items straight from UK and bring it out to you, or anywhere in the world that we are sourcing it. We do the packaging and we bring it to you. And also we do the third-party logistics. In case you are buying or you have a donor support from anywhere in the world, and you need to bring it down to Ghana or to your research lab, we help you with that. Even if the items were not hasn't been purchased from us, we are still there to support you to that regard. So we are out there on the first floor. So if you want more inquiries, you can come to us and we'll be there to support you. So this is one of our delivery we are making to WACCBIP. That was on the last conference. So we do a lot with WACCBIP, and we are very grateful for that. And we look forward to have the partnership going. Thank you very much.
Okay, so when we started on Wednesday, this book seemed quite intimidating and challenging. Together we've walked through the book, and we want to acknowledge all the presenters. So let's I want to express special thanks to the keynote speakers. The keynote speakers normally are people who are busy, but they take the time to prepare a talk and come here and deliver the talk to us. And so we want to acknowledge my friend Nadia, Bismarck, and Peter. So let's acknowledge them. Before we round up, we have some few duties to perform. First one is evaluation. So we normally do an online evaluation, and then we also do an interactive evaluation. So Lydia will come and tell you about the online one, and then I will do the more interesting one.
Hello again. So if you are connected online right now, kindly check your email. You would have received an email from either WACCBIP leader or WACCBIP manager. So I would wait 30 seconds for you to check your email. Eight seconds in.
The evaluation gives you a coupon to dinner.
You better not go back. Great. So it takes literally 1 minute to 1 to 2 minutes, maybe, to fill the evaluation. Most of them are check buttons that you need to indicate your level of approval or disapproval on the point being asked. And then there's a last session, which we actually want to hear from you. So it's a comment session. So that's where it will take probably another minute to fill. Kindly spend some time doing this, because we need to report back to our funders how we are utilizing the resources that they give us. So please, everyone, I entreat you, those online particularly, please try and do this as well. Thank you.
Merci beaucoup. So, whilst you are doing the evaluation, out interaction and feedback, we want to get better every year. So, please feel free to tell us what worked well and what can be done better. If it's your first time, you can tell us how how your experience was and what could have improved it. If you have been here several times, if there are changes that we did this year that you liked or did not like, we'd like to hear. So please feel free. We take it all in good spirit. Yes, Mauli. Mauli doesn't fail. Have you done your online one? Okay. Make sure you do your online one first before you start talking. Please, where's the microphone? It's not working. Go ahead.
Yeah, thank you very much for this opportunity. On a more personal note, I'll say this is my first WACCBIP conference in person, because last year, unfortunately, I wasn't able to be here. But the organization has been top-notch and everything has run smoothly. I only have a suggestion which has to do with the session we had, I think yesterday or so, with the keynote speakers and the panel discussion. Because the way forward was not to wait for someone to come to our aid, but for us to take the initiative. So I'm putting that with respect is important. Why they need to spend money, or you keep saying that government needs to invest in research and all that. So I believe that having a representative from the media as part of the panelists will help enrich that aspect of it. We go back to our labs. So I think that's my suggestion on that note. Thank you very much.
Yeah, I think you're right. Maybe next year we should even partner with a radio or TV station and they'll carry it live or something like that. It's a good suggestion. Thank you very much. Didier, are you taking notes? All right. Any other good suggestions? Yes, there's one at the back.
Thank you, Prof. I realized yesterday I tried to watch some of the presentations, but they were lagging. I think the video was way ahead of the audio.
Oh.
So I don't know if the virtual parts of the conference can be really looked up on. And also, there were a few complaints about the Facebook Live and other streaming platforms we're using. So if that could be checked on going forward, that would be very awesome.
Okay.
Thank you.
Good. So,
yes, thank you for that comment, Reynard. We did notice it, and all the live feeds, especially the ones on YouTube, are going to be re-edited and put back online. especially to eliminate the breaks and to make up for these lags. And then also for the media being invited to the panel discussion, because we have it recorded, we are going to move forward with our public engagement team to try and disseminate it a little bit further.
All right. So, but our ICT team, what can we do to improve the live feed? It's always been a problem. Last year, I think we had similar problems. I tried to watch some of the talks from my office when I had to have meetings alongside, and it was breaking. When it's working really well. The picture is clear and the sound is good, but then sometimes it breaks. Well, okay, yeah. So I was wondering whether it was my internet in the office or what, but I guess other people had other challenges. So we'll continue to try to do better, but I think that overall our picture quality and everything has been great. So I think that that's something that has improved from last year. So we also, yesterday, is it Yahoo was talking about the smuggling of goods into the country. Yeah, so anyway, this projector is a new one, and I can see the difference. So Lily, thank you very much. That's all I have to say, it's coded. Yes, any other, Dominic, yeah. So you mean the screen for the presenter, you think is better over there? Yeah, because it's more natural to stand this way. Yeah.
Prof, when the presenter is, we've been asked to look in there.
Yeah.
So when they project it, online you are fine, but the audience here, it's a little bit.
They are looking away.
And then your slides are also on that side, so it's a little bit confusing. Thank you. Great.
I think it's a great suggestion. It's a great suggestion. So I think naturally I would like to look over there than turn like this. So I think it's a great suggestion. And that's easy. You just move this thing to the other end. It's easy. Just that maybe you have to do some wiring across, but it should be done. That can be done. Thank you. And you wanted additional screens? Or cameras, additional cameras, so that you can see the room from different angles.
Yeah, I tried something interesting in Gambia. I thought after my Viva, I still wanted to have it. Just a quick one. Just a quick one. So I planned for a small dinner, but there were people outside who wanted to be part. So we just mounted laptops at the corners of the room and put it on Microsoft Teams. And then it looked like everyone was in the room with a big projector. So I'm just trying to suggest a similar approach. Thank you.
Thank you. Okay. Yes, it's getting interesting. Any more?
I think it was a fantastic conference. So far, as usual, it was like too short, but that's how this time. Maybe it's me getting old, but time is passing fast.
Should we cut the beer time?
We can close at 9. We're not going to cut on that. Yeah, that's like that's explicitly why I'm coming. But so Regarding the media presence, what I would suggest is, I mean, we heard a lot of fantastic talks, some highly technical in nature, and that is not what the media is interested, right? So, so for us scientists, everything shades kind of in gray, yeah? It's not black or white. So, but maybe it would be a good idea to have like a little offer summary with the submission of the abstract, capturing the relevance in a rather easy language and language that is presentable to the mass media. Because I don't think that like Miss and Mrs. Average knows about T cells, B cells, p-values or whatever. But maybe it would be a good idea to wrap it up and attract more media attention. But by that, if you have like of each talk a little a summary capturing the relevance and the purpose of that.
Okay, great, thank you. Yes.
Hello everyone, my name is Priscilla, and this is my first time of attending any conference at all.
Oh, really?
Yeah.
Wow. Conference virgin.
So I must say that
we have broken her conference virginity. Please go ahead.
So I must say that it's been an awesome time for me. Has really been interesting and educative. Although this is not my field, but I've learned
What's
a
your
lot.
field, please?
Chemistry.
Okay.
Yeah. So I've learned a lot from here, and I really want to appreciate you, Prof, for the work you are doing at WACCBIP. It's really commendable, and may God bless you.
It's not me. Me, I just talk. The people who work, they are not here. Me, I just talk. So I thank you for acknowledging my talking, but I'll acknowledge those who actually work when we finish. I'll tell you those who actually work. Okay. Yes, I think Samira wanted to speak.
Yeah, good evening. So personally, I've enjoyed the conference. However, a few people complained about not being able to register for the conference. Therefore, they had to take part virtually. And I think they also had challenges, a few hiccups with the virtual platform. So since we now have a database with their email addresses, maybe moving forward, we can send them emails about registering for the conference ahead of time, apart from posting the conference online on our various social media handles. Thank you.
Okay, thank you. Did you get an email asking you to register? You didn't? Really? Okay, that's a mistake. Normally, there's an email that goes out to all faculty and all students who have ever been in this program, including those who completed 10 years ago. So that's a mistake. I don't know why that happened.
Yeah.
Anyway, we'll check that. Comms team, please. It's very crucial because I have some faculty who also said they didn't get the email to register. So let's make sure that our email lists are up to date and everybody is in the database, so that when we send out an email, everybody gets it. Thank you very much for that feedback. Yes?
I'm here. Okay, just concerning the media issue, a thought just came to mind. I was just wondering if we could have a sort of press conference after everything, to just bring up the highlights of, our findings and also our challenges, so that the media can take it up from there.
Okay, thank you. So normally after the opening, we do some media interviews. These days I don't get time to watch TV, so I don't know where they showed them. But I did some interviews after the opening that day, and Lydia did some in local languages and others. So, and we tried to put a point across about the need to support research and so on and so forth. But you are probably suggesting another media event at a closing where we sort of do a communique. Yeah, I think that's a good idea. I think there was one year we did that. I asked the comms team to sort of write a release that they will give to the media for them to use. So maybe we can do that. Thank you. Yes. But by the way, they are tallying the scores for the winners of the poster and oral. So we are not wasting time at all. We are actually using the time properly whilst waiting for the scores.
Hi, good evening. Thank you very much for this wonderful conference. I think my problem has to do with the times allocated for the presentations. I feel like some of them were too short, so some of the presenters had to rush through and then not fully present the data that they had. So maybe we can work on it next time.
Did you rush yours? You wanted more time? Well, so it's a trade-off. We could give 25 minutes to each person, but that means we have only 10 talks. So how many students will get to present? You know, so it's always a balance. We're trying to give as many students as possible an opportunity to talk, and then we'll give others the poster option. So if we increase the amount of time, it means that fewer people get a chance to talk. So it's a balance. Yeah. So we think that, you know, globally, talks are usually 15 minutes. Even when I go to a conference, they give me 15 minutes to speak. Unless I'm a keynote speaker, I get 15 minutes to speak. So it's quite standard. And it's part of your training to learn how to have 10 slides and deliver your message. So yes, we appreciate the feedback, but there are considerations in choosing how many people present and how long. Thank you. Yes.
Good evening, everyone. I'm very happy to have participated to this conference, and I know that one of the things we are striving to do is to bring up rising scientists, and one of the things I was amazed with is a young scientist that traveled by road from Nigeria to come here, which was not sponsored, and I think that these are the kind of things we should encourage. I don't know if the organization is aware of such things, but I think having an undergraduate leave Nigeria in the night to come here for a conference is something which we should praise. So I think that
Who's that, please? So, yeah. What's your name? Give her the microphone, please. Okay, wow. What's your name and which institution are you coming from?
My name is Eseka Amazing Grace from the University of Medical Sciences, Ondo State, Nigeria.
So what motivated you to come here?
Okay, actually, I've been wanting to attend a conference for the longest time. So I spoke with one of my lecturers, and then she mentioned this conference. Yes, I told my parents about it.
So your parents, paid for you to come?
Yes, they did.
Oh, you're lucky. Thank you very much.
Yes.
So what can we do to make your journey back easier? All right, we'll find you some kind of incentive for coming.
Thank you.
So, you know, it reminds me of another instance where a student came in a similar way for one of the workshops. I don't remember which one. Do you remember which one? Where the student came by road also, no accommodation. He came here and arrived here in the evening and said he had nowhere to sleep. And he said he was willing to sleep in the room under the stairs there, which is like our switch room. He was willing to sleep there. So I came and he was actually, you know, getting comfortable to sleep there. So we had to look for space for him at a guest center or something. Who is this? Who? Come again? Is it a trend workshop? Yeah, so, we had to find him somewhere to sleep. But, that's the kind of determination that we're talking about. He has no clue where he's going to sleep, but he wants to be here. So he shows up and he's willing to sleep under the stairs just to learn, so that you have to respect that type of commitment. So, yeah, thank you. Okay. All right, any more?
Yes.
Oh, somebody has a microphone.
Thanks for the opportunity. As a sponsor, I'm very happy about where we have a place for the lunch. It's just within the posters and our stand, so that's brought more traffic to our place and also on the poster. So with that, we had more engagements that effect. So we'll be grateful that we repeat something similar in the subsequent conference. Thank you.
I think some people disagree. Who disagrees? Because I got the opposite feedback. Who disagrees? Yes, somebody disagrees.
Yeah, I feel like the posters and the dinner place being at the same place is not good enough for poster presentations. So we should find a poster room for poster presentations.
Yeah, it's quite interesting. You have to always balance these things. So normally we have the lunch outside. All the meals, we have them outside. And then the floors are just for posters. And then people don't go to the posters. People just eat and sit there and chat and wait for the next session. They don't go to the posters. They don't go to the exhibitors. They don't pass there at all. So then this year we tried to compel us to interact. And now we get the opposite. So anyway, these are things we have to balance. Yes, there's a hand there. Christian, I'll come back to you.
Yeah, so the book, I think it fell short of some of the abstract. This ended at 79, and we go all the way to, I think, 83 or 6. So if they could check that out.
Okay. Is your abstract missing?
Yes, mine is missing. It's missing.
We apologize profusely. Yeah. Yeah, so this year we're a little late in handling the abstracts. And also we got more abstracts than we normally do. So again, these are adjustments we have to make. So there was a lot of pressure in putting the abstracts together. So some may have been missed. So apologies. We'll compensate you. All right. Okay. Yeah.
I want to suggest some form of sightseeing on campus, aside from the normal conference and exhibition for our foreigners who come into the country.
It's true. In fact, I was supposed to do that yesterday. I forgot. I got so busy. I forgot. I was supposed to organize for the driver to take them around. Did that happen at all? No, it didn't happen. Yeah, we need to do that. It's true. Our campus is a city. So if you come here, at least you have to get a drive around. You can't exploit just by walking. It's huge. So I think it's true. We need to allow some 1 hour on one of the days for optional campus tour. So that's a good suggestion. Those of our collaborators who have traveled here, if you are still interested in that, we can organize one for tomorrow or so. Places will not be open tomorrow, but at least you can drive around and see the buildings and the facilities. So if you are interested, let us know. We can still do that. I promised to do that yesterday, but I forgot. Yeah. Okay. Thank you. Yeah, Christian.
So I also found it rather difficult to see the poster due to the lack of space. But to give and I see the difficulty in balancing the outside, inside lunch area. So a suggestion, I once was at this wonderful conference, and they had a very cool incentive. So they gave every participant a wine glass, and they gave every poster presenter a bottle of wine. And it worked wonderful. Yeah. So they had a good mixture of red and white wines, and that worked just wonderful. Yeah.
Was there any session after the posters?
Well, maybe those with the more shallow topics.
Because if you do that after the posters, then you're done for the day. You just close. So you must as well have the poster session in a bar. All right, thank you. Okay, you have.
So,
okay.
Stephen Opoku online is asking, will it be possible to get a soft copy of the Book of Abstracts?
Is that not online yet? Andrew, where is it? On our website? Okay, normally the brochure will be on our website. If you go, you even have the previous conferences brochures on the website. So this should be on the website probably shortly. But you shared the link where? Well, some people didn't get your email, so so let.
We did a scan.
There's a
we generated
code.
a QR code and then we put it on Zoom for the online.
So it's online now. How do
yes.
can you post it on?
Yeah, we'll upload it to the website and then also share the link.
Or you can share the link right now to those who are online watching,
right,
so that they can just click on it. You can just share it on the Zoom chat. All right. Peter, I thought I saw Peter coming.
All right, Prof.
Yes.
I want to comment on the assessment of the posters.
Who's talking?
Tego.
Oh, Tego, okay.
Right. So I attended a conference and the poster assessment, they used the QR barcode. So each poster has that. I don't know that's what we are using here, I don't know. So the assessors go there, they scan the barcode, and just after assessing, they get the results straight away. I don't know whether we can do that.
Yeah. Where's Peter? So yeah, I guess we can do that. It's I think this year they had an Excel chart or something that they were using to score. So yeah, I guess we can innovate a little bit more on that front. So I thought you were going to say that we vote. You know, there are some conferences where the posters is done by voting. So the participants go around and they are given coupons just like the ones you get for the dinner. And then each poster has a little box that you put your votes in there. So when we finish, we go and get all the boxes from the posters and count, and then you see who wins. So anyway, there are ways of doing this. So we'll look at how we can make it easier for the scorers. Okay. All right. Where's Peter? It's still writing the news. Okay. Okay.
This is from Ishmael Tego. I would like to propose if time can also be allotted for new research proposal representation, as this gives opportunity to gain possible funding from keynote speakers, sponsors, and invited guests. Thank you.
What did he say? New research.
I would like to propose if time can be allocated for new research proposal representation, as this gives opportunity to gain possible funding from keynote speakers, sponsors, and invited guests.
Okay. So like proposals. Okay. Okay. All right. Yes. Last one.
Just a follow-up on the comment that was just made. I think there was a time I think there was a meeting like this. I proposed if we could have a research wish list, whereby you have a proposal or the research you want to do. You know the research items you need, but you probably are not being sponsored by the normal funders, so that someone outside may look at it and say, okay, you need about $10,000 to complete this research. I'm willing to give you $1,000 because I'm interested, or 500. I think it's similar to what I don't know whether I say he or she, but yeah, so if we can create a wish list so that during the conference we can research wish list of items that you need, and maybe sponsors and people can say, okay, I'm giving you, you don't have money, I'm giving you pipette tips, or I'm buying you a PCR reagent, so that next conference you also come and tell us what you did. Yeah.
Okay. All right. Thank you. Okay, so I think we've had great feedback from all of you. So thank you very much for all the contributions. So I'm going to start the thank yous now, so that when the results come, we can just present the prizes and go. So first, I want to thank all our collaborators who traveled to come here from neighboring countries, far east and west of Africa, north, south, Europe, North America, you know, all over the place. So let's acknowledge our collaborators. I want to thank all our students for their participation and for the enthusiasm that they show. Anytime we have these conferences, you know, we are always, you know, basically encouraged and re-energized by the enthusiasm of our students and the way they make their presentations and cherish the opportunity to be up here. And that is what encourages us to keep giving them the opportunities to do science and to come up here. So thank you very much. Now, I want to thank those who actually do the work so that I can come here and talk, so that you don't stone me. If the conference wasn't good, you throw stones at me. So since you are not throwing stones at me and you are giving me all this positive feedback, we need to thank the team that put this together. We have something called the WACCBIP machine. Do you know what that is? Who knows what the WACCBIP machine is? Philip, what's the WACCBIP machine? Eh? It's what? No, it's not just the secretariat. You can't put a finger on it. Yeah? It's a system. It's a machinery that works. It includes cleaners, drivers, accountants, communications people, lab technicians. I mean, we have a whole machinery. Whenever we have an event like this, then the machine just rolls. And everybody has to play their role for things to work. Cleaners have to clean. When you come here, everything is clean. All the peanuts you dropped here are gone. There's water for you to drink. Meals are on time. Drivers pick you up from the airport. I mean, everybody plays their role, and that's what we call the WACCBIP machine. So let's give it up for the WACCBIP machine. And we don't take their work lightly. It is because we have created a culture or a work ethic here where people are held accountable for their work, and they're also empowered to take initiative. And that's how we built the machine. It's for people to be empowered to do their work and do it with pride and dignity, and also be recognized for the work they do. So this is something that we have deliberately put together, all of us together. We built it together, so we thank you very much for all your efforts. But you know, a machine needs to be driven. So this year, because I've been so busy, I wasn't on the ground most of the time. So we had our two deputy directors, Dr. Lydia Mosi and Dr. Peter Kwashi, who were driving the machine. Lydia was chair of the logistics committee, and Peter was the chair of the scientific committee, and both of them did a fantastic job, so let's appreciate them. Under the scientific committee, we had several faculty who helped review abstracts, staff who received the abstracts and indexed them nicely and packaged them for the faculty to review. So we want to thank the entire scientific committee for all their efforts sorting out all the abstracts. Thank you very much. We also had those who had to score the presentations. That's a tedious job. Listening to all the presentations, you can't miss any of them. Going through the posters and scoring all those posters. Some of you only looked at 2 or 3 posters. They had to look at 80 posters. So let's appreciate the scorers. No, normally they are anonymous because we don't want you to bribe them. So I will not be able to mention their names so that you don't go and bribe them for next year. All right, so thank you very much. And then when we come to the logistics side, several units under the logistics. We have, the grants management team and accounts people who make sure that we have money to do what we are doing. We have the comms team led by Andrew, who this year has really done an amazing job and, put together this brochure. This was all done in-house, okay? This brochure was all done in-house. Okay, we didn't have to contract anybody to design a brochure for that. This is done by our comms team in-house, so we really want to appreciate their talent and their hard work. Thank you very much. We want to thank our ICT team. Like I mentioned, I think this year we've had a much more, steady, presentation, and the projector hasn't gone off, and and the picture has been sharp. I'm really impressed by the picture quality. And then Chris and his people who did the live streaming and everything, we're very grateful for all your efforts and your sleepless nights, so thank you very much. I hear Sika and the others have taken the lead to go and organize the dinner, so in their absence, we still want to recognize them. Sika is such an amazing staff to have. I'm sure all of you have interacted with her. She's so efficient. This year she had to go on maternity leave and come back, but when she came back, she basically did enough work to cover for the period that she wasn't here. So I want to appreciate Sika for her amazing work. And everybody else for all your amazing contributions. Everybody is so important to the functioning of this enterprise, and we're grateful. All right. Before we do the prizes, we want to recognize our staff. Usually we give an award for the most outstanding staff for each year. So this year we're actually giving out 2 this year because people have really stood up in the midst of very challenging circumstances. So we really want to honor 2 of our staff for the efforts they've put in this year. In the midst of everybody putting in extra effort, some people put in extra super effort, and so we have to recognize them. So I want to recognize, first of all, our most outstanding next generation sequencing manager, Miss Joyce. Joyce, please come. So I'm going to call on Professor Kivgor. Please, can you come and do us the honors and present the Meritorious Award for Outstanding Commitment and Service to WACCBIP. missing? The money. Where's the envelope? I'm going to talk about the envelope. Congratulations. So when you hear about all the sequencing and all the variants and all that, this is the lady who does that. So she's been outstanding. Yeah? All right. And also, outside of the lab, our support staff, this year we had one outstanding staff who had to pick up the slack. Two of our key staff, head of accounting and then Sika, the grants manager, had to go on maternity leave at the same time. I think they coordinated their pregnancy. I don't know how to do it. But they went for maternity leave exactly at the same time. They were away for 3 months, and then they've come back and they're working half-time for the last few months also. And in that period, Marian Nano had to take additional responsibility to make sure that we didn't feel their absence. And she did an outstanding job at that, so we want to appreciate Marian. I don't think she's here. Yeah, she's not here. She's gone to get the dinner ready, but we want to appreciate her for her outstanding contribution in 2022. Well, 2021, 2022, okay? So each of them will get a handsome reward of $500 United States dollars. Okay. So, do we have the drums to roll? Now it's time for the awards. Okay. Okay. So, where's Peter? Peter, please come and tell them what you did. So that if there's a riot, you will be the one who will be... I don't want to be stoned.
Okay, before we go into that, I'd like to thank all the faculty members who took their time to grade the orals and grade especially the posters. Some did almost all of them. So let's give them a big round of applause. So what we did was we ensured that everyone had at least 3 people scoring. So that way we could try to get a good average amongst, and it was quite a wide and diverse and also competitive field. Yes, so we have, what we'll present is the... Should I present?
Just give the positions out.
Okay, so there will be best overall oral, best overall poster, and then normally we have best runner-up PhD, runner-up masters for oral, and then runner-up PhD, runner-up masters for poster. What we noticed, or I should say what I noticed, is that in the poster session, the PhD runner-up was so down the line, and the best poster, or the best runner-up, the runner-up poster was actually an intern. So what we decided was to have runner-up intern and runner-up MSc. So there's no runner-up for PhD in the poster session.
Thank you. All right. Thank you very much, Peter. Okay, so today I don't have the scores, so I can't tell how close people were. And normally I give the fractions of the percentage that they were close. So we start from poster session. So this is runner-up for poster presentation, master's category. Fatima. Bismarck, can you, Bismarck, Can you come and make the presentation for us? Bismarck Dinko. Bismarck Dinko. Okay. So this is the other runner-up for poster, and like Peter mentioned, this person is an intern, so that makes it even more impressive. An intern is somebody who has finished their bachelor's, they had to start their master's. Okay, so she went into competition with master's students and PhD students and held her own. Ethel Deborah. She's not here. She's still at her poster. Oh, okay. Jack Santa, can you please make the presentation for us? Jack. Good. All right. So then we go to the overall best poster presentation. This goes to Claudia. Bill, can you come and make the presentation? Bill, yes? Please, this way, yeah, so you can. All right, then we go to the oral. First of the two runners-up, this is for master's category. Nelson Edu. Lydia. All right. The other runner-up, this PhD category, Elvis Chumesi Abaji. All right. So now the coveted prize, overall best oral presentation. Any drums? Drum roll, please. Godwin Insult. Keep going. Professor.
Wow. Congratulations. The big ball. Well done.
Okay. So I just realized that I was supposed to sign the certificates and I didn't sign them.
No,
So please make sure you bring your certificate. I'll sign it for you. I'll autograph it for you. Thank you. So usually the winners get to go to an international conference fully paid by WACCBIP. So look for a conference of your choice, and we would pay for you to go and present your work there over the next 1 year. So thank you very much. All right. So I think my work is done. Your work is done. Our work is done. WACCBIP Research Conference 2022 over.
I can't top that now, can I? I just have housekeeping. So those of you who received a second ticket when you registered this morning, there'll be buses in the front of the building to convey you to eat and feed. So I think that's pretty much all I have to say. And thank you all so much for a wonderful experience. Thank you.
Established in 2002 in Pretoria, South Africa, Inqaba Biotech is a rare and unique company, as suggested by the name, and we thrive to give reliable and efficient services to all our customers. How it all began. With the concepts developed in the year 2000 to speed up the arrival of primers in molecular labs in the country, Dr. Oliver Pricer, co-founder and executive director, together with a venture team of 5, secured a loan from Swiss government and got rights to be the very first company to synthesize primers locally, and thus it all began. With capacity to synthesize over 1,000 primers a day, Inqaba remains the
Recording stopped.